पहले तो ओके स्विच कर
check, 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 check.
Sure. Good morning, ladies and gentlemen. Welcome to the day 2 of Agri Vision 2024. Dr. Channa S. Prakash, Dean, College of Arts and Science, Tuskegee University, Alabama. He is already there online. His talk is over the video conferencing, so we'll start that in just 5 more minutes. Sure, sure. Sure. Dr. Channa S. Prakash, Dean, College of Arts and Science, Tuskegee University, is already there on the Zoom call. Can we connect with Dr. Channa, please?
Yes.
The first session.
Can I share the screen?
Yes, so we are looking into it.
Thank you.
So your screen is visible now.
Okay, can I go ahead and start?
Yes, sir, you can.
Thank you so much. Good morning, everyone. I'm really honored and delighted to be invited by AgriVision to share my thoughts on a very, very transformative technique that is literally literally taking over the plant breeding and plant genetics crop improvement around the world, including India. It's a new technique called genome editing. So I just wanted to share with you what are some of the applications of this technique in Indian agriculture, giving some examples on how it could be useful in specific instances, and also talk about how we can, as scientists, how we can contribute to greater understanding of this technology and hopefully its acceptance. So genome editing, to place it in context, we must understand that we've been changing our food crops for almost 10,000 years. Ever since we walked out of the caves and we started beginning agriculture in many parts of the world, we literally took all the weeds, the wild plants that were growing around us, and slowly through selection, a slow process of selection, we domesticated them and improved all our crop plants, all our wild relatives of crop plants into crops. Just to give you an example, on the top panel, you see this is how the banana looks like in the wild. And you can see how the banana looks now. And a more dramatic example on the right here, I show you how a maize or corn looked before its domestication. Its wild relative teosinte looked like a pencil, and we all know what corn looks like now. And so this process of change has been happening all the time since the dawn of agriculture. But more recently, using scientific plant breeding methods in the past 100 years. We have practiced several methods of breeding, including mutagenesis, hybridization, and that is a very important reason why we have been able to feed this humanity. Even though our world population has developed so much, we've been able to do a very good job in feeding that through scientific agriculture, and plant breeding through many of these techniques has been very important. The transgenic breeding or the use of GMOs was developed in the early 80s, and the first GM crops were released in 1996 in the US and 2003 in India, and they have been now been practiced. The GM crops have been grown for more than 20 years. The genome editing, which is essentially invented about 10, 15 years ago, involves it essentially represents a continuum of all the techniques that we have been using. And so the GM crops, the BT cotton is one of it is the only crop that is grown here in India. And you can see if on a cotton crop that is unsprayed, that you see on the right, you can see a complete 100% destruction. This is an extreme example, but it's not uncommon to see a cotton crop that is completely devoured by bollworm. But on the left, you see the GM BT cotton, how it protects. And again, a similar example I show you with the corn against the fall armyworm. And so compared to the GM crops, which involves an introduction of foreign gene editing represents a much finer approach, and it involves much smaller changes, and this is very precise, very targeted. And if you are following the first two techniques, the SDN1, SDN2, it does not introduce any foreign genes in it, and so this gene editing is very similar to the classical mutagenesis. And in many countries, including India, as long as no foreign genes are introduced, genome-edited crops or gene-edited crops go through much less regulation. In other words, they're not regulated as GMOs. And so this is one of the biggest attractions for many of us, especially those of us working in the universities, because we do not have to go through very high burden of regulation. And in fact, in many countries, even GM crops are not even allowed, especially in many European countries, as you know. And India has not approved any GM crop other than cotton. But on the other hand, there is the genome-edited crops are very attractive because this can be developed very faster, and there is a cost of development is also much less. And most importantly, public acceptance and regulations, there is much ease compared to the GM crops. So what can genome editing do? And we can alter many traits. One of the most important traits that are being altered through genome editing is introduction of disease resistance. Here you can see tomato, a control tomato on the top with powdery mildew, and just genome edited crops. You can see there's 100% resistance to this very important disease of tomato. Similarly, just this week, there was a publication coming from Africa where they were able to take a banana and just knock one important gene out through genome editing, and they were able to develop a banana that is resistant to a very important wilt that is an important, very destructive disease across Africa for banana. And similarly, a citrus resistance to canker has been developed, and this is a bacterial speck resistance in tomato, and you can see how damaging this disease could be on the left panel. In a wild type, in a gene-edited tomato, you can see very high resistance. And again, we can also improve quality traits, and this is one where a banana that is non-browning and slow ripening has been developed through gene editing. And moving forward, rice is an important crop. A lot of work, including India, is going on genome editing. And this is one example where a deadly blast disease, where scientists were able to develop a resistance to that through few alteration of few nucleotides in the rice genome. And there's another disease which is also very rampant in India. It is a bacterial blight disease of rice. And again, you can see there has been a resistance to this developed very early on. And another very important work on gene editing came from Indian Institute of Rice Research at Hyderabad, is where that they were able to take the Samba Masuri, a very important rice variety, as you know, and they were able to increase yield by about 20–30% in this particular rice variety, which is again a very, very encouraging research coming from India on a crop of very high importance to not only India, but across Asia. And it's not just qualitative traits like disease resistance that could be increased. One could also work on quantitative traits, which are polygenic in nature, and genome editing of multiple genes can increase the size of the grain here in wheat, as it is shown. And editing 6 genes improves the size even further. And one other very important trait that could be altered is the plant architecture. As you know, the Green Revolution was primarily achieved by transformation of the height of both wheat and rice by development of dwarf varieties of wheat in Simet, and later in Erie and other places, dwarf varieties of rice helped achieve Green Revolution. And this is again to show by genome editing, we could do the same where we can alter the plant architecture, as you can see here in the case of a cherry plant, a relative of tomato. And one other trait that could be improved is also the nutrient efficiency. We know in India, one of the negative effects of Green Revolution has been the very rampant and pervasive use of fertilizer, leading to many ecological problems, including soil degradation and loss of soil fertility. And 80% of the fertilizer we use on our crop plants gets washed off and in runoff. And so if we increase the nitrogen, phosphorus, and potassium use efficiency, we can cut down the amount of fertilizer that we use. In that way, by making the plants more efficient in the uptake and utilization of nutrients. And there are again many very examples of that in the literature in the past few years. And moving forward in the quality traits, and about 50% of the fruits and vegetables grown in India are thrown away because they perish because of the lack of storage, lack of marketing, and lack of processing in many of our rural areas. But if we can delay the ripening of many of our of vegetables and fruits, including tomato that is shown here, we can conserve, improve, and help improve the marketability of these crops. We can keep it for a longer time. A potato which ripens and gets perished in about 2 weeks, we can extend that window to about 3 or 4 weeks. Moving forward, scientists have also developed golden rice and golden bananas with improved vitamins using gene editing. And as you can imagine, this is going to be a very dramatic biofortification, addressing a very serious challenges that we face in many developing countries, increasing lycopene content in tomato, and knowing diabetes is a very big important problem, especially in India. And Indians, we consume a lot of rice, and gene editing also has potential to develop low glycemic index rice. And this is just again one example that I'm showing here. And this is a potato that where a browning has been reduced, and much of potato, again, once it is cut, it accumulates phenolics very quickly and browns and needs to be thrown away. And this is one example where we can conserve and improve the marketability. Genome editing has also been used to develop better beer. Who doesn't like a better beer? And this is an example from Australia in barley. And finally, one of the most important challenge facing humanity today is a global climate change. It's due to a variety of reasons. And the most important negative effect of global climate change is on agriculture because of the increased disease, lower yields, and lower nutritive quality. And again, here, genome editing has shown value that we can develop crop plants that can tolerate high salt conditions, for instance. And but there is even a company that is trying to see if we can grow rice. This is an Israeli company, if we can grow rice in seawater. And so these are just some of the examples that I'm showing how we can develop crops against global climate change. This is a Arabidopsis with very deep root system that is developed. So it can be a very valuable trait for drought tolerance. If it could be done in many crop plants. And this is a high temperature tolerance gene editing that is done in tomato here. And we know the temperature increase is common, and we have seen in the past 10 years how this is our temperatures all across India has increased. And one of the most important effect it's going to be is already in agriculture, we can see. So we have a potential here, a tool that would help improve, confer our crop plants to be more resilient to all these parts of climate change, including flooding that is seen here in a plant, a rice plant from Odisha is known to have the greatest flooding tolerance of any rice, and those trait can be easily, by studying the trait, we can edit the same gene in many of the other rice varieties and make them also tolerant to flooding. And finally, we know this is the year of millets, and the Prime Minister Modi has been very gung-ho and advocating for millets, rightfully, because they are very hardy, very nutritious, but they are not necessarily highly productive and also suffer from susceptibility to diseases and pests. And by taking many of our hardy millet varieties, we can make them productive very quickly and bring about a greater improvement in the resilience of Indian agriculture and also contribute to the nutrition. So genome editing has, as I have shown, can increase the crop productivity. It can increase the nutrient levels of our crops. It can make our crops more resilient to climate. It can build in a greater disease and pest resistance. And it can also bring about an element of food safety, improve the food quality, and also eliminate some of the undesirable effects, elements in our food, such as the neurotoxins that we see in kesar dal, and then the cyanide that we see in sorghum and cassava. And so, as I said, many countries, and those countries that are in the green here, they regulate gene-edited crops as almost a conventional new varieties of plant breeding with very little of regulation. And so we are very fortunate that India has moved forward in a very science-based fashion to regulate or deregulate GMOs. And so if we foster productive, increase research, increase funding for genome editing, I think India can benefit. But we also need to ensure that as scientists, we need to communicate as to what this technique is. This is essentially an advanced mutagenesis technique. We have been using radiation and chemicals for inducing mutations for over 100 years now in a very safe manner. We have used all kinds of other techniques of plant breeding. So this is just one other more science-based, more knowledge-based, but more precise way to develop plants. And we need to make sure that we provide evidence on the safety and address any specific concerns and acknowledge any ethical concerns that we may have, that if people are uncomfortable with the use of modern techniques. But most importantly, we need to highlight the benefits of this technique, like I have done now, and bring about a greater dialogue amongst the various stakeholders. Be very transparent about any risks that the technique may have. So far, there is none. but clearly delineate the benefits and work collaboratively with all the stakeholders, especially the farming community, the media, and to the scientific community in a way to help address to bring about good policy changes in the acceptance of this. And this I'll be sharing my slides with the organizers, and so please feel free to share with them, share with the audience the whole slides, and I'll be very happy to take any questions if there is. But I do thank you for listening to my presentation. Thank you very much.
Sure. Thank you so much, sir. Do we have any questions for him? Any questions? Yes, sir. So we have one question. Please be there to answer it.
Thank you, sir. Regards, sir. Mostly, sir, it was a nice presentation and also a good milestone that gene editing will offer to go for also disease resistance. Particularly, I am plant pathologist and it will take care against blast disease, other disease also. Sir, another thing, till now, research has been done vividly by different rice researchers, plant pathologists, and also breeders throughout the country and also world. But till now, no resistant genotype has been developed against sheath blight disease in rice caused by Rhizoctonia solani cone. So, sir, whether this gene editing will achieve in that aspect. So, if he by gene editing will develop the sheath blight resistant variety, then it will be a good milestone. Second question, sir, whether gene editing or a development of a gene edited crops, whether it will have any harmful effect on human body after consuming that crop grains? It's my two questions, sir. Thank you, sir.
Well, thank you very much, sir. I appreciate your question. I did not specifically get the name of the disease that you talked about, but I'm not a rice. I have worked in plant pathology for both for my master's and PhD. And so I have some good understanding of many of the diseases and disease resistance breeding. So I am pretty sure that the specific disease that you mentioned, although I'm not familiar with it, one could develop resistance to that. It requires basic research and understanding of the disease resistance, and it requires a considerable genomic knowledge of that particular resistance to the gene. Once that knowledge is developed, then I am very confident through gene editing one could develop resistance to that particular disease. And secondly, in terms of the harmful effects, you see, a particular plant will have about 3 billion nucleotides, as you know, and through gene editing, we are just changing literally 1 or 2 nucleotides in that. And so the level of change is so small, we do not expect any harmful effects. But nevertheless, if there are some pleiotropic effects of where, for instance, we know there is one classic case of developing a powdery mildew resistance in barley led to the susceptibility to one other disease. So things like that might happen. And so we must follow the regular plant breeding testing, and whatever means we develop the genotype or the germplasm. And so even in gene editing, all the plant breeding protocols must be followed to make sure that there are no negative traits associated with any development of the positive trait through gene editing. And so far, all the results have been very good. And when you breed your rice, when you cross it with a wild rice, or when you are breeding tomato for resistance to nematode, and then you cross it with wild tomatoes, you literally disrupt the whole genome. You disrupt the probably thousands of genes are changed, right? The alleles. But in contrast, gene editing requires a very minor, minuscule change, and so we do not expect any harmful effects in most of the time, according to science. Yeah. Thank you.
Sir, we have also advanced with respect to breeding aspect in our National Rice Research Institute with taking the help of rice breeders, and we plant pathologists are proceeding to go for a QTL mapping with the background of most susceptible Sitholite variety Tapasini and Sorna Sawan, so that we are thinking that we in future will get a Sitholite resistant variety. And also, sir, biotechnologists are there. We are so taking their help also. Sir, thank you for a nice presentation, and also
Thank you. And I wish you good luck with your breeding.
thank you, sir. Thank you.
Any more questions? Yes. So there would be one last question for you.
Sure.
Good morning, sir.
Good morning.
I was fascinated by your report, and I would like to ask some questions like, was there any of those genome edited crops that you mentioned already produced largely? And the second question is,
Sorry, can you repeat that again? The first question again?
okay, so is there any of those genome edited crops you mentioned earlier are being produced widely now? Second is that, is there any incidents that those edited genome become resistant to disease but susceptible to pests, to insects?
Very good question. Thank you very much. Yeah, this is a very new technique. While literally you will see thousands of research papers have been published just in the last couple of years on this. The commercialization of this is just beginning. We have one variety, a nutritionally improved variety of tomato that has been released for public in Japan, and also an herbicide tolerant varieties of soybean that have been released in Canada. And so, but in the next but when we look at into the USDA approval process and the application process, there is literally dozens of genome edited crops that are going to be released for commercial cultivation in the next couple of years. That is my further answer to your first question. And again, second question, if it's if it develops a resistance to a disease, can it develop resistance to a particular pest? That can only happen if that particular gene has a pleiotropic effect. You see, if and then we do know occasionally there are genes like that where when you bring about a positive change, there may be an associated negative change. But in those cases, we would not be using those. We will not be editing such genes because we do not want to bring any new complication. So whether you use gene editing, whether you use hybridization or mutagenesis, you need to perform extensive testing of your plants and your populations through regular breeding procedure. And when we do that, if there are any such susceptibility, we are going to detect it. So far, there has not been any such evidence in the literature so far.
Thank you so much.
You're welcome.
So that's all. Thank you so much, sir, for your wonderful talk.
Thank you.
So now we'll move ahead with the next one. We'll connect with Dr. M. Nageshwar Rao, genetist and ornamental project lead, USDA ARS, Miami, USA. If he's there online, we are going to hear his talk again virtually. We are waiting for him to join. Please wait for a while. Check. Okay, so Dr. M. Nageshwar Rao has not joined the link, so probably he is not available. We are moving with the next presentation from Professor Johnson Adesoda, Ekiti State Polytechnic, Ishan Ekiti, Nigeria. He'll be talking on the topic carbon footprint and aggregation of an alphisol in response to different tillage and maize. Sir, can we please have you on stage? Give him a big round of applause, ladies and gentlemen.
Good morning, everybody. Good morning. My name is Professor Johnson Adesodun. Good morning, everybody. My name is Professor Johnson Adesodun from Federal University of Agriculture, Abeokuta, Nigeria, and presently from Ekiti State Polytechnic, Ishan Ekiti, Nigeria. I'm the current rector of the Ekiti State Polytechnic. I bring you greetings from Africa and particularly from Nigeria. I'll be speaking on the research topic, carbon fraction and aggregation of an alphisol in response to different tillage and maize cowpea intercrop. Well, agricultural management practices involving land preparation, sowing, plant biomass, and fertilizer application have the potential to contribute to anthropogenic greenhouse gas emission to the atmosphere. And therefore, the development of a low carbon agricultural system to store carbon in soil is a measure of that requires global attention. The strategy in reducing greenhouse gas emission from agricultural soils is to adopt improved farming practices such as conservation tillage and intercropping in crop production. However, tillage, while practicing tillage, we should take into consideration the effect on soil properties, particularly with regards to soil aggregation, which involve the binding of soil particles and into secondary units. Sorry, I forgot to mention that I'm a soil physicist by training. Carbon footprints, on the other hand, is a product, is sum of greenhouse gas emission and removal in the production system, which is expressed as carbon equivalent and based on the life cycle assessment on climate change. That is like intercrop I'm going to talk about, and tillage, that is from the production, from the planting, use of tillage to harvest. Simply put, carbon fraction is a calculated value or index that indicates the total amount of greenhouse gases that's an activity out to the atmosphere, usually reported in tons of emission per unit of the activity. Now, the objective of this study, is to evaluate improvement in soil aggregation and short-term carbon fraction in crop production system to assess the impact on climate change. The methodology, that's materials and methods. This experiment was conducted in the planting season of the year 2018 at Federal University of Agriculture, Cuttack, Nigeria. The soil is well drained, sandy loam with gravelly sandy clay loam subsurface. The experiment had tillage as main plots, cropping system that is sow maize, sow cowpea, an intercrop of maize and cowpea as subplots, an amendment that is NPK fertilizer 15-15-15, and urea, and organomineral fertilizer that is a chemical fertilizer mixed with poultry manure as amendments as subplots. So for maize, the variety of maize used was Oba Super 2 hybrid, and the cowpea was IT 10K plus 8364. These planting materials were sourced from International Institute for Tropical Agriculture in Ibadan, Nigeria. Soil aggregation was measured by the standard West Steven techniques in sieve sizes of 5.6 down to 0.25 mm, that is the micro aggregate fraction. The soil retained in each sieve were oven dried and weighed to determine the percent water stable aggregates and the mean weight diameter. The mean weight diameter was used to measure the stability of the soil, while the water stable aggregate was used to assess the distribution within the aggregate fractions. Total organic carbon was determined by the standard by the standard acid dichromic oxidation method of working on black as adopted by Nelson and Sommer, 1996. Total nitrogen was determined by Keja analytical method as described by Brenner and Morfessi, 1982. The climate change impact as influenced by the management practices, that is the tillage, the cropping system, and the amendment was evaluated by carbon sequestered below the ground, and then direct emission of the greenhouse gases, and in the whole life cycle of the crop, the tested crop, that is maize and cowpea, and carbon footprints as presented below. That is the sequestration in this study was determined from the difference between the total elemental carbon minus the control plots. And the carbon stocks was calculated using the concentration of the carbon multiplied by the bulk density. Total greenhouse gas emission in carbon equivalent through the materials added, that is the chemical fertilizer and organomineral fertilizer, and from tillage operation in a single life cycle were assessed as presented by Eli et al. 2019. that which shows the summation from the equation 2, we can see that AI denotes the amount of all agricultural inputs in kilogram per hectare, and AF is emission factor as a unit of materials. Then the total greenhouse emission was determined as the sum of the input factors. Okay, like I mentioned, chemical fertilizer, the organomineral fertilizer, and the crops. This was done using, as described by Hannah et al. 2015, where the direct emission of nitrous oxide as presented in kilogram carbon equivalent per hectare in this related to the inputs were also determined as shown in equation 3. That is, the N applied in these studies. What was applied is the standard for Nigeria, which is a 60 kilogram N, 60 kilogram nitrogen per hectare, and then minus total nitrogen as measured from soil following the chemical analysis, and then multiplied by the molecular weights of nitrogen gas and 298, which represent the global warming potential as by IPCC 2006, and also by the molecular weight of CO2. Sorry for that presentation. I'm a physicist, and just pardon my ignorance there. Also, it was shown, as was shown by Ma et al. And Inlet 2009, that 75% of total carbon footprint from crop production results from nitrogen input. The nitrogen input contributes that is from the fertilizer, contribute to CO2 emission. In these studies, we use urea as nitrogen fertilizer, and then the emission of the CO2 from urea-based fertilizer were calculated according to March 2012, as shown in equation 4. Now, total carbon fraction, that is kilogram carbon equivalent per hectare of crop in a single season, that is cowpea and maize, were then expressed as a summation of all of the individual, from the individual plots, that is from the life cycle of maize and cowpea production system. Therefore, carbon fraction now is total gas, greenhouse gas emission in kilogram carbon dioxide per grain, because the whole life cycle, the endpoint there is the grain yield, which we use in these studies. Grain yield data is not presented in this, it's not shown in this presentation. Now I go to results. Oh, this is clear? Yes. Like I mentioned earlier, the physicochemical property of the soil, of the site and organomineral fertilizer. The soil is a sandy loam surface, and the sandy clay loam subsurface very gravelly. And other parameters are shown. The analytical composition of the organomineral fertilizer is as shown in this table 1. Yes, from this table, table 2, which shows the soil aggregation, soil aggregate distribution, and midway diameter relative to the treatment. It shows from, you can see from here that in NT, which is a non-tillage, non-tillage or zero tillage plots, with the amendment, organomineral fertilizer in maize plots gave the highest mean weight diameter. That is improvement in the aggregation. The stability of aggregate was higher in under no tillage in soil amended with organomineral fertilizer planted with maize. But generally, you can see that these amendments, these treatments, the tillage and the amendments and the cropping system improve the generally improve the micro aggregate fraction, which is the range of 5.66 to 0.25 than the microaggregate fraction. But generally, if you look below here, under conventional tillage, you will see that the soil were disaggregated more with under conventional tillage. Conventional tillage here involved plowing and harrowing, while minimum tillage was just plowing alone, and then the crop were planted. Table 3, that is the total organic carbon and total nitrogen and sequester carbon and nitrogen is shown in table 3. From the studies, we can see that tillage, that is no tillage or zero tillage, had more total organic carbon and total nitrogen concentration, and sequester more carbon and nitrogen than other tillage practices. Okay, now coming to the amendment, application of chemical fertilizer, which is NPK, was able to store or sequester more nitrogen than other amendments. Generally, for the sequester carbon and nitrogen, the negative value here indicates release of CO2 into the atmosphere, while positive value indicates carbon and nitrogen being stored within the soil. So there was significant sequestration of carbon and nitrogen in no tillage plot than other tillage system. Likewise, for cropping system, plots planted with sow maize were able to sequester more nitrogen than other cropping system, while cowpea and intercrop of cowpea and maize alone were able to release more carbon into the atmosphere compared with other cropping system. For specific greenhouse gases emission relative to the treatment, that is carbon dioxide emission and nitrous oxide emission, we can see from effect of tillage that conventional tillage was able to release more carbon dioxide and nitrous oxide into the atmosphere compared with no tillage and minimum tillage. These observed differences were very significant and 0.5 probability level. With regards to the amendments, the control there supposed to be not applicable because the chemical fertilizer, that is NPK and urea, in the studies, what happened at the 4th week, NPK 15-15 were applied, and urea was also applied at 6 weeks. You can see that amendments with chemical fertilizer only was able to emit more carbon dioxide into the atmosphere and nitrous oxide compared with organomineral fertilizer, which was least. Okay, the effect of cropping system showed that intercrop of cowpea and maize were able to emit more carbon dioxide and nitrous oxide into the atmosphere compared with maize and cowpea alone. Table 5 shows carbon fraction related to the treatment. In these studies, well, generally, the carbon fraction lower value indicates less release into the atmosphere, whereas higher value indicate more release to the atmosphere. So from these studies, you can see that zero tillage was able to mitigate against carbon dioxide release to the atmosphere compared with minimum tillage and conventional tillage. Also, with regards to the amendments, organomineral fertilizer was able to mitigate against greenhouse gas release to the atmosphere compared with chemical fertilizer. With regards to cropping system, maize alone, that is sow maize, was able to release less greenhouse gases into the atmosphere. That is, plus planted with maize was able to release less greenhouse gases into the atmosphere. So the summary of this work, this study showed that the stability of the soil was better improved with no tillage than conventional tillage and minimum tillage. Application of organomineral fertilizer improved the soil stability than application of chemical fertilizers, while planting of sow maize aggregated the soil than sow cowpea or intercrop of maize and cowpea. Results also showed that greenhouse gases was lowest in no tillage system than other tillage system. Also, application of chemical fertilizer significantly increased the total greenhouse gas emission than organomineral fertilizer, while carbon fraction was found to be least under no tillage system. Planting of sow cowpea led to higher carbon fraction than sow maize and intercrop of maize and cowpea. Finally, for these fragile tropical soils, it was concluded that no tillage and minimum tillage with use of organomineral fertilizer are better options for maize and cowpea production with less carbon fraction and consequent reduction of greenhouse gases into the atmosphere. These are some of my selected references. Thank you for listening. Thank you. Any question?
Any questions for him?
Really fascinating presentation. I enjoyed it. I guess two questions. One, do you have any data on or any thoughts on what's going to happen with the fractions of phosphorus and other micronutrients under the tillage system?
Can you come again?
Do you have any thoughts on what's happening to the fraction of phosphorus or other micronutrients in the no-tillage system?
No, sorry.
And do you think there are significant changes in the soil microbiome, the bacteria, the eukaryotic microorganisms that are important?
Yeah, it's very important, but we didn't go into that area in these studies.
Okay.
Yeah, we didn't. We just limited to assessing the emission of the greenhouse gases and looking at the stability of the soil.
Okay.
Well,
Maybe one last question.
Yeah.
So you're showing significant increase in the nitrogen storage. Do you think that could indicate the next growing season you could add less nitrogen as a phosphorus or as a fertilizer?
Yeah, sure.
Okay.
Yeah.
Very nice.
Thank you.
Thank you so much. Any other questions? Thank you, sir.
Thank you very much.
We will now have a coffee break for 15 minutes. We resume the session sharp at 11.
और ये डीप नहीं है, मतलब सॉलिता जिसको बोलते हैं। और ये का है, क्या बोलते हैं, उसके ऊपर कपूर डालना। कपूर नहीं डालेगा तो जलेगा नहीं। आ रही है, कुछ आ रहा है। थोड़े वाला
Hello, hello, hello. Hello, check. Check, check, check.
Check.
Hello, hello. Check, check, check. Check 1, 2, 3. Hello. Volume थोड़ा इसका दो. Hello, check. So is Dr. Dev Mishra over here? Dr. Dev Mishra?
Yes, I'm
Ye-
here.
Yes. Thank you so much, sir. So we actually have the meeting session right after this. So we will hear from Dr. Dev Mishra first. He is from Bayer Bioscience Private Limited. We are going to have a virtual session with him. Pathogen informed disease management strategies in crop for sustainable farming. He is talking on the same topic. Let's hear it live on screen.
Yeah, so I'm just sharing my screen. Is it visible?
Yes, sir.
Oh, so thanks. So I'll just go through and talk on the very important topic that is typically on the disease management and sustainable farming. So as we all know that disease is the outcome of interaction between pathogen, host, and environment. So I will be taking the inputs and the information what we had generated here in Asia, specifically focused on the pathogen. So how the information linked with pathogen can help to devise these strategies for the disease management. And as we know that the mega trends in agriculture that is driving the modern agriculture, we see a lot of challenges there. And one of the big cause of those challenges is the population. And what we are seeing by 2050, we are going to add more than 2 billion population again, and that will require more than 50% food that is being produced now. So this is also aggravated because of a lot of loss is being happening, what is being produced because of the climate change. And also the arable land is decreasing day by because of the urbanization and all those. So it is putting a lot of pressure on the agro ecosystem. And that's where we need to use the natural resources very efficiently and responsibly. Over and above, the increasing pressure of the diseases and insects and beasts because of this climate change is also becoming enormous. And that's where the innovation in agriculture is going to really take this forward, where we need to really grow more on the small available arable land. And this is one of the example. If you see, by in 1940, to produce 250 kilogram of yield, yield of corn, so it was required to have 1347 square meter area. But with the innovation in the agriculture and a lot of technologies, as we were hearing in the morning from Dr. Channa Prakash also, so that much amount of this yield we are able to get now in 167 square meter area, if you see in this screen, in 2019. And in future, with the advances in the technologies like the genomics, genome editing, and all those, so this area we can further reduce. Like in 120 meter square area, we can produce the amount of corn, for example, that was produced in 1940s in 1347 million square meter area. So that is possible because of the innovation. But the important topic here today is on the crop health, and we see that a lot of diseases that is becoming and appearing every year, they are posing a lot of risk on the humanity, because crop is being lost. And some example here in banana, fusarium wilt. Then in blast recently, that was got introduced in Asia, for example, in Bangladesh. And last year we had seen in 2022 a new disease that was appeared in rice, specifically in North India. That was a viral disease for the first time. So these are the diseases that keep on coming and devastating a lot of crop, and that's where it becomes very important to address these diseases. But what is important when we talk on the disease? So as I told in the beginning, so three major components: the host, pathogen, and environment, we need to understand. And pathogen becomes more important because they keep on changing with the kind of host genetics and the change in climate conditions. So that is very important. And what we see, whatever resistance we are deploying as of now, they are not going to be the important one going forward, because they are going to be broken. So in that scenario, the basic understanding for the sustainable agriculture in terms of the disease management is whether we can monitor the pathogen shift very early and correctly, so that we can help the farming community to address the diseases in very accurately manner, so that they can save the yield loss. And second is also the strengthening the plant immune system. So we'll just go and take some example from rice, because rice is major crop in Asia, and that is a kind of crop that is helping humanity in terms of providing the sustainable food system for almost half of the world population. So, and from plant health perspective in rice, so you see the diseases, insects, and weeds, they cause a lot of damage on the crop. And if we focus on the major diseases here, so you will see the bacterial leaf blight, that is important one, rice blast and brown flower hopper insect, specifically in this region. So we'll take the example of bacterial blight disease, and what we have estimated around 30 million acre area is affected annually in the region, and it causes a lot of damage to the crop. From looking from the topic, specifically from the pathogen side, so we will go more deeper. But if you see the disease per se, so it can cause, means it can come at the flowering stage, and it can be much to harvest. So it is important because the pathogen of this disease, that is bacteria Xanthomonas oryzae, Pathobacter oryzae, is having a lot of variability. And if you see on this slide, so we could identify 12 major pathotype out of 1410 isolate that we have characterized as of now. and none of the single genetics, what we see here, XA1 to XA23 is able to protect at all. So that's so that pathogen variation is very important. And that's why its monitoring becomes very important to save the crop. And also, if you see the distribution pattern, so they are widely distributed. Like, for example, all these races, 12 races, so they have very wide distribution across all the regions in India. Some of them, they are having high population, like third pathotype is having around 38-39% population structure, but still there are others that is present in all other geography. And the kind of risk what is seen, like as of now, some of the major genetics that has been deployed. So when we search that, what is the spectrum of their effectiveness and what are the percentage of pathogen isolate that can break it. So we found that XA21, the major gene that is being deployed by public and private sector. So there are already around 14% pathogen that can break it. So the major question is if we can really detect them quite early in the cropping stage, then that becomes easy to manage them in the field itself. So how we want to advance those technologies to detect the pathogen very accurately and diagnose. So as of now, it is more on the manual, like sample collection and then doing a screening in control environment or in control field condition. But that is not a kind of high throughput. So we need to go ahead from manual to more automated and predictive way, utilizing the genomic knowledge and the genomic understanding. And that is the major shift we are talking about in the pathogen understanding. And that's where we started collaborating with different institutes across globe. Like, for example, in India, CCMB and IMTech. So we just went ahead and did the genome sequencing of 100 isolate to understand that how their genomic architecture looks like. And based on those genomic architecture, we were able to pinpoint what are the major changes in the genome structure that define and decide new race development. So if you see on this slide, so based on those genomic architecture, we were able to identify the major change in the pathogen genome, which is making that particular race to cause disease on XA21, for example. So it was like any isolate, if they are liking this RAXX gene or they are mutated at that locus, so they are able to break the XA21 resistant gene that is deployed. So the whole point is that if we are able to identify these and we are able to pinpoint these regions in the genome, so we are also able to make some assay so that we can start detecting that kind of race directly from the field sample. And that is the direction what we are taking. Not only to that resistant gene, we have also tried to look some of other major resistant gene in bacterial blight like XA5 and XA13. Because now we are deploying the genes in combination also. So these three resistant genes like XA5, 13, and 21, they are the major one. And we were able to identify the genomic region for the effectors in the pathogen, which are helping them to really break these resistance genetics. So like, for example, if you see these two effectors like PTH-XA1 and XA4, XA7, if they are present, then these isolates, they are the virulent on the XA5, means they can break XA5. And same way, if PTH, XO2 and 3, if they are present in any pathogen isolate, they can break XA13. So that's where we are taking all these examples into consideration to devise some of the technologies going forward so that we can start detecting the pathogen directly from the rice tissues, for example, in the field. And now, since the genomic technologies, they are very advanced, and we see that it is possible in the field itself, like some of these sequencers, nanopore sequencing system, that is now available. We can take that machine, it's very small like our phone, and we can take this in the field and we can just take the sample and cross it, and then we can just do that, the sequencing itself. So that way it becomes very efficient and also important. We can avoid a lot of process during the pathogen isolation and screening. So we can just go in field and see that if any field is affected, so what is the race, whether it can break the genetics that we are deploying or not. So that way it becomes very easy. So this is the approach that we are following, and that we are going ahead to monitor the pathogen quite early in the field. And it is not limited to the one disease, like this is the disease that we have made a lot of progress, but we are also trying to address with other diseases also. So rice blast, we have seen that it is having a lot of variation in the pathogen. So that's why we need to understand this also. So similar approach we are taking in this one also. Even in other crops like in corn, so like for example stock rot complex, so that is also a major disease in the region where multiple pathogen is involved. So it is really important to understand what is the pathogen that is able to cause the disease, and how accurately you are able to diagnose and recommend the kind of important genetics that can be deployed in those geography that would be effective against the dominant fungus, pathogen, or species or race. Same way, we are working on another disease in corn, that is downy mildew, and that is important specifically in Philippines and Indonesia. And that disease is caused by 3 species of Sclerospora, that is Medes philippiensis and Sorghai. But we need to really understand what is the dominance of these species in different regions in Asia, so that the genetics that we are going to deploy, it should be correctly deployed in those important regions so that it can provide the required level of tolerance. Otherwise, if genetics is resistant to one species and susceptible to other, then it will not be effective and cannot save the crop. So that is how we are looking, and that is how the whole scientific community is trying to address the pathogen risk, means detecting them in proper time, predicting the pathogen race evolution, and also going forward modeling that the risk. So that is the way forward. But that information is also utilized to strengthen the plant immune system, like the kind of varieties and the products, hybrids they are being produced. So they are having good level of tolerance or resistance against those evolving pathogens. And the example what I talked on the pathogen, so based on the pathogen information, the resistant genetics that we are trying and we have deployed, so that is working well. For example, here you will see based on all these pathogen information, we looked at what are the genetics that is effective against all those 1500 isolate. And you will see here in green color that is showing the effective genetics like 13, 23, 21. So some of those genetics have been deployed and they are in the region, and you can see the kind of effect they are providing. If resistance is there, so they are green. If there is no resistance, then they are destroyed by the disease. And you can see the yield loss, for example, if there is no resistance, 1.7 ton, and if we have resistance, 8.3 ton. So that way, it is helping a lot, and we are putting the right genetics in right geography based on the pathogen understanding. And that's where it is making all those genetics more sustainable and providing a lot of relief to farming community so that they can do the farming in more sustainable way by managing the disease. One example is the way it is helping the farming community. I will take this example of one trait, the bacterial blight. So we worked on and we launched first product in 2008. I'm just talking about the product that Bayer is putting in the market, means that is being grown by the farmer. So that product only is being grown in now around 4 million acre area and saving a lot of means the loss for the farmers. And also, as we all know that for bacterial and viral diseases, there is really very difficult. They are very difficult to control. We don't have a good chemistry. And for example, in bacterial case, a lot of antibiotics are used to manage, but that is not good option because that antibiotics, they go into the food chain system, and we as human being and our animals, they are becoming resistant to antibiotics as well. So that is not good practice. So that way they are really helping a lot, and they are means farmers, they are saving a lot on growing these resistant cultivars. So this is not only the Bayer or other private companies. A lot of public sector institutes, they are also coming with a lot of varieties that is resistant to these diseases. Like for example, NRRI, then IIRR, IRI, PAU. So all those institutes and universities, they are bringing a lot of product in the farming system. So that's where the farming is going to become more sustainable from the disease perspective. And what impact these products that they have brought in the farming community. So you can see here. So we have launched first product in 2008 as Rajdhani BLB resistance, and then we brought BLB plus BPH resistant in 2017. And one example here, the farmer is being recognized by our Honorable Prime Minister by growing one of our product, and there that farmer was able to realize enormous yield, like 11 quintal yield per acre in the variety versus 30 quintal in the product that was bacterial blight resistant. So that kind of impact it is bringing in whole farming community, and also it is making the farming more sustainable. And this is the journey, for example, in Bayer itself. So we started putting the product in 2008, and now we have multiple products with multiple stress tolerance, and that is helping the farming community a lot to manage their crop. And going forward, our innovation will also help to define and design some tailored solution where we can integrate multiple options together based on the disease prediction. We can recommend that this variety, this chemistry can go together so that farmer can really have a tailored pest management solution going forward. So that is the direction that we are taking going forward, and that is going to help our sustainability commitment that we have made in United Nations, that we are going to help to reduce the 30% greenhouse gas emission by 2030. And we are also reducing the environmental impact of crop protection chemistries by 30%, the way we are developing the resistant products, for example. And we are also empowering the smallholder farmers across the regions. Our target is to reach 100 million smallholder farmer by 2030. So this is our commitment to smallholder farming community, and the way we are working and the way we are advancing the innovation and technologies. So we are very sure that we are going to touch the farming community and help them to grow healthier crop going forward. So all these activity was done in collaboration with different stakeholders. For example, CCMB, these scientists, Dr. Ramesh Sonti and group, then IMTech in Chandigarh, Dr. Prabhu Patil and his group, and then IIRR, Dr. Ramesh Sundaram and his team was there. We collaborated, and IRI, International Rice Research Institute, Dr. Vera Cruz, and also in California University, Dr. Pamela Ronald, and Colorado State University by Dr. John Leach and group. So this is the continuing effort, and that we are on the right path to define and design the technologies that can really help to farm community in better way. And I believe we all together, whole scientific community is towards that, and only to help farmers to grow healthy crop and have less crop damage. So that's all from my side. And thanks a lot for this opportunity. If there are some questions, I will be happy to take. Thank you.
Thank you, sir. Is there any questions for him? Sure.
Good morning, a great lecture. Hello.
Good morning. Yeah.
So I just want to ask if the sweet proteins have now been advocated to be, able to protect plants from Xanthomonas infection. Do you have any
Hello.
comments on this? Should we work on sweet proteins? Sweet proteins also reduce plant growth, also overexpression in the rice.
Yeah. So yes. So if I could understood your question, so you are talking about sweet protein. So means there are a lot of effort that is being done on that direction. And when we were working with IRI, so they were targeting some of those genetics to edit, because those are the sweet genes, those are targeted by the pathogen. So what we have seen, those genome edited lines, they are not having any yield drag as of now. So as Dr. Channa Prakash was also mentioning in the morning, so whenever we are doing or we are editing some of those genetics, we need to be really very careful, and that need to go in enormous testing so that we are making sure that there is not any kind of yield drag and all those. But what we did is through conventional breeding, and what we have found, there is no yield penalty and all. But there are certain reports, you are right, that sweet genes, because they are the sugar transporters, if they are being hijacked by the pathogen, maybe because those sugars, they are required for the grain filling also. So means we need to really test enormously before launching any product. But as of now, we haven't seen any yield drag.
Thank you.
Right.
Anyone else? No, so that's all. Thank you so much, sir, for joining and for a wonderful talk.
Thank you. Thanks a lot.
Well, we are now about to begin the meeting session, which would be continuing for the next half an hour. So all the other sessions will be happening after that. So we are waiting for our dignitaries to join. They're already outside. Yes. Check. All right, ladies and gentlemen, sorry for keeping you waited. We were actually waiting for our dignitaries to join inside the hall. They were having a visit to our stalls. This is the meeting session that we are about to run for the next half an hour. And we have some felicitation to the dignitaries and the speakers over here. They're already outside the hall, so we are just waiting. So our chief guest for the day, Dr. Mrityunjay Mahapatra, DGM, Indian Meteorological Department, Government of India, New Delhi. Ladies and gentlemen, give him a big round of applause. Also, our guest of honor for the day, Shri Sudhir Ranjan Mishra, Head Corporate Affairs, Paradeep Phosphates Limited, Bhubaneswar. Along with our Program Director, Agri Vision 2024, Dr. Engineer Divyanshu Prasad Das. Requesting our dignitaries and to please join us on stage. Please give them a big round of applause. Dr. Mrityunjay Mahapatra, DGM, Indian Meteorological Department, Government of India, New Delhi. Sir, please join us on stage. Also requesting our guest of honor, Shri Sudhir Ranjan Mishra ji, Head Corporate Affairs, Paradeep Phosphates Limited, Bhubaneswar, to please join us on stage as well. Alongside our Program Director, Agri Vision 2024, Engineer Divyanshu Prasad Das. As we commence any beautiful day with the lamp lighting request. Okay, as we commence any beautiful day with seeking the blessings from the Lord above, I'll request my dignitaries to please do the honor of lamp lighting first. Also requesting all of you, if we all can stand for the moment. Thank you so much, everyone. As we do it in Odisha, let's say this together. Jai Jagannath. Thank you so much. Also, tomorrow is a very auspicious day for all the Indians. So, as we know, Prabhu Shri Ram ji is coming to their own home. So, एक बार उनका नाम भी ले लेते हैं, रघुवर सिया राम की. Thank you so much. Chaliye. And as we proceed to the day, let me tell you all, we are not taking much of the time for this meeting. We will just take half an hour. But yes, I must tell this in one sentence that all the people you are watching over this dais are the people who have always been there for AgriVision and for always been there with the good things that we think about agriculture in future. Give them a big round of applause. And with that note, I'll request our guest of honor, Shri Sudhir Ranjan Mishra ji, Head Corporate Affairs, Paradeep Phosphate Limited, Bhubaneswar, to please speak to the gathering.
Thank you, ma'am. Thank you, Divyanshu, for inviting me to in this very precious audience and along with sharing the dais with Mr. Mrityunjay Mahapatra, one of the greatest son of the soil, the pride of Odisha. It's really nice meeting you today in the morning, our oral discussion and everything. And as we saw the stalls and the participants were explaining the beautiful thing that is happening, root level things, and that are being cumulating the central effect overall India, what is happening in the sector of agriculture, horticulture, fishery and everything. It's a nice study in the morning of a weekend and we have come over and discussing with you. So whenever I really welcome from the family of Paradeep Phosphates Limited. So whenever it coming about the agriculture scenario of the state, I always feel that CRRI, the central, when we Rice Research Institute, whatever we are reading in schools, it's the Asia's biggest research institute that time. So it's still continuing, and that is the research foundation if it's CRRI. If OEOT is the education foundation for the state for the agriculture point of view, then I always feel that from industry point of view regarding agriculture, Paradip Phosphate Limited is the guardian of the state. We are there with the farmers since 40 years. We have the plant at Paradip, and on that offices and our field officer everywhere distributed entirely dispersed and distributed throughout the country and helping the farmers, you can say 24/7 in terms of our product, fertilizers, and also with the techniques what is happening. And with the farmers, we are growing, and with us, the farmers are also growing in terms of techniques, in terms of new fertilizer, new products, in terms of pesticide that are coming to them, and we are exploiting them day and night. And for that purpose also, we have our social responsibility in handholding the farmers. So we have our plant at Paradip, as I told, and we have an office at Bhubaneswar, the corporate office where the central marketing team sits. And we have our central soil test lab. We have a MSTLs. We have, like Government of India, Odisha has given us 30 mobile soil testing vans, which is operating throughout the 30 districts. When anytime you can call the MSTL, they will go to your people and have their soil test and everything done and certificate given at the doorstep. And beside that, we have a farmer training school at Baramunda in Bhubaneswar, where we are regularly, we have the training mandates from not only from Odisha, throughout the country, from Chhattisgarh, from Jharkhand, from Maharashtra, everywhere farmers are coming and they are getting trained over there from the panelists, from CRRA, from the OUT, and everywhere is coming, and the latest techniques, and they can do at their doorstep, at their villages, everything. They are coming and they are getting trained over there. So overall, farmer, I mean to say that it's not only about product we are promoting, it's about the entire inclusive backbone of the central farming system of the nation, you can say. Paradip Phosphate Limited has a great say in that. And regularly, whatever the soil test certificate, soil test samples, and the training mandates we are giving, that has been slowly day to day, every day, you can say, building up the knowledge level, the source level, and the resource level of the common farmer, not only in Odisha, but in the entire country also. So that way, I always feel proud that no, we are not at the level, we are selling only our fertilizer things, but we are handholding them day-to-day basis. Like that, from our CSR point of view, every time in the villages and all that, we have our in the Jagatsinghpur district, Khordha district, even Puri, everywhere. We have that farmer, we are community-based, helping them in the availing agriculture loans, availing the new technique. Recently, only in the Jai Kisan Day, you can say that our Charan Singh's birthday, that the Kisan Divas, we have given 3 drones to the 3 SAGs in Odisha. That way, in there, the nano DAP or the nano urea, that technology is coming, so they can distribute the pesticides or the real fertilizer in their land through drones. So that technology or the latest whatever is PM office or the central system is promoting, that we are also bringing them to their doorstep. So in that way, we are slowly attaching to the entire level what you are researching over here to the farmers development, that we are doing also in point of the industry point of view. So I'll not take much time. Thank you very much for attending the session in the morning and the Saturday morning. Thank you very much for coming over here. Thank you, sir.
Thank you so much, sir, for quickly taking us through the journey of PPL and always being there for the farmers and for the community of agriculturals. I'll give him a big round of applause, and now we move ahead. Can we hear the applause, please? Thank you so much. Well, now we proceed with our chief guest of the day, Dr. Mrutyunjay Mahapatraji, DGM, Indian Meteorological Department, Government of India. So please guide us and please speak to the gathering.
Namaskar. Dear Sri Sudhiranjan Mishra, DGM Corporate Affairs, Paradip Phosphate Limited, guest of honor of the day, Engineer Dibyangshu Prasad Das, Program Director, Agri Vision, Professor Vaishnav Saran Tripathi, former Vice Chancellor, Ravenshaw University, Dr. S.S. Das, Botanical Survey of India, Dr. Arup Mukherjee, Scientist, NRRI, Cuttack, Dr. B.K. Sahu, former Vice Chancellor, Showa University, distinguished professors, delegates, students, ladies and gentlemen. At the outset, I congratulate Agri Vision and SARM for organizing this excellent program for consecutively for 6th year. The objective of this program, as you can find out, has been to bring out awareness among the public, to connect the public with the various resources in terms of the institutes, organizations, the professionals, and the entrepreneurs. As I have been associated for quite some time, I have seen the growth of this organization, the contribution of SARM, and especially the contribution of engineer Dibyangshu Prasad Das. Wherever and whenever you find any success story, you will find certainly there will be an organizations, but there will be one person or two sometimes steering the organization to the pinnacle. So therefore, I congratulate and thank Mr. Dibyangshu and his team for growing these organizations, bringing to this stage and helping the people to understand the role of agriculture, the role of technology in agriculture, and the relationship between technology and the success stories, which has been witnessed today when you go through from stall to stall. So all of us know that India is an agrarian country. The majority of the population depend upon still agriculture, even though India is moving towards the industrial economy. At the same time, we also know that the irrigation facility in the country has not improved so much, and still more than 50% of the cultivated area is not irrigated, and hence it is rain-fed. And the majority of rainfall comes during the monsoon season. And monsoon season rainfall, if you just look at it, is just random, erratic. It can vary from one place to another place. And also, the total quantum of rainfall distribution during the season itself varies, leading to sometimes dry spells and weak wet spells or the floods and droughts. It also shows large-scale variation from year to year, leading to the annual droughts or annual floods. While the rainfall affects all strata of life, all types of activities, including human health, agriculture, industry, etc. But being an agrarian economy, and being more than 50% people dependent upon the agriculture. So any kind of deviation from the normal pattern affects our economic health. So in this background, Agri Vision plays a very important role to educate, aware, and utilize all the technological inputs for not only minimizing the input cost of the farmers and enabling them to utilize all types of technology so that their cost is minimized, but also to enable them to utilize all latest tools and technology for all types of agricultural operations, be it sowing or be it crop seedling or be it irrigation applications or be it the pesticides or fertilizers. And finally, it helps to improve the yield. When the government of India has the program like doubling the farmer's income, you cannot proceed without all these components being safeguarded and strengthened at the individual farmers level and also in the organizational level. So while I went through the exhibitions, I found that there are many innovative initiatives by the people across the country. There is a cohesive force, you can say, in this field, which has sustained our economy. We can all remember the COVID period for 3 years when the major economy in the globe failed, but India succeeded. It was mainly because it was an agrarian economy. Agriculture could survive, it could sustain our economy, and we did not fail. Our growth continued and was comparatively higher to many leading countries of the world. So as a meteorologist, I just want to say that the agriculture has been supported largely by the scientific community. We have done many things starting from 1932, when the first agro-meteorological advisory services started in the country. The organization was set off. In 1945, state-level agro-meteorological advisory services started in the country with Maharashtra state. Then, as for the Planning Commission's requirement, the country was divided into different agroclimatic zones, 128, and afterwards it became 130 or 31. And the advice was provided for agroclimatic zones. Then came another era when we went to resolve the products to higher resolutions, and we came up with the district level in 2008. And since 2021, the products are being provided at the block level. So these products are being generated in a public-private partnership, where we have the Indian Council of Agricultural Research, India Meteorological Department, state and central agricultural universities, central agriculture department, and also the state agriculture department. Every Tuesday and Friday, there is a meeting among all the stakeholders. And finally, the weather advisory given by the India Meteorological Department, supported by the crop advisory generated by all these KVKs, district agricultural meteorological unit, and universities like UAT. They support all this. And finally, it goes as a product in their own language. A farmer can understand in his or her own language. To further reach out to the farmer, we have got the mobile app, that Meghdut, by which it reaches and people utilize these informations on day-to-day basis. There are also participation from various other groups, be it the fertilizer companies, or be it the NGOs like MS Swaminathan or Reliance Foundation. So it is a success story in the country where there is a public-private partnership, there is a partnership from the stakeholders, and there is a partnership among the academic institutes and R&D institutes, and of course, central and state agencies. I will say it is one of the most important innovation, and it is to grow. We have to handle the farmers. There are many studies, as we have seen in the field, and as we have seen in this exhibition also. The application of this information has enabled a farmer to gain in economy. We conducted an independent survey. If a farmer utilizes weather information along with the crop advisories being generated in the agrometeorological advisory services, a farmer who is a below poverty line farmer, a farmer who belongs to the rain-fed area without any irrigation facility, a farmer who has got marginal land holding less than 2 acres, he or she, if utilizing the informations, gains 12,500 rupees per year. So you may think that this is what this is actually. This is nothing, but in this era, 12,500 is nothing. But yes, it is a big amount for a poor farmer who belongs to below poverty line, who does not have much land, who doesn't have the irrigation. With this 12,500 rupees, just imagine, he can purchase the dresses for the children, he can repair the house, he can dream for educating the children. So like many other initiatives. And when you consider the country's gain, so we made a rough calculation that if you consider all the below poverty line farmers with land holding of less than 2 acres, it comes out about 13,300 crores per year. So you can just imagine how much it is benefiting the society, the country, and the individual farmers. And that second, I will tell you, we have reached only up to 3 crore farmers in the country, and there are 10 crore farmers in the country. So if we just took it as a revolution, if you just partner with each other, if you utilize all these technologies what AgriVision has enabled to understand today, then I am sure the economy of each and every farmer can be not only doubled but multiplied in the coming years. Not only it is helping in agricultural operations, but also the information being provided is helping to minimize the loss of lives and properties from the disasters. Perhaps you might be knowing that Odisha is such a state which experiences all types of disasters like cyclones, floods, droughts. Every year you will have the impact. Also, you have got the heat waves, cold waves, fog, thunderstorms, and lightning. So while number of deaths has been reduced due to the cyclones or heat waves, but number of deaths is still rising. And every year almost 300 people die because of the lightning. And we made a study. Who are dying? We found that these are the farmers. The maximum death in the country occurs to the farmers. And again, you will believe or not, the maximum death occurs in the month of July and August, because farmers are exposed in July and August. They are in the field, and also there is a monsoon. The frequency of lightning is more, so therefore death is more. Another study we found out that even though frequency of lightning is maximum in Northeast India relative to Eastern India like Odisha, death is more in Eastern India like Odisha, Jharkhand, Bihar, West Bengal. It is because that lightning occurs or thunderstorm occurs in Northeast India at night or early morning where people are not exposed. Day before yesterday, I was in a meeting. So someone pointed out to me that in USA, the frequency of lightning is more, but death is quite less. It is natural. It is a developed country. People do not work. People are not exposed. People are not working in the field, unlike our people in the country. Our people are poor. Our people are not equipped with all technologies. They have to be in the field for a longer time and hence exposed. So therefore, people die. So what I want to say that we have to take care of the farmers also. The technology has come up. We need awareness. We need some kind of hand-holding in a campaign mode so that people can save their lives and properties due to lightning. I sometimes see that a fan is damaged because of the lightning. You just imagine, you may think that भाई fan में क्या है यार, हमारा तो AC ठीक, AC चला गया 30,000 50,000 रुपये और fan तो ठीक है यार. But for a farmer, a fan is very precious. He cannot purchase a fan again. So therefore, he needs some kind of awareness campaign so that he can safeguard his whatever property he has due to lightning or other phenomena. So I'm talking about lightning especially because we found that farmers are worst affected nowadays because of lightning, because people are not dying because of the cyclones now. Farmers were worst affected because of cyclones, but now they are not dying. But loss of property is there for farmers. But still, they are able to minimize the losses because of the 5 days warning of the cyclones. They are cutting the harvest and making all arrangements, etc. So this is one of the worry. And then second point is that the fishery. The fishery community, if you just look at, they were the worst sufferers. But now, no fisherman is dying because of the cyclones. So that is the great success story. And someone wrote a book. They told the cyclone warning is one of the 75 most important innovation in the country after independence. It is published by Mr. Dinesh Sharma. So that is the country. It is a country's success story. But what I want to say that still the fishermen are the great contributors. If you look at the exports from India, the fishery sector is the most important sector in terms of export. Maximum export takes place from the country, from the fishery. So therefore, we should salute to those people, the farmers, the fisheries, fishermen. Those are really contributing to our economy. Those are really providing a sustainable economy, because of whom we are getting the foreign currency, because of whom we are fighting the poverty, and we are ensuring ourselves, even if the time of COVID. So on this specific day, I salute all our farmers and fishermen because of their outstanding contribution to the Indian economy and also to the Indian people, ensuring food security in the time of the climate change. I'll just 1 or 2 points more. As I told, the climate change. Yes, climate change is a threat. And all of us know that the climate change threat is more towards the poor people, towards the poor countries. And therefore, we have to do something more. Even if there is a projection, and whatever we've seen during past 100 years, that rainfall is not going to affect it, but rainfall distribution is going to be affected, and therefore we have to be careful and we have to guide our farmers. For example, the total amount of rainfall remaining same. The onset date of monsoon has shifted for central India, starting from Odisha to Maharashtra and Gujarat. It has shifted by about 1 week or 10 days. So withdrawal has been delayed. So therefore, the seasonal pattern of the rainfall is changing, and accordingly, therefore, you can see, and the temperatures. The temperature in winter is changing and is rapidly increasing, about 1 degree Celsius in winter, temperature rising if you go towards north. So therefore, there are certain impacts on the optimum temperature requirement for each and every agricultural system. So therefore, we have to go for massive campaign, explain to the farmers what type of adaptation is required, and there is the role for agricultural scientists and academicians, R&D institutes, and of course, agrometeorologists. So the knowledge which has been gained over the years, that should percolate to each and every farmer. Because nowadays, the literacy level has also increased in the country. The farmers are also able to understand the various types of phenomena which is occurring in the country. So there is a good scope here now to work together and help the farmers, to help them in terms of increasing their knowledge base, to help them, to capacitate them, to intervene in the farming sector with the new technology, which is low-cost technology, which can help to minimize their cost and increase the yield. At the same time, also we should make them aware about the various types of disasters, the impact of the climate change, what type of adaptation they should go, and what type of mitigation they can go for in the coming years. Yeah, I was very happy to know that various types of varieties are coming up, which are resistant to the change of climate. And certainly it will help. But we have to think about next 50 years. And research should start now, keeping in mind what will happen, say in 2050 or 2075 or 2100. Because research is a slow process. The development of a particular seed or development of a particular technology does not come in a day or two. It takes time. So therefore, the foundation should be built up, keeping in mind what will happen if there is a rise of temperature by 2 degrees Celsius, which is projected by the end of this century, that is 2100. At that time, what type of agriculture would be? Because all of us know there are projections based on this 2 degrees rise temperature. It is projected that the paddy yield will decrease by 6 to 10% and 10 to 15% in the irrigated area. Similarly, the wheat, a major crop I'm talking about. So we should think now what type of paddy or what type of wheat or what type of seeds we should adopt for, so that this projected decrease can be counted and we can go on increasing. That is possible. I'll tell you, when we got a study 10 years back that climate change will have the adverse impact on the predictability of the weather, especially the rainfall. They predicted that because of the climate change, if you are predicting a heavy rainfall event 5 days ahead, now you will be able to predict 2.5 days ahead. That means predictability will be halved because of the impact of the climate change. So what we do then? We went for mitigation adaptations. We found that, yes, this heavy rainfall increase is because of the mesoscale events, very small scale heavy rainfall events occurring because of the low pressure systems. So therefore, if you want to predict these with the same predictability of 5 days in advance, then you have to increase their detection. You have to increase the number of observational systems, radars, and satellites. And accordingly, the data collected should be ingested in the numerical models. And numerical model resolution should increase, science should increase, physical concept should increase, and hence accuracy should be sustained. And that happened. In the last 10 years, if you just look at, the accuracy has increased by 50% in spite of the threat due to the climate change. So that I am sure. So this type of intervention of science leading to sustainability, not only sustainability, but also enhancement of the capacity in all fields, be it agriculture or be it agriculture technology or be it the early warning, all should be aimed at so that by 2050 or 2100, when we realize the actual climate change, our growth, our economy, our agriculture is not affected. And hence we can sustain our economy. So if you just look at the climate actions, the 13 ensures the climate actions, and that is meant for the sustainable development. That is SDG 15 I'm talking about based on the HIGO framework aimed at 2030. So it says the first one is that to ensure life. Second one is the livelihood. So all these things depends upon our actions. So on this occasion, I urge upon all the students, the practitioners, researchers, the professors, to be a guiding force to our poor farmers and ensure the sustainable development, keeping an eye about the possible climate change in the future. I once again congratulate Dibyanshu and the entire team, SARM, for this continuous effort to help the country, to help our system development, and finally, to help the farmers for their sustenance, their livelihood, their safety, and also growth of the country. Thank you very much.
Thank you so much, sir, for always boosting our morale. Now we move ahead with the academic and business awards. The first few felicitations are for Dr. Sabut Sahu Memorial Lifetime Achievement Award. I'll request my dignitaries to please step ahead. So the first receiver of award for Dr. Sabut Sahu Memorial Lifetime Achievement Award is Dr. Sarat Kumar Pradhan, ADG, ICAR, New Delhi. He'll be receiving his award virtually. So can we please connect to him on screen? Dr. Sarat Kumar Pradhan, we will connect to him
I'm
on
connected.
screen virtually.
I'm connected. Hello. Hello.
Can you please
Hello.
connect to him with the Zoom call? Dr. Sarat Kumar Pradhan.
I'm able to see from here. Hello. 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 Hello.
Yes, Dr. Pradhan is over there.
I'm able to listen. Please go ahead.
Dr. Pradhan, can you hear us?
I can hear.
Thank you, sir. Thank you.
Hello.
So we are now felicitating you with the Dr. Sabut Sahu Memorial Lifetime Achievement Award. You are receiving this.
Thank you very much. I'm able to listen. Go ahead.
Please accept it, sir. You are receiving it from Dr. Mrityunjay Mahapatra, DGM Indian Meteorological Department, Government of India, New Delhi, along with our guest of honor, Shri Sudhi Ranjan Mishra ji, Head Corporate Affairs, Paradeep Phosphates Limited, Bhubaneswar, and our Agri Vision Program Director, Engineer Divyanshu Prasad Das. Congratulations, sir. I guess.
Hello.
Yes, sir. We can hear you. I guess his screen is freezed. Everybody, give him a big round of applause. Yeah.
Hello.
Yes, sir. We can hear you.
Should I?
Okay. So, okay, the next one for the Dr. Sabut Sahu Memorial Lifetime Achievement Award goes to Dr. Bijay Kumar Sahu, former Dean IAS and Advisor SO University, Bhubaneswar, Odisha. Sir is already here. Give him a big round of applause.
Tell there is some problem here, network issues.
Congratulations, sir. . Okay. Thank you so much. Thank you. So next award is for Krusak Bandhu Award, which is being received by Shri Lalit Sahuji, a farmer. I guess he's not present, but we will definitely give a big round of applause for him. Okay. And the next felicitation are for some of our plenary session speakers. First one goes to Professor Johnson Ed Sudan from Ekiti State Polytechnic, Izan Ekiti, Nigeria. Sir, can we please have you on stage? For the next, I'll request Dr. Om Prakash Dhankar, Professor Biotech, University of Massachusetts, Amherst, USA, to please join us on stage. Thank you, sir. Congratulations. Requesting Professor Jason C. White, Director, Connecticut Agricultural Experiment Station, to please join us on stage. And next one is, last but not the least, Dr. Sudhanshu Shekhar Das, Scientist F, Botanical Survey of India, Kolkata. There are plenty of felicitation in this category, but we are doing it every day, couple of felicitation. Now I'll request our program director to felicitate our dignitaries. First, our chief guest, Dr. Mrutyunjay Mahapatraji, DGM, Indian Meteorological Department. Thank you, sir, for giving us your time. Ladies and gentlemen, please give it up for the Cyclone Man of India. And now to our guest of honor, Sri Sudhi Ranjan Mishra ji, Head Corporate Affairs, Paradeep Phosphates Limited. Thank you, sir, for always being there with us. And now, before we close the meeting and proceed with the plenary sessions again, I'll request our program director, Sri Engineer Devyanshu Prasad Das ji, to propose the vote of thanks.
Namaskar, Jai Jagannath. This second day of 6th International Conference on Agriculture for Sustainable Development, Agri Vision 2024, which is being hosted by Society for Agricultural Research and Management in collaboration with ICAR-National Rice Research Institute. I'd like to thank our host institute, NRRI, for being our collaborative partner in organizing this international conference. I'd like to thank chief guest for the day, Dr. Mrutyunjay Mahapatra sir, for sparing his most valuable time for us and blessing us with his presence. I'm really thankful to the guest of honor of our day, Sri Sudhir Ranjan Mishra, DGM Corporate Affairs, who joined us and graced the occasion of AgriVision. I'd like to thank all our revered speakers, renowned scientists who joined us from India and abroad and enlightened this August gathering. I'd like to extend my thanks to all our sponsors, our diamond sponsor, State Bank of India, silver sponsor, Tata Agrico, and not in order, IREDA, Coir Board, National Rice Research Institute, Botanical Survey of India, India Post, Fishery Survey of India, Ministry of Ayush, Government of India, NFDB, National Fisheries Development Board, Spices Board of India, NALCO, Annapurna Finance, Odisha University of Agriculture and Technology, CRIPCO, Central Institute of Fisheries Education, Central Warehousing Corporation, Centurion University, Central Marine Fisheries Research Institute, Central Tuber Crops Research Institute, Central Institute of Freshwater Aquaculture, Indian Institute of Horticulture Research, Odisha Hydropower Corporation, Odisha Language, Literature and Culture Department, Central Institute of Women in Agriculture, Coconut Development Board, National Medicinal Plant Board, OMFED, Directorate of Veterinary, Government of Odisha, Paradip Phosphate Limited, Central Cattle Breeding Farm, CCBF, Sambalpur, Chiplema. I would like to thank all our participants, our students and researchers who joined us from different universities, from different corners of India. I'm thankful to all our supporting staff who are tirelessly working in making this event a grand success. I thank all the print and publicity media for widely publicizing this event. I'm thankful to all our crew members without whose support this program is not possible. I thank to all the founding members and the members of Society for Agriculture Research and Management for their guidance in organizing this conference. With this, I close my vote of thanks. Thank you so much. Jai Jagannath.
Thank you so much, sir. Now we'll proceed with the plenary session. Thank you so much, sir. It was an honor having both of you here. Okay, all right. We'll now proceed with our plenary session as it was already planned. So, requesting all of you to kindly be settled. Okay, so the next one is by Professor B.C. Tripathi, Professor and J.C. Bose National Fellow at JNTU, New Delhi. He is talking on the topic evolution and application of light-dependent chlorophyllite oxidoreductase enzyme to increase photosynthesis and plant productivity. So please. Get a pointer. Answer it up number.
Bus. Go back.
Okay. It screen along.
Okay. Okay, the talk is approaches to reduce generation of reactive oxygen species. Okay. And increase photosynthesis and plant productivity. There are two different approaches we have taken, but due to shortage of time, I'll only skip one. But before I go, I must say this, you might have said this, but these are chlorophyll biosynthesis pathway written here. One is synthetic pathway, another is degradation pathway. But I am saying here that chlorophyll biosynthesis and degradation pathway are also interconnected. Okay. And we are modulating one of the enzymes of chlorophyll biosynthesis. Here I have said two enzymes were modulated, but here I am talking about one enzyme today. We modulated chlorophyll biosynthesis pathway. So I'll skip one because you don't have time for that. So I'll just go to one of the enzymes we have modulated for to increase photosynthesis and plant productivity. This is protochlorophyllite oxidoreductase. You see, plants have, an enzyme, photoenzyme called protochlorophyllite oxidoreductase. Its function is to convert protochlorophyll to chlorophyll during chlorophyll synthesis. When seedling grows, germinates and grows, massive amounts of chlorophylls are synthesized in the seedling. Okay, otherwise in response to light. But you see that this I talk about, I don't know here. This is chlorophyll biosynthesis pathway in nutshell. Glutamic acid goes right to the delta aminolevulinic acid, give rise to protoporphyrin 9. If magnesium inserted, it goes to chlorophylls. But if iron inserted, it goes to heme and cytochromes. In plants, both the pathways operating magnesium pathway and iron pathway, both are operating. By the animals, the magnesium branch is shut down. Okay. So we have protoporphyrin 9, magnesium inserted with magnesium protoporphyrin 9. Then you have protochlorophyllite. So finally synthesis protochlorophyllite. Then protochlorophyllite is converted to chlorophyllite by the enzyme in presence of light by the enzyme protochlorophyllite oxidoreductase called POR enzyme. Okay. POR enzyme is a light dependent enzyme. It needs light as an obligate requirement for synthesis of chlorophyll. And once chlorophyllite is formed, it is phytylated to form chlorophyll. Okay. So this is a nutshell the chlorophyll pathway. I'm talking about one of the enzymes of in the nature is the proteochlorophyll reductase. There's another enzyme, also widely known enzyme, which needs light as a substrate for to its action. The DNA photolyase, which is needed for DNA repair. Okay, and pyrimidine dimer repair in UV light repair. And I'm not talking about that today because it's not present in higher plants either. So, okay. So we are talking about this enzyme here, which needs light enzyme. Okay, photoenzyme called photoenzyme. If you see in the process of evolution, this is protochlorophylite structure, and this is chlorophyllite. What is happening here? The double bond here is reduced to single bond here. That is the difference between protochlorophylite and chlorophyllite. Okay. And this enzyme, protochlorophyllite oxidoreductase, converts protochlorophylite to chlorophyllite. Now, once chlorophyllite is formed, then phytyl tail is attached here to form chlorophyll. Okay. All right. So this protochlorophyllite and can be converted to chlorophyllite by light dependent enzyme and light independent enzymes. If you go to algae, fungi, algae, bryophytes, pteridophytes, and gymnosperms, pines and all, they can green in dark. Okay, they can synthesize chlorophyll in dark because they have light independent pathway, light independent chlorophyll biosynthesis pathway, light independent enzyme. Okay, these light independent enzyme has 3 subunits: CHL, CHLN, and CHLB, and these 3 genes code for a light independent protochlorophyll oxidoreductase, a light independent pore, and these are present in chloroplast genome. Okay. So in the process of evolution, these 3 genes have been deleted from angiosperm genome. So angiosperm chloroplasts do not have these 3 genes. Okay, therefore angiosperms cannot green in dark. Okay, they need these 3. They are only have photoenzyme, the light dependent enzyme, photoenzyme, and it has 3 isoforms: pore A, pore B, and pore C. Pore stands for protochlorophyllite oxidoreductase, the enzyme that I am focusing today. Okay, to synthesize chlorophylls. Okay. If you grow a plant in dark, the plant shielding is yellow, not green. That because plants do not have, angiosperms do not have these 3 genes in their chloroplast genome. Okay. All right. So when I say pore A, pore B, pore C genes there, so there are, in Arabidopsis, there are 3 genes: pore A, pore B, and pore C. And their similarity is very high. Well, you can see how much they spare. Pore A is 1 to 108 base pairs, pore B 1 to 06 base pairs, pore C is 1 to 06 base pair. You can show significant relationship by this all happened by gene duplication events. But this pore has, but their similarity ends. Okay. So while looking at the Arabidopsis genome, we came across a gene which is called pore C, and we put this genome, this protein, this gene in E. coli, expressed in E. coli, and found out, yes, it's similar to pore A, pore B, but yes, it can also synthesize chlorophyll in light. So what we did, we expressed in the bacteria, clone this, expressed and purified in nickel column, and the protein. Then we took a cuvette, fluorescent cuvette, added protochlorophyllide oxidoreductase, isolated protochlorophyllide from the seedlings, put them together, okay? And this is the absorption fluorescence spectrum of the substance. This is protochlorophyllide absorption fluorescence spectrum at 635 nanometer. And as you see shine light, you can see here the protochlorophyllide fluorescence goes down and chlorophyll fluorescence comes up. Okay. Okay. So from this experiment, we proved that this enzyme is really can convert protochlorophyllide to chlorophyllide. Okay. So this is a C isoform of protochlorophyllide reductase. We call it pore C. It is present in Arabidopsis and most brassicales, including Brassica gentiana and many other crop plants, but not most, very few. But it is one thing very important for this gene expression is that it does not express in dark, only expresses in light and is highly expressed in highlight. Okay. So pore C gene expression we observe is highly expressed in highlight. Okay. Therefore, you clone this gene. I'm not going to all these steps here because of time. So you have cloned the other one to overexpress this gene in Brassica itself. We took this gene Arabidopsis and cloned in Brassica and overexpressed in Brassica. Okay. And we saw we got the transgenic plants here. Okay. And these are the gene express. This is a southern blot showing that gene is already incorporated and single insertion. And we got a good expression pattern. And also you can see northern expression and also western blot. Okay. Showing that the protein is overexpressed into the wild type plant. These are two transgenic lines. Okay. Showing that this plant is overexpressed in transgenics in Brassica. So once you got a transgenic plant, we measure the chlorophyll content. The chlorophyll content increased. You got a chlorophyll, carotene content also increased. Okay. Okay, I'll skip this slide because I don't have time for this right now. Yeah. You see, protocol. What is important here? We don't want protochlorophyllide to accumulate in plants. Okay. What happens during, you see, protochlorophyllide synthesis takes place all the time, but day and night, both. Okay. But in the dark, protochlorophyllide synthesis is reduced. Okay. Because protochlorophyllide is photosensitized. What it does, when I showed you slide here, once protochlorophyllide accumulates in the plants, it inhibits its own synthesis by feedback inhibition, where glutamic acid is converted to ALA. So this by feedback inhibition, it inhibits its own synthesis. Okay. Because plants do not want to accumulate protochlorophyllide, therefore, because protochlorophyllide is a photosensitized. And once it absorbs light, it can go to excited state. It is tetrapyrroles and this is protochlorophyllide. The photosensitizer, it can absorb light, go to excited state. When it come back to ground state, gives energy to oxygen, which becomes single oxygen. Okay, who is highly toxic. So here the light energy absorbed by the pigment is now converted to ROS, reactive oxygen species, called singlet oxygen. Okay. And this singlet oxygen is very highly reactive and can kill the plant, can harm the plant. So plants, therefore, do not want the accumulation of a protochlorophyllide. Okay. So you wanted to reduce this. So this is the picture of a chloroplast in a cell. The nucleus, the mitochondria, this is chloroplast. We have shown here where the potential sites of generation of single oxygen in the chloroplast. It can also be clear generated mitochondria because they have also protoporphyrin 9 there. And this could all give a signal. The single oxygen produced can give a signal to nucleus as retrograde signaling for, for changing gene expression. Okay. Okay. So, okay. Yeah. So we spread the plants with ALA. You see, a delta aminolevulinic acid is, as I told you, is a substrate here. You can lead to photosynthesis. Okay, you substrate here for photosynthesis. As I told, protochlorophyll feedback inhibitor glutamic acid, glutamate. But if you give ALA, to spray the plants with ALA, you'll accumulate a lot of protochlorophyll overnight. If you spray ALA at sunset to the plants, they will all synthesize protochlorophyll overnight. Okay. And when in the morning, when sunlight comes, there are a lot of protochlorophyll in the plants because they cannot be converted chlorophyll in light. And therefore, this lot of protochlorophyll will absorb light, go to excited state, when come back to ground state, give their energy to oxygen, that makes singlet oxygen and kills the plant. So these are the examples here where you can see here, we are sprayed with ALA, minus ALA, the wild type and transgenic. There is no ALA treatment here, but you can always see transgenic plants are doing better than wild type, so that's better for you. But when it's treated with 2 millimolar, 3 millimolar ALA, you can see here the wild and expose them to light. Okay. So we spread them at sunset, okay, and expose them to light in the morning. As you can see here, all the wild plants are dead, where 4CX plants are alive because the ALA was absorbed by the plants. They synthesize the protochlorophyllide. Protochlorophyllide absorb light, went to excited state. When they came back to ground state, gave the energy to oxygen, with single oxygen, they killed the plant. Okay. Whereas the overexpressors, okay, did not die. Did not die because they have enough pore enzyme, the photoenzyme, the protochlorophyll reductase, which were efficiently transformed to synthesize chlorophyll. Okay. So therefore, protochlorophyll did not accumulate. Okay. Therefore, there is no damage to plant. Minimal generation of single oxygen, and then the damage to plant. And that's what we saw here. Even after 3 millimolar AL, you can see the wild type plant all dead. So, yeah, this is all again after 5 days, 10 days of light exposure. All right. So, this is up to one of the examples we are saying. This is the wild. This is after AL exposure. This plus ALA. You can see the wild type plant is green, and transgenic is in red. You can see the photosynthetic efficiency of the transgenic plant is quite maintained. The ABFM value of wild type plant has gone down very, very rapidly. Within hours of exposure. This is only 6 hours of exposure. So you can see how fast the plant is dying, okay, in response to, light exposure. Okay, similar electron transport rate, you can see here wild type plant, transgenic plants. You can much difference between the wild and transgenics. Okay. All right. I won't show you details there. Okay. And now we measure single oxygen in plants, and you can see the single oxygen production in the wild type plant in response to ALA treatment is extremely high, where transgenic plant is very low. Therefore, transgenics are survived because of their reduced minimal production of single oxygen. This is another experiment, SOSG staining experiment. We isolated protoplasts from wild type and transgenic plants, and this SOSG staining to measure the green fluorescence measured by induced by single oxygen. And you can see the protoplasts isolated from wild type plants without ALA, with ALA, the 4CX plants, and the mutant. So you can see here the single oxygen production is very high. The fluorescence you can see wild type and with AL, you can see that the chlorophyll, chloroplasts are disorganized, and you can see a lot of chlorophyll fluorescence here. Okay, single oxygen production. But POR6 plants, we have seen very low. But if you give POR6 to mutant plant, you can where no protochloroxidase present, you can see they are dying very, very fast. Okay. And also we saw the tunnel assays showing that the programmed cell death, the single oxygen produced in the chloroplast is able to execute the cell death in the nucleus. Okay, and therefore the programmed cell death executed in the nucleus by tunnel assays, so that therefore what you're saying, the retrograde signaling by the single oxygen which killed the plant, even killed the cell through some through necrosis and some through programmed cell death, PCD. Okay. All right. All right, this is basic. So we just go ahead a little bit and further and stop saying, yeah. These are your wild type plant. Yep. Wild type control plants, POR6 control plants. You can see a clear difference between this. And we gave them stress, hot stress, after a few days of stress. And after 2 weeks of stress, you can see wild type control, POR6 control. That's doing so good, but you give wild type stress and POR6 stress, you can see why there is no flowering took place. In wild type plants, or POR6 plants are gone and flowering. And also these electron stages of growth. And we measure the fluorescence electron transport rate. You can see wild type transient. You can see clear cut increase in photosynthetic rate. Okay. And you can see the stress condition is the wild type plant with very, very minimal photosynthetic mode. Yeah. And we also measured the CO2 assimilation rate by the leaves. You can see the wild type and transgenic, showing the photosynthetic capacity of the plants are increased. Okay, not only its photosynthetic capacity increased, but also it is resistant to short stress. As you can see here, the short stress have changed. Like quantum milli photosynthesis. So, and okay, this is the dry weight, this is the control. You can see here, control wild POR6 is always red, wild type is always blue. So you can see wild type POR6 overexpressors, and control plants always they have dry weight higher, 150 millimolar NaCl, 200 millimolar NaCl, showing that the wild type plants are being transient plants being better even stress conditions. And starch content you can see also much more in transgenics, even 200 millimolar NaCl. And this is the weight of the seeds per plant. You can see the yield also has reduced, but not as much as in wild type. 1,000 seed weight is also much higher in the transgenic than wild type. So I come to conclusion here. Attempts are made reducing the oxygen concentration in plant cell or stress conditions. I will skip this portion here. Overexpression of protochloroxide reductase, C, efficiently phototransformed, protochlorophyll to chlorophyll, leading to reduced accumulation of photosensitizer, protochlorophylline, and minimal production of oxygen. Reduce accumulation of oxygen, protected plant space, and increase plant productivity. Okay. Thank you. Thank you very much for kind attention.
So any questions for sir?
Yes. No,
sir, I'm coming.
Yes, yes.
And I heard you talk before as well, and nice work. I have 2 questions. You showed the with the POR6 overexpression, it's increased the chlorophyll contents, And then finally, under stress condition, it was salt tolerance. And
Yes.
so have you tested this under different stress conditions? Because chlorophyll increased photosynthetic efficiency will be affecting, under other stresses like drought and high temperature and others, it will be much better.
We tested drought.
Uh-huh.
Now we are testing high temperature. We have already done drought experiment, which I'm
Okay.
not showing here.
Uh-huh.
But drought also, there's a drought tolerance to a certain extent.
Uh-huh.
But we see better high temperature tolerance.
Okay. And my second question is because the glutamate is key to the synthesis of the chlorophyll. So under this, when you overexpress this gene, you are basically pulling more glutamate. So the carbon-nitrogen partitioning will be affected. If the plants don't have enough nitrogen fertilizers, then the glutamate will be limiting. So how was the carbon-nitrogen partitioning? Have you tested those in these plants?
Yeah, you see, only if there's nitrogen limitation in the soil,
Mhm.
then the probability may arise. But under our experimental conditions, where there's no nitrogen limitation in the soil, or we did a pot experiments, also did soil experiments also later on, okay, on the field with special permission. We saw that they're being much better. I mean, and not only that, they have also a small increase in their protein content also.
But because most of the time the farmers has to, the nitrogen will be limiting. Farmer has to, they have to put the fertilizers.
Yes.
And if in this scenario, it is pulling more nitrogen towards this photosynthesis. So that will be.
It is side change, you know, you see how much chlorophyll is there compared to protein. Much more protein and very, very less chlorophyll.
Okay.
Milligram to microgram
Yeah.
situation. So in that case, we don't expect much protein to be transmitted to chlorophyll.
Okay. Yeah.
Okay.
Thank you. Yeah. Thank you.
Anyone else? Okay, that's it. Thank you so much, sir.
Thank you.
Well, now I'll request Dr. Sudhanshu Shekhar Das, Scientist at Botanical Survey of India, Kolkata, to please join us on stage for his talk.
Good afternoon to everybody. Really, I'm very much thankful to the organizers, the Agri Vision, to give me opportunity to come here and talk to you before. And as today morning, our chief guest was saying that the Agri Vision was doing a great job for bringing all the stakeholders of the biodiversity or all the stakeholders of the agriculture in a common platform, so that it will give a great opportunity to us to interact with each other, and we may be helpful to each other. And today, as in the previous talk, like Professor Tripathi has told regarding some of the experimental of the chlorophylls, I will take you all to the field of the real science, or the field of the real botany. In fact, I'm from Botanical Survey of India, and we work basically for the floristic surveys and inventorization of the country, of the world resources. And also we look into that, what kind of different resources can be tied up with a different organization so that we can work together. And today we will look after this plant explorations opportunities and the germplasm collections in the Indian Himalayan region, and for the sustainable development. When we talk about sustainable development, as today morning our chief guest was saying regarding the SDG, the basic things is the generation, the livelihood. And that is one of the most important factor for the generation of the livelihood. And for this purpose, and this is particularly the Indian Himalayan region. When we talk about Himalayan Indian regions, this is a vast area, and more than the 50% of the plant diversity of the country is in the Himalayan. And when we go to if we start from this one, the biodiversity, the term biodiversity, as we know that the biodiversity, this is the totality of the species or the ecosystem of the region. And more precisely, when we go for that, this is the level of the diversity. The level, when we talk about the level, different level of the diversity, this may be the genetic, that is the variation within the species, that it may be, or the species, that is the variation among the species, or the ecosystem when the common, the group of common functions of plants are situated with, they interact with the biotic and abiotic environment. And and many kind of this kind of ecosystems is makes the ecosystem diversity. And where they are, when we can able to understand, because all the biodiversity of the country is basically located in the tropical region of the tropical region of the world, and otherwise known as this is the megadiverse countries. And these megadiverse countries, when we talk about the megadiverse countries, this is basically the rich in the floristic diversity, rich in the endemism, and also this is also the rich in the different kinds of the threats. And when we consider this particular, these 3 points, but is rich in the diversity and the endemism and the threats, and this comes around the hotspots. The biological hotspots, when we talk about the biological hotspot, this is a 0.5% of the total floristic area, or the floristic component of those regions are basically, it comes from the endemism. And we have two of the four, we are lucky that for example, the Eastern Himalayas are part of the biodiversity, or the Western Ghats is the part of the biodiversity hotspots. And when we come to the country, in our country level, this is a part of, this is a 4 biodiversity hotspots. This is Western Ghats, Himalayan biodiversity, Indo-Burma, and the Sundaland. And this is 10 vegetation zones with in 25 biotic provinces and 525 around biomes. And you can imagine that the different kind of ecosystem diversity in the country. And when we particularly comes to pinpoint to particularly the forest area, the Himalayan region, this is part of the 2 biodiversity. Not only the floristic diversity have the great, not only the great diversity of the floristics, but also this is the ethnic diversity, also a different kind of the ecosystem diversity in this country. Because you can see that around 50%, 58% of the total diversity, the total country's flora is present in this Indian Himalayan region. And out of which 35% plant is endemic. That is the most important thing. And the altitude variation is 200 degree to 8,500 degree, and the temperature difference is -25, -35 to +35. So that every 500 meters of height or every 500 meters of altitude, the floristic component or the floristic diversity changes. That's why we can able to understand that how many kinds of forest the wild species can interact in here, and what are the new prospects of this area. And many of this country, this area also a part of many of the botanical curiosity plants. Maybe this is from Sapria Himalayana, or maybe the Monotropa, or the pitcher plants, or the Rheum nobile. This includes also many of the economic plants which has a great gene pool in these areas, and this great gene pool makes the agricultural scientists, the horticultural scientists, or many of the commercial plant scientists to make a venture into that. And if you will see that in component wise, the Indian Himalayan regions is having, as I told you, the 58% of the total total floristic composition of that area. This is around 11,157 species is found, and out of this is if you will see that most of the germplasms comes from the Poaceae, Asteraceae, Orchidaceae, Papilionaceae, which gives the mean to say the first 10 germplasm, the first 10 families of the plants, the wild families of the plants, gives the maximum wild relatives of the plants of the country. And if you see that around 44% of the complete enumeration of the plants is present in the first 10 families only. And out of that, the maximum Poaceae or the Papilionaceae and the Rosaceae or Rubius or the Ericaceae, this makes the most commercial plants of the commercial cultivars of the things. And we know that this is one of the most important factor, why the region of this country, this region is most rich. This we can attribute it to the geological lifeline or geological scales perspectives. Maybe the Gondwana flora has been shifted to this area, or the great variation in the altitudes, that is creating a long topographic relief in where, as I told, 200 meter to 8,500 meters, so that it is make a different kind of ecosystem, the different kinds of microhabitats where the wild plants grow. And this is also the large number of endemics of the primitive families. That is one of the most important thing. What makes the Himalayan flora is one of the most important thing. And this also because of the plant, these kind of endemic or the primitive species are found in this area. This implies this is the primary center of the species, the many plant groups. As we know that this is also a primary center for domestication of plants for the millet and pulses. And not only the millet and pulses, this is a center of origin of the rice, sugar canes, mango, citrus, oriental cotton system. And if you'll see the 175 different kind of the wild, only wild rices are found in the northeastern states and also particularly in the Himalayan belt. This is one of the, apart from the eastern parts of the country, this is one of the most important factor. This is one important region where many of the things many of the wild plants has been originated. And this is not only the rice or sugarcane, but also many of the orchids has also been come into the commercial factor. And also the legumes, that is one of the most important factor. Legumes, you can say the Tephrosia, or you can say the different kind of the legumes are also starting the high altitude ophiographies to the low altitude, the Tephrosia is also found here. And this, in fact, when we talk about this great floristic wealth of these regions, the botanical survey of India, we have 4 the regional centers there, the eastern regional centers, that is having more than 275,000 herbarium specimens with us, and northern regional center, 135,000 herbarium collections, and Sikkim regional, Himalayan regional center, and Arunachal Pradesh regional center. And also we have a central national herbarium, that is we have more than 25 lakhs collections of plant specimens there. The importance of this all the huge collections is not only this documents the gene pools of the horticulture important wild plant or the crop plants, crop relatives, but paves a way for the prioritizing the future collections, the technology interventions of the research of the crop development or the drug development or for the food security or the livelihood generation. Because the entire huge collections, this opens a great collaboration opportunities for the different sectors of the agriculture, maybe a agriculture scientist or the horticulture scientist or any of the floriculture or and this is as I told you the crop wild relatives, because the last 2 decades the concern has been changed from the wild plants to the wild relatives, because if you see that it is estimated that last 2 decades, last 20 years, we have collected more than 60% of the total the wild crop relatives what we have already present. And this conservation efforts, because Botanical Survey of India, we also have having the botanical gardens throughout the country, and this conservation effects, the impacts, what we are planning to do what are the main challenges, and the emphasis the specific richness and also the infra-specific richness of the variations inside the species. And the wild relative, many of the wild relative cereal groups, we will see that, as I already told, the 175 type of wild rice, in fact, is found in northeast states and also the Indian Himalayan regions. And also the wild relative, the solanum, you can say that solanum, we have a 69 species of wild plants of the solanums in the particular Indian Himalayan regions. And if you see that's cucurbitaceae, from we have around out of 104 cucurbitaceae, we have around 65 to 70 species of cucurbitaceae in there. And piper, only in Arunachal Pradesh, we have a different kind of 21 species of piper species in Arunachal Pradesh. That's why this makes a great, huge wealth of the wild relatives of the many of the crop plants and of the commercial plants which can be explored or which can be utilized. And you can see the taros. This is different kind of alocaceae. 19 species of found only in the Arunachal Pradesh we have. And if you see the Dioscorias, the 29 species of different kind of Dioscorias, and this is most important thing. The species, many of the species are been edible by anyone, edible in the traditional way and edible in the commercial way. And when today morning I was seeing in one of the stall, the Tuber Research Institute, where they have displayed the different kind of this particular alocaceae and also Dioscorias. And there is a great scope for these particular species or particular wild species can bring into the cultivation, and so that it can be not only the livelihood of the local area and not only the livelihood, but also this will secure the food security of this region. But the most important thing is the dynamism, the taxonomy, where we place a important role, because the naming of these plants has been changed. The dynamism of the collections, the dynamism of the different kind of the identity of these plants has been most important things, because some of those plants has been poisonous too. That's why where this taxonomy plays is very important thing. And sometimes before, when the different kind of digital era was not there, many of the species which have been found in the endemic to this particular region has now been found in the other part. Or what are the things, the adjacent countries, what are some of the species has been found in adjacent countries is now also found in our country. That's why you can take the example of the archidendron, one of the species, the archidendron, which is equivalent to the pongamia, and this is very rich in, the seeds are very rich in oil. But however, these species were not found in India, and this was only found in the China and the Vietnam and Laos. But recent explorations in these areas, particularly in this Arunachal Pradesh, we have targeted. We have collected around 18 to 17 species of archidendrons from this area region, and where we have new discoveries also included. Similarly, the rubus. Rubus is one of the most species which can be bring into commercialization, or which can be bring into the culture, the development of fruit. And this has been recently worked out, and we have taken that. Minimum 29 species of rubus species has been eaten by the traditional population. This is Echiniaceae, one of the species recently been collected and described from the Arunachal Pradesh, and this is one of the very important species for timber productions in the tropical forest. And this is many of the species which is found in also the endemic plants is found in this region, Himalayan region, is one of the most important thing is about the orchids. And you can see bulbophyllums or the gentiana. What is the wild relative of the gentiana curo and the archidendron? This is a fruit, one of the simplest. The fruit is developed from the 5 millimeter of the flower to this type of a fruit, and the full of oil content. And this is Stapletonea arunachalensis, one of the bamboo species which can be used the food also. Delausia bracteata, the plants are used, the flower petals are used for the traditional use, and also begonia, one of the most commercially utilized plant in the USA. More than $50 million plant, the $50 million business they do in the floriculture, and particularly in the begonia. Many of the begonia species which is found in the European market or the American market, it's not, though it has been taken from the wild from India, it is not found wild in India. You can take the example of the begonia berkeley or the begonia tassiocarpa, like that. So that's why there is a great opportunity for this, for the collaboration. And being Botanical Survey of India, we also work on the economic resources of the economic resources of the country. And we will see that the many of the species, the particularly Eastern Himalayan species or Western Himalayan species, if you see the Western Himalayan species, we have a great collections of prunas, sorbus, rubus, or ripes, or particularly carom, caesar, or cucumis. This is the center of secondary center of domestication or secondary center of diversification of the wild species. While the particularly in the Eastern Himalaya, we work on the genre of the different genus of the Musa or Eliocarpus, or you can see the Vigna, Trichojanthis, that is one of the most 13 species of Trichojanthis is found only in the Sikkim, and 18 species of Trichojanthis is found in the Arunachal Pradesh. So that's why there is a great opportunity for the agricultural scientists to work together and prioritizing the collections and the explorations, because we have some member of understanding with NPBGIR and also ICAR for the collection purpose particularly, because there is the particularly when they work for the livelihood mission, though we need to explore the efficacies of the wild plants. That is one of the most important thing. And this is the ripes. This is one of the gooseberries. This is the least utilized plant of the country, because the one of the most many of the species are edible, and many of the species has, though some of the species has been gone to the agriculture, but in the low profile. So that's why we have to see that how these natural resources of the country can be utilized properly. This is again another species. This is blackberries, that is known as the rubus. The rubus, as I told you, we have recently completed one of the revisionist studies of rubus. When we started, it was only 29 or around 30 species were described from the rubus, and it is only the reference was the Hucus flora from 100 years back. When we worked it out, around 180 species of the rubus has been documented, and out of the 80 species of rubus, this is around 30 species of rubus species are being used as a food material. And that's why there is a great opportunities for that. It must be come into the cultivation, and the particular gene has to be brought out, and it may also be hybridized with the strawberries or the gooseberries, so that the local farmers can be utilized, or the different kind of the forms of the not only the livelihood, monetary gain also be there, and also the food security can be assessed. And this is the rubus paniculatus. If you will go to the Arunachal, any area around subtropical region, the area is full of like weed plants only, and the food is very tasty and also edible. And this is the you can see the variety of the fruits which is edible, and these fruits has to be brought out to the commercialization or agriculture now. This is another plant known as the Codonopsis. The Codonopsis is known as this is the poor man's ginseng. This species is present all the characters of the ginseng particularly, but this is also not been utilized. Out of this, the particular Codonopsis species is the secondary the part of the speciation of this area. Out of 19 species throughout the world, we have around 18 species of Codonopsis only in the Eastern Himalaya. Only one species of Codonopsis is found in the Western Himalaya. Otherwise, all the 17 species are found in the Eastern Himalaya. That's why there is also a great scope for this one. And this is also again Codonopsis for some of the species of Codonopsis, and the root of the Codonopsis is used as the aphrodisiacs, or this is used as the part of the ginseng like that. And gingivers, I need not to say about the gingivers, the potency of the gingivers. Hedychiums, this is one of the most fragrant species of the family. Around 69 species is found itself in the Indian Himalayan region. Around the Larsinanthera, this is another new genus what we have recently worked out. And this is also having a potential different kind of the genes or the different kind of the fragrance material which can also be hybridized. And also Alpinia and Curcuma and also Gluba. This is another potential genus where we can work, because this is around 250 species. Out of 250 species of this gingival members are found. This is around more than 70 species is found in the Indian Himalayan regions. And this is a hippopy. I didn't want to talk about the hippopy. This is one of the most healthy tonics nowadays in the Western Himalayan. But unfortunately, the species all this has been collected from the wild, and it has to be come to the cultivation so that the more and more people can be benefited, and more money the commercialization can be done. And we have, as I told, we have the proud honor of the around 11 botanical gardens throughout the country. And we have more than 7,050 species around the live germplasm of the country. We are conserving on that, starting from the rhododendron to gingiber, and all the tree species, and all the edible and fruit species, wild edible fruit species. And the largest garden, we have the Agency Bose garden in the Havra. We have 110 acres of land. And we have also a garden in Yarkau. There the orchidarium, the National Orchidarium is present. The Eastern Regional Center of Barpani. We have the medicinal plant garden over there. But the problem is that when we talk about all these, the vulnerability is one of the most important things. The vulnerability due to climate change, or vulnerability due to any of the drivers, what we talk about, the proximals or maybe ultimate, all the global drivers. When we talk about the proximal drivers, this affects the local impacts, the forest fires, or the extension of agricultural lands, or the introduction of the monoculture of the many of the things. The Caryota, some of the species, this has been introduced in the Himalayan ecosystem, which has been the monoculturally they produce that. And this has a great adverse impact on that. And recently, we have also worked on that in the climate change things. Today morning also, I was speaking to Professor Mahapatraji, and there he was asking that there is a difference of 2.3 degree of temperature difference from the last 1 decade in the Himalayan, particularly Himalayan region. So that's why when we talk about this, then the climate change, when the climate change occurs, then 3 things basically it happens to a plant community. Either it adapts to the plant to the new regime, that is the selective plasticity of the plant, or this is make a migration to the higher zones, that is a shifting of the particular species to a higher zone, or it may lose. It extinct from the ecosystem because of the different kind of impact on the climate change. So that's why when the past exploration or past opportunities were there, so we usually focus on the broad range of species. Now the focus has been changed. Now we have to talk about the prioritizing the species, when and how to collect the species. Because unless we don't collect these species or the wild species, one day it will come definitely these species will extinct, and many of the novel genes or many of the important genes may also lose. So then what are the greatest challenges for this one when we talk about when we amalgam between the agricultural scientist or horticultural scientist or a pure taxonomic scientist? This is the lack of the comprehensive knowledge of the species concept of the plant. Because what the people is talking about the variety, or what the people are talking about the subspecies level, it may not be the subspecies level, it may be ecotypic variation. So that's why the most important thing is particularly the scientists of the agriculture or the novel genes. If the taxonomy also be amalgamated with this pure science of the applied science, the more and more positive result can become. And the another thing is that expertise in the plant taxonomy. This is one of the vanishing subject nowadays in the country, and not in the country, throughout the world. The problem is that particularly though we work on a lot of the things in the applied subject, because sometimes we fail to identify a particular species for which the entire experiment depends. So that's why the most important thing is knowledge of the plant taxonomy is one of the important things. And as you know that many of the universities has also deleted the particular basic taxonomy thing. And people now a botany, and they are not able to understand, they are not able to identify a simple plant on which they are working. Because many times you work on the plant part or flower or a leaf. That's why the most important thing is imparting of this particular science, to plant taxonomy, the basic things is one of the most important thing. And also the knowledge regarding the distribution of the plant. Because we have the Botanical Survey of India working throughout the country. We have a great data of the plants, what are the plants is found in the country. So we have exact distribution points of the plant and where these plants are found. So that's why it will also, the collaboration will also helpful to the other people to get and to give the flower. This is recently, I am just going to give an example. This is one of our scientists is working on the Paris polyphylla. And Paris polyphylla is found, although it is found in the higher altitudes of the Himalaya, but the exact location is most important. And when we surveyed the exact locations and we found that this is not only what the talking about the Paris polyphylla, what we are thinking of the Paris polyphylla, this is a mixture of the big complex. These are different kind of species are found there. So that's why that is the most important. When we are doing on the experiment, this is the different kind. The identification of the particular species is one of the most important thing. And fund constraints, this is unknown. This is not new to everybody. The fund constraint is one of the most challenging for this one. And loss of genetic diversity, because due to the natural and also in the man-made problems, a lot of genetic diversity is going up and destroying. That's why we need to collaborate for this one, for this. And when we talk about the relationship between the climate change, and this is the relationship between the different kind of the species found in the natural habitat, some of the indicators things we need to take. That is maybe a phenology or maybe a community of the complete component of the particular ecosystem, or the prediction modelings of different kind of in what will happen to the 3 years or 30 years or 50 years of time. And if you see that the species composition, one of the most important thing, because any of the change in the climatic condition, you take the example of the saprophytic orchids. The saprophytic orchids are basically, these are dependable on the microclimatic condition of the particular plant, microclimatic condition of the particular ecosystem. When the temperature changes, when the climate changes, or the rainfall changes, the conducive atmosphere or conducive microclimatic of that ecosystem loses. So that's why it may not flower, and maybe the particular saprophytic orchids, or it may shift to the other places. Or a change in the life cycle. If the change in the life cycle, as I was talking, the shifting of the altitudinal shifting, then take the example of the Paphlopadium. Paphlopadium is one of the orchid which is found only in the particular locations of the Himalaya. So if any changes that happens there, so if the ecosystem level destroys on that, then the life cycle destroys, automatically the life cycle will change. Many of the pollinator will not come, and many of the associated mechanism of the life cycles will lose, and change of the abundance, that is one of the thing. That's why the prioritizing, now we need to prioritize the plant exploration. When we talk about the prioritizing plant exploration, it may be a potential for the medicinal plants or the economy, which is directly converted to the economy, which is directed to come to the SDG goals of the country. Or the taxonomically uniquely isolated. This is rare and narrowly the endemic species, insufficiently represented in the species or diseases of high priority. Very narrowly. If you will take the example of the Pleonis scarflorum, this is only found in the single locality of Barsay Hills of the Arunachal Pradesh. If you will take the example of any of the Paphlopadium, you take the example of Panax pseudo-ginseng. The Panax pseudo-ginseng is particularly found in the limited altitudinal variations. That's why those areas, those things has to be taken care first. And the superior plants. When we talk of the superior plant, this is a very cold hardiness or the tolerance level of the stressful conditions. You can take many of the examples of the high cold desert plants. The cold desert plants are the very much cold hardiness, and those plants has to be referred, and those genes has to be preserved. And samples for the multiple population. That is one of the thing. When we work on any of the experiment things, we need to work on the different kind of the populations or different kind of the ecosystem. It may be, you take the example of the Polygonum nepalensis. This found in the one of the, which is used particularly in orthotitis. And this is found in the 200 altitude to 4,500 meter of altitude. That's why if we'll have a different kind of the different kind of the samples from the different kind of the habitat, then this will help us for getting the new novelties or getting the new ideas or new results. And prioritizing species in both the species and the geography, that is most important thing. And insufficiently represented taxa, which has not been represented properly. Maybe it has been collected in 100 years, 150 years ago, and now we are not getting any species for that. So if there are new species for that or new discoveries of those things, then it will have a great importance for that. And this is some of the species which we have worked on, this Parthenium. this is one of the invasive species, which is we have predicted for the next 70 years, and how these species will be found in the eastern, this is the Indian Himalayan region. If another species, this is Chloroma odorata. This we have worked in the Mizoram, and we have predicted for the three RCPs, that is in 4.5 and 8.5, and in the how the scenario of this area in the 1930, the 2030, 2050, and 2070. And we found that if the same condition prevails, then it will increase, and it will one day it will cover up everything, all the natural forest of the country. That is the most important, because now it is gone up to the 2,500 meter of height, and if the same trend continues, it may have the adverse effect in the natural forest. And the current issues and challenges, because we work on the field, actually, in fact, we got some of the challenges in the field itself. This is the most challenging is the getting the permissions to the entry into the restricted areas. And maybe the government have the different policies, and particularly those people who work in the particularly in the ground, they didn't get easily permission to enter into the wildlife sanctuaries, biosphere reserves. That is one of the most things. Though we have a technique to the highest extent to the ministry, though we are getting being Botanical Survey of India, we get the permissions. Many of the researchers of the particular taxonomy, they don't get it. And need for the consortium, that is one of the most important thing, because we work in, sometimes we work in the very silos. And unless you don't work together and collaboratively, many of the results which is supposed to be come up with a collaborative result, it may not come. It may come in isolated ways. That is one of the thing. And emphasize in collecting on the red listed plants. This is one of the things. The red list plant is one of the most important things, that is the threatened species. And because we need to work in the red listed plants, and what are the importance and what are the different kind of the components is present on that. And simultaneously, we need to work on the conserving the ex situ and also for the in situ conservation. And the multi-species versus the single species. As I already mentioned that, if the one species has been used for the different kind of diseases, if different kind of species are used for the particular diseases, then all the group of plants has to be collected, and all the group of plants has to be studied properly. And the single, because this is debatable topic on that, whether the single genus or the few genera or the wider genetic diversity. Because when we work on the particular species, and we not to restrict ourselves to collect this particular species from a particular region. Because suppose you take the example of the Dioscorea of the complete Himalaya, you will see that the same Dioscorea pentaphyla is having a different kind of constitution and different kind of alkaloid content. So that's why that is the important thing. We have a variety of collections for this one, and a wide range of collections for this. And when we talk about the way forwards, we need to signify the importance of the systematic explorations and the collection. And because we need the novel genes and the availability of the sophisticated biotechnology, these tools is come up with the biotechnology, and also the breaking wide hybridization barriers. When we have able to do the hybridization and break the hybridization barrier, it is very much wiseful and very much useful for work on the wild species versus the cultivated species. That is one of the most thing. And problems of species concept. And recently we have been published the entire checklist of the country. And this book is also available in our stall, which is present in the exhibition ground. So the species concept, what the problem is that species concept we haven't changed, because that was a long pending aspiration of the country for having a working checklist of the country. And because after the CVD 92, the country was committed to bring up the working checklist of the wild plants, and the working checklist of the wild plant has already been done. And also these are also available in our website, the entire checklist of the plant. And till now we have recorded around 20,700 odd plants, and the entire checklist is found, so that the species concept has been changed, and we have given the exact accepted name of the particular species, wild species, and also all the relevant synonyms for that. And we also need to the gap existence between the CWR genetic resources, that is the crop wild resource relative, that unless we sit together and work together or collaborate together, this cannot be done. And that's why we invite all the agricultural scientists to have a look into that, And so the wild plant resources can be amalgamated with these research, their own researchers, so that it can be useful for the human mankind. And prioritizing conservation, we have also, as I told, we prioritizing conservation. We are committed to have around 60% of the total threatened plants of the country has to be conserved in a botanic garden, and for which the BSI, being a government agency, is committed for that, and we are working on that. And more than around 50% of the threatened plants has already been conserved in our own gardens. And except that, because we have MOU particularly with ICAR and NPBGR, we work with them for the collection of the wild resources for these seed banks, gene banks, and in vitro collections for them, and botanic garden collections for us. And my ministry also gives one of the flagship program, that is assistance to botanic garden, where we also fund to the different kind of organization and different kind of universities for developing of gardens, and also conserving other plants in the botanic gardens. And when we talk up way forward, this is one of the most important things what we stressed upon that, how to balance the need of the access to the genetic resources and also the benefit sharing, because we also work on the traditional use of the plants from the Northeast and Himalaya region. And unless the end of point is not that the collection of the plants and the use of the traditional knowledge, but the end game is that when we bring it into the commercialization and also benefit to the particular community, that is the most important thing. And the possibility of collaboration, all the great like-minded people has to come together and sit with each other, and they have to track out the different kind of the collaborations. We have different kind of collaborations of the NPPGR. We have some of the collaboration of development of this is wild plant resources and also wild edible plants. We have a collaboration with DRDO, where we are giving them the complete list of the wild edible leaves of the country. And we have a project also with the DRDO, where we are asked to give them the wild edible fruits of the Northeast states. So that's why the like-minded people has to come together and for a great challenge, to face this great challenge, for do the greater power, for greater things for the natural mankind. And thank you very much.
Anyone up for any question?
Sir, thank you for the elaborative lecture. Actually, Botanical Survey of India is doing really great job. And my question is that you are serving the usually forest regions, is it?
Yes.
But what's about rural areas? Because for example, I'm telling, in my village, I have seen in my childhood number of medicinal plants are there, different varieties, types of plants are there. But now they all are replaced with either acacia or eucalyptus.
No, sir. Actually, you see, we have been designed to work with the wild resources of the country. But we have projects like that, the All India Coordinated Projects for the Ethnobiology. On that project, we have already surveyed the entire the Northeast and also Himalayan state for regarding the medicinal plant, wild edible plant, wild what we call this is wild edible leaves. Because you see, as I told you, DRDO has given an assignment to us to bring out the wild edible leaves of the country, only wild edible leaves. So, because although many people or many institutes have also been assigned those works, but the authenticity of the BSI, what we are giving is the pure. Because we want particularly in this ethnobiology study, we have given around 3,500. Already the uses and also pictures. So now the next step comes to the scientist of agriculture, how that can be used. Suppose you take the example of Dioscorea, what we see in the Dioscorea. There are 21 species of Dioscorea has been used. So those, we have given the identity of the plant, the photograph of that plant, from where this plant has been collected, we have given, and also what is the quantity and what is the distribution of that area, that also we have given. So like that, we also survey in the rural areas, not only the inside the cultivated plant, but only the wild resources of the country. Thank you. Because the cultivated is been assigned to the NPBGR, they are working, and recently we had a MOU with them. So wherever we are going, one of the scientists from the NPBGR is accompanying with us, or whenever they go, one of the scientists of BSI accompany with them, so that it can be worked out thoroughly.
So there's one more question from there, the back. Okay.
Thank you. Very insightful presentation. I just have a simple question. I just wanted to know, like you say, there is a lot of species available for cultivation. So my first question was, for example, for the solanum, you say that there is 51 species, but according to you, how many are actually edible? And among all the species that you have identified are actually edible that we can consume and cultivate for commercial purpose?
Ma'am, in fact, around 58 species of particular solanum plants are found in the country. Out of the 58 plants, around 22 species has been edible. Different kind of the wild, I'm talking about the wild plants. And which the plants I'm talking about, these are the wild cultivars, actually, wild relative plants, actually. Unless these are some of the genes or some of the, if you can hybridize with some of the species, then maybe some drought resistance will be come up. My only intention is to show that this is the wild relative plants, and these wild relative plants, if can be crossed out, you can take the example of the saccharum. And when it been the saccharum and your sugar cane, and when it has been the gene which has resistance to the particular disease has been hybridized with the saccharum, the regulation has been changed. That's why this is right now to identify the gene, and unless you don't study and don't see the genes, what is the drought resistance or what is the particularly disease resistance, nothing can be done.
Okay. Thank you so much, sir.
Thank you, ma'am. Thank you very much.
So we have two more sessions for this time, but this particular sort. But what we'll do is we'll have the lunch first. That will be and we'll resume the session sharp at 2:30. And the sessions which we could not do now, we will do it after lunch. If any of you want to give your talk to us, so please, you can give it to us now.
Hello. Hello.
Okay, due to lack of time, we'll start the presentation that we left early. So for now, we'll request Professor Samrendra Mahapatra, former professor and head of Agribusiness OUAT Bhubaneswar, to please proceed with his plenary session. His topic is emerging trends in agricultural marketing, issues and challenges. So can we please have you here?
This spot is coming. Thank you. So good afternoon, all those who are present. I thank the organizing committee of Agri Vision for giving me an opportunity to present my paper. So I'll be giving a brief idea about the emerging trends in agricultural marketing in India. What are the issues and challenges? So with respect to so far agriculture in India is concerned, we have moved from deficit oriented to surplus oriented as a transformation. And also, as you know, we have shifted from the basic agriculture, that is growing food grains, to commercial cash crops. And also we have shifted to the high value agriculture, especially the horti business that includes fruits, vegetables, floriculture, the dairy sector, dairy and value added products of dairy, then fisheries sector, and also poultry, etc. These are high value agriculture sector. Especially after 90s, when we have seen the changes came with respect to the liberalization and globalization in India. It has totally changed the market scenario and environment. So with respect to the corporate's entry into agribusiness model, as you know, the major Indian corporate houses like Mahindra and Mahindra, Birlas, Tata's, Reliance, they have entered into agribusiness models. And as they found it is highly potential, and the commercialization of agriculture sector with corporate farming and tie up with farmers. With respect to agriculture exports, if we see, because of the bilateral and regional trade agreements, there is a reduction in the tariff and non-tariff barriers, which has facilitated for free flow of goods between the countries across the nations in the world.
and that is supported the international trade in agribusiness. We have witnessed the changes of the LPG era, that is the market-driven economy. And also with the introduction, starting of the agri-commodity exchanges, especially the National Spot Exchange, we started in 2005, as well as the major commodity exchanges, which gives a platform for transacting valuable products, including agri-commodities, especially NCDEX, National Commodity and Derivative Exchange, the multi-commodity exchanges, which it gives a platform to the producing farmers as well as the registered traders and vendors to transact the listed agri-commodities, which brings a transparency in the, and also it ensures profitability to both the stakeholders. If you see the renewable energy applications in the field of agriculture, we have seen the benefits we have seen the benefits of solar energy, biomass energy, and wind energy applied in the field of agriculture and allied sectors, especially for energizing the remote rural regions, villages with power source, with the pump sets operated by solar energy. Then also it is water heating system, etc. A lot of devices are being energized through the solar energy photovoltaic cell system. And that is totally environmental friendly source and is alternate source of energy, which is very much viable with the entry of various agencies and organizations. So they have got every state, they have got state level renewable energy development agency, including one at the central level. So with respect to the ICT in agriculture, we have seen the transformational model, that is the ITCE Chopal, with the ITC has introduced, introducing lot of kiosks in the villages for the farmers with required training so that they can use and get the market informations. Besides, there are some other websites like iKishan, like krishibihar.com, agriwatch.com, etc. also gives lot of market information, which helps the stakeholders to take the data-driven decision making. With respect to the agriculture marketing process, if you see, so as we know that around 85% of the farmers in India, they belong to small and marginal farmers category with less than 2 acres of farmland. So though the individual wise, when it is assembled, it gets in a smaller quantity, but ultimately with the help of the farmer interest group, when the farmers are joining hands together, it is consolidated to a higher quantity, which is feasible for transacting with the institutional buyers. And so far the grading and standardization is concerned, we know that the grading of the food grains is carried out, and also grading of agri produce is carried out in general with respect to the size, color, and utility. So the value also differs. The market value, market price based on the grading, which fetches a higher value to the producing farmers. So sorting grading is carried out at the marketplaces, especially in the regulated market committee, the RMC, which is operated by the state level marketing boards, agriculture marketing boards. So with respect to processing, if you see, value addition is very essential in order to fetch a higher market value for the agri commodities when the raw agricultural products are processed in primary, secondary, and tertiary level with a highly converting into highly value added agri products, which is fetching a high market value, high market demand, and highly profitable also for the producers. With respect to the storage, if you see, because the surplus of agri commodities need to be stored in a controlled temperature, especially majority of the agri products are perishable and also highly perishable. So they need to be stored under the controlled environment, controlled temperature and humidity. And those support is provided by major organization is the Food Corporation of India, and also the cold storages that is operated on cooperative basis or with the support of corporates, and also some with the initiative of the farmer producer organizations in India. So with respect to the transportation part in marketing, as you know, the agri commodities need to be transported from the farmhouse or farm gate to the nearest marketplace. So proper connectivity, road conditions and transport arrangement need to be provided. Cost-effective transportation mode need to be provided to the farmers. And with respect to the distribution, physical distribution, so because the farmers always wants that how to dispose the perishable agri commodities timely. So it needs proper arrangement for physical distribution so that it is also available for the consumers at the different retail outlets platforms. So it is a win-win solution for producer as well as the consumers. With respect to the opportunities, if you see, first, we have seen that the global trading that is happening because of the changes that has taken place in India after 90s with the support of the apex body, that is the World Trade Organization, WTO, when the bilateral and regional trade agreements taking place between the member nations, which provides free trade of commodities, including agri commodities across the international borders, reducing. It also helps to reduce the tariff and non-tariff barriers to facilitate the stakeholders in the process. With respect to the advanced level of processing, we have seen the benefits of converting the farm. It is the converting the raw agri produce into highly value-added agri commodity. So when there is a change in the form, there is a change in the demand and change in the value and price, resulting in high profits for the producers. With respect to the livestock resources, we have seen the demand for high value sectors like dairy products, like fishery products and poultry products, etc. is catching a very good demand in the marketplace and also very profitable for the producers. With respect to the biotechnology application, we have seen in the case of seed production, also in the case of the bakery industries, so biocontrol agents are also developed. And with respect to the organic farming, if we see, there is a concern for the health oriented among the consumers in general. So they are always looking for healthy food, healthy diet, and organic. When it is a question of organic farming, we need to take care of the right from inception while using the inputs instead of external chemical inputs. More focus should be given for using the organic inputs, especially the vermicompost and the like the locally available cow dung and etc. The organic inputs which is available free of cost instead of spending high amount for purchasing the external chemical inputs. So far, the water and labor use efficiency is concerned. Now the trend has changed. How to use water efficiently with the support of micro irrigation system, that is sprinklers and the drip irrigation, to save the water and use the available limited water efficiently for the crop. And also we have to take care of the labor use efficiency with the help of mechanized farming and agriculture. As you know, there is lot of improvement have taken place with tractor-driven operation and combined harvesters and all, which does the harvesting activity very fast, and also timely harvesting is feasible with the support of mechanized farming. If you see the scope of employment, when there is an increase in production, it also leads to increase in employment opportunity because of the marketing opportunity for the produce, transportation opportunity, logistics, etc. So in every field, there is an increase requirement of the manpower in the rural areas, which is a source of employment generation in the villages. If you see the challenges in agriculture marketing, we have seen the variation in the marketing fees. So the marketing fee, which is chargeable as per the APMC Act, it is actually varies from 0.5 to 2%. If you see the state to state also, there is variations, which can only help you if the fees can be standardized and made it uniform instead of variation in the marketing fees charged to the traders and producers. Are the RMC market yard, regulated market committee yards governed by APMC Act and also monitored by the state level agriculture marketing boards. With respect to the rural harts, we have seen that it is the periodic hart taking place in the villages where there is lot of problem with respect to providing the basic amenities or the infrastructure facilities for transacting the agri and non-agri products directly to the consumers. The producers with interface directly to the consumers in the periodic rural harts. And if you see the standardization, there are different set of standards. The standards set by the AG mark, that is a standard for the food products. There is also standards set by the Food Corporation of India. So there are various organizations for standardizing the products in different approach and criteria, which creates a confusion that need to be considered. And with respect to Essential Commodities Act, so the central government has already removed regarding the storage and transportation restrictions, whereas some of the state governments still, they are enforcing the same to the stakeholders, which creates a problem. And if you see the infrastructure facilities, many of the areas, they don't have the good road conditions, power supply or water supply, and they don't have the facilities like the weighing machine, sorting grading machine. So these are very much essential for developing the market infrastructure. With respect to the digital platforms and e-markets, if you see the blockchain technology, which is very much helpful in tracking and tracing the products. That is how to ensure knowing the origin source of the production, which relates to authenticity of the produce, and also authenticity, whether it is organic or not organic, can be known. And if you see the concept of precision agriculture, it is nothing but the data-driven agriculture, and it relates to how to optimize the resources, how to ensure timely harvesting, and other farm-related activities can be done with precisely, not only saving time and money, but also it increases the efficiency and productivity of the farm operations. If you see the agri-tech startups, it facilitates the farmers through the apps to knowing the information readily available through the apps. And once they are acquainted, they can know the information, timely information about the market prices of various commodities, which will help to take the decisions accordingly to benefit the stakeholders, both linking the buyers and sellers. If you see the smart packaging and labeling, which has been carried out with RFID and barcodes, so that you can scan and can know the details, which is informations in detail, which is very much required by the consumers. With respect to the concept and approach of sustainable practices, so throughout the process or the supply chain, we have to ensure during while sourcing the inputs, using the inputs, while during the crop production, during the harvesting, logistics, and also distribution and marketing, in entire chain, how to make the activities eco-friendly without affecting the environment in any manner. If you see the social media marketing, it is very much popular to spread the message of success story of the producers, also to to improve their brand image, all the related information, since it is widely used, social media. That can be also one of the areas of focus. If you see the government initiatives, the government of India has given lot of support in providing incentive and subsidies to the beneficiary farmers through DBT, direct benefit cash transfer. Also, they encourage to empower the 85% of the small and marginal farmers of India by formation of more and more of farmer producer organizations in general, and also farmer producer companies in specific, where they can deal in bulk quantity with the institutional buyers and can be benefited. And the government also supports through introducing various schemes and programs, and thereby encouraging agri-entrepreneurship and agri startups. With respect to the vertical farming and agriculture, as you know, it is a source of getting fresh agri-produce in the urban towns and cities with a limited space. If you see the application of artificial intelligence and predictive analytics, it helps the farmers in knowing the details regarding the pest and disease attack, and also timely estimation of the demand for the products. All can be known with the use, provided they should know the application, how to use it, how to get the benefit out of this technology. If you see the development of initiatives in agriculture marketing, we have seen the Kisan Rath mobile app, which is also helps the stakeholders, especially the farmers, producing farmers, in areas of logistics and other support. If you see the eNAM call center implementation, there are certain discrepancies, certain problems like the inadequate infrastructure and amenities to provide for transaction, and even storing the items under controlled temperature. That is the need for cold storage and cold chain facilities. Expansion of eNAM platform has already carried out in 193 for 193 commodities in India. But still there are challenges and issues in eNAM implementation. So far, eNAM is concerned, 2016, it is started, and also the stakeholders like the producing farmers traders, commission agents, all are being benefited in the process. And there is also, it will also benefit if all the RMCRs can be connected centrally to the eNAM portal, that is Online National Agriculture Market Portal. The guidelines for the farmers and traders are in eNAM is the sorting, grading of infrastructure, quick settlement of online transactions, inter-market trade between the mandis, awareness and orientation programs for the stakeholders, logistics and infrastructure availability, transparency and accountability in the implementation, better internet connectivity, as well as regular meetings of the APMC officials. Those were monitored by the state level agriculture marketing board. If you see the concept of empowering the small and marginal farmers through farmer producer organizations in general or farmer producer companies in specific, that is a non-profit organization. And it is, you can say it is a hybrid structure of cooperative and corporate, taking the benefit of both, where the farmers are the members of the organization and they will take interest how to, for the growth of the organization and for their benefit by involving themselves in various activities, commercial and activities across the entire agri supply chain. And but what is needed actually, they need to be trained. So the capacity building aspect has to be taken care, which is already being supported by the national level organizations like NABARD, NCDC, National Cooperative Development Corporation, then NAFED, National Cooperative Agriculture Marketing Federation, as well as NDDB, National Dairy Development Board for the milk farmers. So they are already there, including SFAC, Small Farmers Agri Business Consortium, for providing the necessary seed capital for the farmer producer companies. So if you see the recent trends in agriculture marketing, since India is one of the largest population country, obviously food for all, there is a growing demand for food production. Hence, there is an opportunity for large scale production and organized initiatives like the developing the entire distribution network and channels for providing timely agri commodities in all the regions across the wide geographical area, and state-wise can be monitored. And also another area, that is investment in infrastructure development. Government of India has already during 2022 invested a huge amount for developing the agriculture infrastructure, which is a great support to the farming community for all their activities, production, processing, and marketing. With respect to government policy and regulation, which is needed is favorable and supportive government policy, which will help the farmers and the other stakeholders related to agri-allied sectors so that they can take benefits out of that and they can continue in agriculture farming profession. What is happening is there is a switchover. There is many of the farmers, they are switching from primary to secondary and tertiary sector only because they found it is not profitable as compared to secondary and tertiary sector. If you see the ICT application in agriculture marketing, we have seen the benefits of these websites and also ITC, eChopal and others, those who are providing the information with real-time value of agriculture commodities across the country, across India, so that they can take the decisions, right decisions accordingly to benefit them. With respect to Regulate Market Committee, if you see, there are number of regulated marketing committee which is operating in every state under the which is monitored by the state level agriculture marketing board, and the related acts which helps the Essential Commodities Act and the Agriculture Produce Market Committee Act and Model APMC Act, which is the revised one, which is now operating, to ensure to provide fair price to the stakeholder, especially to the farmers, and also they should ensure that consumers should get the products at affordable price. Should be a win-win situation among the stakeholders in the business. With respect to the quality control aspect, as you know, quality, every consumers are very much quality conscious. They always expect quality agri commodities. There must be quality control checks by the different agencies and especially government side, and they can ensure that quality right from inception, from the seed stage itself, input stage, they should ensure quality production with the use of quality inputs and also good agricultural practices. Then market infrastructure can be enhanced with the investment of the government, because government has given a huge investment plan year-wise to support the agri-allied sector. If there is any dispute arises among the stakeholders, that is amicably resolved with the dispute settlement mechanism set up by the state level agriculture marketing boards. And with respect to information dissemination, as you know, timely information is the power source of, and should be reliable information. Organized database should be available for agri-allied sectors, which can be retrieved as and when required in order to decide and prepare the meaningful reports for decision making. Risk mitigation is also one important aspect, how to mitigate the risk in production, processing, and pricing, market risk, and all the areas. So risk is involved in the agriculture entire chain. So how to mitigate the risk with different approaches. As you know, production risk with the support of the insurance, agri-insurance, and the price risk can be supported with the contract farming, and also with the agri-commodity exchanges, which gives a fair price, and also the win-win situation between the buyer and seller. If you see agri-commodity exchanges, it is a diverse commodity trading. As I told you, the National Spot Exchange started in 2005. Then NCDEX, National Commodity Derivative Exchange, as well as Multi-Commodity Exchange, started in 2003 in Mumbai as its headquarters with their zonal outlets in India. If you see the price discovery system, it is very important, which the platform, agri-commodity exchange platform, provides the valuable real life, the price information for taking informed decisions by the stakeholders for transaction of different agri commodities between the registered buyers and registered sellers. So risk management can be an important factor for the farmers, which can be obviously prevented to greater extent by participating in the agri commodity exchange, because they can fix up the price for the future trading also, in addition to present transactions, spot transaction. With respect to market integrity, there has to be fair transactions, and with transparency, clarity should be there, ensured by this platform, by these electronic trading platforms, where the parties can join from any part of the country. And also the agri-commodity exchange, they tie up for storage and warehousing facilities for the store of the safe storage of the perishable agri-commodities, with the support of the institutional members. And a lot of support is extended to these stakeholders. And the market informations is a very important support by participating the agri-commodity exchanges. And government, they also link with the government as per the government guidelines they are operating. And some of the agri-commodity exchanges, they have also tie up with the global commodity exchange marketplaces for export of the agri-commodity, that is leading to international trade in agriculture. If you see the APO principle, it is open membership with democratic control, with the economic participation. They have got autonomy and independence, education, with the training and information provided to them, with the and cooperation among the farmer producer organization, and they work for the community development. And also they are getting benefited. In a concluding remark, I can say the group approach is very much supportive for sustainable solutions like the FPO and the collective efforts. The appropriate government policy needed for an efficient marketing system, positive impact of the recent trends as agro-based industries, and also new technology, modernization, direct dealing with the consumers, interface with the consumers, diversification, crop diversification, digitization, global trade dynamics, increase in customer focus, multi-channel marketing, public demonstrations, resource management, strategic partnership, then sustainable agriculture, visual branding, and water management. These are the important aspects which need to be considered in order to improve the overall efficiency of the agriculture system. Thank you.
So since we have couple of lectures added more to the session, we are refraining ourselves from asking any questions, but you can definitely discuss it personally. Now, I'll request Dr. Arlene C. Algray to please come on the podium and do her talk. Is she here? Okay. Dr. Golev Prashanti, ma'am, please join in. She is from ICAR-National Rice Research Institute, Cuttack only, the same place we are hosting this event at, exploring host plants, resistance and biochemical responses in ARC genotypes against the rice leaf folder is her topic of discussion. Also requesting, very humbly requesting all of you to please finish your talk within the given time limits. It's 15 minutes for all. Man, we'll finish this and then call you.
Good afternoon, respected chairman, co-chairman, rapporteurs, distinguished delegates from various institutes. Today, I am here to present on the topic exploring the host plant resistance and biochemical responses in Assam rice collection genotypes against the rice leaf folder, that is Nephlochrosis medinalis. As all of you know that rice is one of the most important staple food crop of more than half of the world population, and this crop is susceptible to various abiotic and biotic stresses. If you see the biotic stresses, this crop is affected by more than 20 insect pests. They are causing serious damage. Out of the 20 insect pests, 5 are causing national and 5 are having national and international significance. Those are stem borer, plant hoppers, gall maize, leaf folder, and remaining all causes 25% damage. If we see the paddy leaf folder alone, it causes 10 to 80% yield loss. And this pest is before 1980s, it was a minor pest. But after 1980s, it has started assuming the major pest status due to the use of high yielding varieties and modern cultivation practices. All these are one of the reasons. So still now we are relying on chemical insecticide for controlling this pest. These are some of the negative effects which are associated with the chemical insecticide, like residual effects, impacts on non-target organisms, and there is a development of resurgence and resistance. And this pest, if we see the resurgence, it has developed resurgence for especially for neonicotinoid insecticide like acetamiprid, imidacloprid, thiamethoxam. At present, we are using this pest already developed resurgence for them. And it is also showing resistance to certain insecticides like carbofuran and phorate granules. So in order to rationalize the pesticide use, host plant resistance is the best measure we can follow. So because this host plant resistance is economically, ecologically stable, and we can, it will be compatible with other integrated pest management tools. So for this study, we have chosen Assam rice collection genotypes. Because why we have chosen these genotypes? These are having high biodiversity and wider adaptability. They can adapt at many places of India, and their nutritional value also very high. And besides that, they are having cultural and culinary significance. Contains some of the special features like aroma, color, and their resistance to some of the diseases and pests also. So for conducting this study, it is conducted under mainly two objectives. That is like screening of these land rices under the field condition. And next one is analysis of the morphobiochemical traits of these varieties against rice leaf folder. So this was the methodology what we have followed. Total number of genotypes for this study, we have used 30 by using the susceptible check as TN1. TKM6 was the resistant check for this study. So after transplanting of these genotypes in the field level, we have covered the genotypes after 35 days of transplanting with a green net. We have chosen green net because it allows the 50% of sunlight inside. Almost for 1 week, we have covered the genotypes. During that period, we released mated leaf folder females, like 50 pairs of adults we have released inside the net. Our main motto of this releasing is to which genotype will be preferred by them. It means our basic screening is happening here. So after releasing of the mated females, after 1 week, we will remove the net and we will do the scoring by using these formulas based on the number of number of leaves per hill damaged by this pest. We will score by using the IRI CES scoring. 2000 recent scoring we are following. This was the actual field layout what we have followed. After every 10 test genotypes, we are planting one resistant check and one susceptible check. And the borders are planted with mainly susceptible checks. 2 rows we have used so that it is what kind of selection pressure it is giving, like that we can easily differentiate between the checks and our varieties. So these are the different genotypes we use, and based on the ADLR scoring, that is adjustable damaged leaf rating scoring, we found that these genotypes are coming under 3 different categories. Those are resistant, moderately resistant, and susceptible category. Among the resistant category, we have seen 7 genotypes. Moderately resistant 11 are falling, and remaining all are coming under susceptible category. So on these varieties, we have studied certain morphological characters like plant height, leaf length, leaf width, and trichome length and density also we have studied. For studying the trichome length and density, we have used scanning electron microscope. So these are our morphological parameters. We found that plant height, leaf length are which are negatively correlated with leaf folder damage, whereas leaf width, which is associated positively with leaf folder damage. So here you can see the trichome length and density. These are some of the scanning electron microscope pictures. So we found mainly two different kind of trichomes. Those are glandular and non-glandular. Non-glandular trichomes are macro, micro, papillae type and prickle type of trichomes. These are non-glandular trichomes, means they are ductless. Glandular trichomes, they are associated with a duct. So these non-glandular trichomes, how they will protect the plant? So what they do, they will act as a physical barrier so that leaf folder adult cannot come and lay eggs on the particular genotype. So these are all acting as physical barriers. Whereas glandular genotypes, these are associated with a duct so that they are associated with the releasing of volatile so that pest attraction towards the genotypes will be reduced. So in our genotypes, we found that in all the categories, we have seen more or less both glandular and non-glandular trichomes. But in resistant category, the density and length of trichomes were more when compared to moderately resistant and susceptible categories. Here, among these 30 genotypes, from all the genotypes, we have chosen nearly 10 genotypes for studying the biochemical parameters. So we have studied the biochemical parameters like total soluble sugar, soluble total phenol, soluble protein, peroxidase, polyphenol oxidase, and catalase we have studied. For this, we have used one control and 3 others are as replicates we have taken. So this was the data. We found that antioxidant enzymes which are negatively correlated with leaf folder damage, whereas the total soluble sugar and protein which are associated positively with the leaf folder damage. So these are the certain antioxidant enzymes like peroxidase, polyphenol oxidase, catalase, and phenol. This data we have studied both on uninsected and insected genotypes. Uninsected is our control. Insected is herbivore pressure-induced plants. So that what we found is after when herbivore attack is there is a increasing in the production of the enzymes like peroxidase, catalase, and polyphenol oxidase we have noticed. And in general also their level of increase is more in case of resistant genotypes when compared to susceptible check. Similarly, we have noticed with the secondary metabolite phenol also. And the other 2 biochemical factors like protein and total soluble sugar, their level is in generality is more in the susceptible genotypes. Whereas after the induction of herbivory, we have seen that their level of increase is high in case of susceptible plants when compared to resistant plants. So now coming to the conclusions, we found that trichome density is highly, we have seen in resistant genotypes when compared to the susceptible ones, whereas sugar and protein, those are high in case of susceptible genotypes, and the other antioxidant enzymes, those are stress busters. The level of enzyme activity we have seen more in case of resistant lines, and the total rate of increase also we have seen more in case of resistant lines. These are the future prospects, and these morphological parameters and trichome parameters. We found the data resistant lines. Those are helpful for the development of leaf folder resistant varieties, and these lines can be incorporated in the breeding programs and later on helpful for the development of effective pest management strategies. And further, we can extend our study for knowing the genomic loci, that is quantitative trait loci, which is responsible for the leaf folder resistance. We will study in the future. These are the things. Thank you.
Yes, ma'am. No, we are not taking any questions. Okay. Thank you so much, ma'am. Okay, there's a small request to the people who have already finished their presentation in this hall, or in fact sitting idle and listening to the other presentation. Some of you, can you please join in the second hall, Subarna Rekha Hall? The second hall. Well, simultaneously the other presentation are running down. Some of you are requested who have finished their presentation, or their presentation is not lined up in this hall. Please join us in the second hall. We want some audience over there as well. Now, Dr. Arlene C. Algre, ma'am, can we please have you on stage? She is from Philippines, Camarines Norte State College. And she's talking on alternative management of queen apple.
Thank you for. Good afternoon, everyone. Good afternoon. So, in behalf of the Camarines Norte State College, okay. In behalf of the Camarines Norte State College and the Queen Pineapple Research and Development Institute, I would like to thank the Agri Vision 2024 for allowing us to share with you our research. Our research is entitled, Alternative Management of Queen Pineapple Mealybug Wilt Disease in Camarines Norte, Philippines. I am Arlene Alegre. Good afternoon to everyone. Suban Handin. Okay. So, Camarines Norte is known for queen pineapple. It is widely grown in our place. The queen pineapple is the small and sweet pineapple. Now, you might also have that here, but in our province, that is very common, and it was introduced to our province in coconut farming for intercropping of coconut. And later on, it became favorable to most of the farmers because they were able to realize the potential of this crop. So queen pineapple became famous because of its sweetness, and probably it's the sweetest pineapple in the country. And also it is very typhoon resilient. For the information of everyone, our province is within the typhoon belt. And we always have typhoons. Every year we experience typhoons. And so because of pineapple, farmers would like to grow this crop because it is very resilient even if we have several typhoons within the year. And also the farmers were able to realize that it gives them 7.7% average annual growth rate of farm gate price, and that is for the advantage. And eventually they realized that the potential income source for 3,500 farmers and traders. And so just like any other industry, queen pineapple has also its own problems and concerns. One is the pineapple mealybug wilt disease. This disease is quite prevalent in our province, and it is being transferred by the pink pineapple mealybug, which is the Demyscoccus bebipes, or the PPMB. And there is a as of now, we don't have epidemiological procedure on the pineapple mealybug wilt disease management. Also, we don't have an available virucide to protect the queen pineapple from pineapple mealybug wilt disease. And because all of this, we also have experienced the unavailability of environmentally safe pesticide to control PPMB. So this all results to poor quality of queen pineapple, resulting to low return of investment. So previous studies, we will learn that we were able to evaluate some botanical extracts that includes Zinzipper officinale, Allium sativum, organic antipathogenic solution, and dishwashing liquid, these two commercial products. And then we were able to observe that pathway can eliminate or control most of the pathogens. And so in this study, we will be using this one as our main control for virus disease. Okay. For the general objective of study is to develop an synthetic management of queen pineapple wilt disease in Camarines Norte. And our objectives, specific objectives, are the following. Possess the incidence of wilt-like disease of queen pineapple, evaluate the production loss incurred from pineapple mealybug wilt disease infection, and develop a methodology of pineapple mealybug wilt disease management. Okay. For the assessment of mealybug wilt disease, we were able to identify 4 areas. These 4 areas are the most the biggest producer of queen pineapple in our place. And each of these municipalities or areas has 2 sites, one site for vegetative and flowering stage. And most of them, okay, there were infected queen pineapple plants were counted and recorded to get the % incidence of the disease. Second is the assessment of production loss incurred from pineapple mealybug wilt disease infection, in which we weigh the queen pineapple and PPMB count at different maturity levels of the fruit were recorded. So for third methodology, the pineapple mealybug wilt management using organic antipathogenic solution, the treatment, the soil was treated with 5% pathway 3 days before transplanting. In queen pineapple, we use the suckers, not the crowns. And for preventive treatments, we use 2% of pathway. And the first month, which is the early vegetative stage, is the first treatment after soil treatment, and following bi-monthly treatment to late vegetative stage, and monthly treatment for 10 to 10 months old, which is the flowering to harvesting stage. By the way, queen pineapple, we harvest after 14 months. Okay. So for the results and discussion, the pineapple mealybug wilt disease on queen pineapple infection is peak at fruiting stage, which gives 37 to 39%. And one site in Basu that recorded in the lowest infestation on both stages, which is 11% and 12%. And this in the picture you can see, these are the pineapples that are infected with pineapple wilt disease. They tend to color red, the leaves, and then eventually it turns to brown and die. But this one, you can see that there is a flower of pineapple, and the other one is a pineapple that is infested with mealybugs. So for the production loss incurred from the pineapple mealybug wilt disease infection, the infestation at different maturity levels of queen pineapple is showed here. And we were able to observe that the pink pineapple mealybug infestation is at peak during the early ripening stage of pineapple. There is a percent count of PPMB amounting to 43% to 75% in areas of San Lorenzo Ruiz, Labo, and San Vicente. Basud has the highest count of PPMB at stage 2, which is 51%. And so this is the one. So here you can see the queen pineapple fruits infested with PPMB at different stages. So on the effect of pink pineapple mealybug infestation on the size of the fruit, there is a percentage of small fruits amounting to 42%, 42 to 66%. And there is a high percentage of small fruits amounting to 42 to 66%. The medium-sized fruits is 15% to 25%, and the large size of queen pineapple is 6% to 18%. So down here, you will able to see the different size of harvested queen pineapple. And so these are the results of the sizes from PPMB infested plants. So the effect of PPMB infestation on the development of the fruit crown. We were able to observe that the PPMB, or the pink pineapple mealybug infected fruits, develop longer fruit crowns when infection occurs at later stage of growth. The small crowns develop when infection sets in at an early stage of growth. So you can see here at the bottom, the harvested queen pineapple fruit showing a varied size of crowns. And when this crown is oversized, that is not marketable. Also when the size of crown is too small or underdeveloped. So on the development of methodology for management of pineapple mealybug wilt disease, a small size was reduced from 62% to 38% after treatment of pineapple of pathway at 2% concentration. The medium size increased from 27% to 39%, and the large size from 8 to 18%. There is a small increase, however, in the production of extra large size from 1% to 3%. Another observation on PPMB infestation at different maturity levels of queen pineapple. The maturity level 3 to 5, there were little reduction of mealybug after treatment of 2% PA or pathway from 62 to to 13%. Okay, so also on the effect of pathway treatment on crown development, there was increase of crown development from 22 cm to 25 cm. So under here, you can see the harvested green pineapple fruits with varying crown size. On the response of pineapple mealybug wilt disease incidence on pathway treatment, the pineapple mealybug wilt disease incidence using 2% treatment from 40% to 8% at flowering stage. And there was a 33% of PPMB incidence at 2% treatment. The reduction of off-size crown development of 2% pathway treatment. So in here, we were able to show you the return of investment of PPMB infected with different pathway concentration treatment. So at pathway concentrations here, 0, 1, and 2, you can see that at 2%, there was a tremendous increase of ROI and a reduction of pink pineapple mealybug count on pineapples. For conclusion, the incidence of pink pineapple mealybug wilt disease in Camarines Norte is at an alarming stage, considering the incidence has a range of 11% to 39%, resulting to production of small fruits ranging from 42% to 66% of total harvest that commands the lowest farm gate price and usually the traders' basis for pricing. The application of pathway organic antipathogenic solution reduced the production of small-sized fruits from 61% to 38%, increased production of medium-sized pineapples from 27 to 39%, and large-sized fruit production to double from 9 to 18%, and also increased the return of investment from 43 to 71% due to increase in volume of marketable fruits. The pathway claimed to be successful in both laboratory tests and field trials for various viruses affecting plants and highly efficacious from both on pathway and was observed to reduce the symptoms of wilt disease. Pathway antipathogenic solution at 2% concentration is effective in preventing the disease, lowering the pineapple mealybug wilt disease infection, thus produce high volume marketable quality of QP fruits. The result proved that the pathway can be used in pineapple mealybug wilt disease management. Thank you very much. And General Variedad. If there might be questions, I'm ready to take in. Thank you very much.
Well, C is here only. In case you have any questions, you can discuss it one-to-one. Well, now moving ahead with Dr. M. Nedu Chensingya. He is from ICAR-CTCRI, Bhubaneswar. Is talking on value addition and entrepreneurship development in tuber crops.
Good afternoon, everybody. I'm presenting on a special crops. Generally, we speak on cereals, millet, pulses. We rarely speak on other crops. We get rare opportunity to see this crop also, to speak also. So today I am presenting on tuber crops. Tuber crops, generally, tuber means potato will come to our mind. Other than potato, a lot of tuber crops are there. We have grouped tuber crops into 6 categories. The main one is cassava, very popular tuber crop in South India. Elsewhere in the world also it is very popular. In India only, South India only it is popular. And part of Koraput, Gajapati, Ganjam district also it is a popular crop. Next is sweet potato. In Odisha it is a popular crop. Southeast Asia it is a very good crop. China contribute 80% of production in the area in world production. Next, a group of aroids are there. Elephant putiam, taro, tania. These are 3 groups. And yam bean, the special crop, it belongs to Eastern India. Arrowroot, it is also a group of 3 crops are there. All the 3 genus you can find in Odisha. Maranta, Arundinese, Karkuma, and Kannayudulis. All these 3 crops are used for starch extraction. You can see that very popular crop. Sweet potato. It's around 40,000 hectares it is cultivated in Odisha. It's a very good crop, contains 20 to 23% starch. Mainly, sweet potato is consumed after boiling or sometimes used as a vegetables. Otherwise, in India, there is no value-added product. But yes, elsewhere, if you see Southeastern Asia, China, Philippines, and other Korea and Japan, lot of value-added products are there. Especially China, whatever noodles they are producing, 95% is from sweet potato, not from wheat. That is for your information I am telling, because it is a low glycemic index food. And we have developed a special varieties. One is Bhusona and Bhukrishna. Bhusona contains 14 milligram of beta carotene, and Bhukrishna contains 92 milligrams of anthocyanin. This is a special variety for industrial purpose, for value addition. So in these 2 varieties, we are popularizing chips production. As we know that in India, chips production means only from potato, but we can produce a very good chips from sweet potato. It's a very popular in China. But these 2 varieties, what we have developed from India, this is a very good for chips production. And also, these are some of the equipments used for chips production. Slicers are there, and small equipment, fat frying machines are there. With that, one can start small business. We need not wait for bigger one. And from these varieties, we can produce a jam also. Very good jam you can make. We used to make only fruits, but tuber crop also you can make jam. Sauce also you can make. It is also a very good product. This can be easily marketed in India. And this is a general way of using. Then apart from that, we can go for flour making. Flour is the basis for many of the product development. And we can also produce a flour from this orange flesh and purple flesh and normal sweet potato also. From the flour, for making small equipments are available in India. This is fabricated in India and especially in CTCRI. And this flour you can use in a locally for tribal people or local people. They can go for preparation of chapati roti like wheat flour. We can use it. You can substitute. We can go and use for like a mandia ragi flour also. We can go for cakes preparation and other things. And this is the way of you have to peel it. First you have to peel it, powder it, and powder you can use it for other snack food production also. And so these are some of the tools, so equipments required for preparation of this product. These are the snack food machines and snack dices are there. With that, one can start the small business. The other one is advantage of sweet potato is it is a gluten-free. And we know that gluten is a major problem in certain areas causing celiac disease. And those areas, generally, they market gluten-free products. Such product can be prepared from sweet potato. Sweet potato is completely gluten-free. And gluten-free cookies and gluten-free breads are also now coming to the market. Elsewhere in the world, it is already, the production is there. And we have a technology in India, and anybody wants to have a small business, they can have a collaboration and they can have our technology. We can produce gluten-free bread and cookies. These are the things. And we can have pizza also, pizza base also we are making. And this is a bakery oven. It's a portable one and very simple, and one can have small business with that. And the technology is also available, machinery also available, and we can guide them. This is a pasta making machines and very small machine, comfortable. One can have a small business using a sweet potato flour. One can go for pasta making. This is orange plus and purple plus pasta. As I said, the two varieties we have released from our institute. Using that one, it is a naturally fortified varieties. And it has a very good market, international market, and also you can produce noodles also. These are the small equipments attached to pasta making and noodles making equipments. And this is a kurkure type of extruded product. You can make it from the sweet potato flour. Kurkure type of product also we can make. This is the equipment. The equipment is indigenous. It's available in India. So we can assist them. Anybody wants to make a business, and we can help them to procurement and other details we will give. So one can make comfortably small business they can make. This is the kurkure making, small demonstrations. And we have a puppet making machines. So sweet potato flour can be used for puppet making, just like wheat flour and maida flour or berry flour used for puppet making. This is the equipment, small equipments, and one can use it and using sweet potato flour. It's a flour like wheat only. These are some of the ancillary machinery required for making business. This is automatic packing machines. The packing machines also available locally, and one can make small business by investing around 15 to 20 lakhs. One can run. Once it's successful, then they can upgrade and upscale the business. And other products also we can make from sweet potato, bigger products. This is a biofortified wine. Technology is available with us. The wines are rich in anthocyanin and vitamin A. And bioethanol, now it is going to come in future. The ethanol production, alcohol production also possible from sweet potato. The other one is arrowroot. It is specifically very popular in Odisha. There are 3 types of arrowroots are there: West Indian arrowroot, Queensland arrowroot, East Indian arrowroot. These all 3 arrowroots are cultivated in Odisha. This is Kanna edulis, very popular in Rayagada and Koraput district. This is the traditional way of starch extraction. All the tribal women, if you go Koraput, they will be keeping 5 to 6 kg of arrowroot starch, because these people are not easily accessible to medical facilities. For their ailment, they immediately use arrowroot starch only. Any disease, any problem, any stomach upset, they take arrowroot powder, dissolve in water, and they will drink. So they keep at any point of time 5 to 6 kg arrowroot starch. This is a traditional way of making arrowroot starch. Of course, it is not hygienic. This is the only thing, but it gives supplementary employment to the tribal woman, of course, but it's not hygienic. We want to make it a hygienic way. That way, we have introduced some of the equipments. This is the equipment, small equipment, not very high cost equipment, around 20 to 25,000 only, machinery crushing equipment. We can crush it, make it pulp, and from the pulp, they can go for starch productions. We have demonstrated. This is the starch. And we have developed a food also, Palwa Laddu, they say, arrowroot laddu. It is a very popular in Badrak, and we refined the technology, and it's an excellent sweet dish. Can be stored for 3 months, up to 3 months, and it can be exported. The other crops I'm just briefing, it is a cassava tapioca, very popular in South India and part of Koraput. And usually it is boiled and taken. This is the things. Just they boil it along with the fish and mutton. They used to consume it, but we can go for commercially lot of value-added products. It's a jam production. This is the chips, like potato chips in North India, South India, especially Kerala and Tamil Nadu. Cassava chips is very popular. And snacks foods also we can go. These are some of the machinery. Bread also very popular in African countries. Bread, wine, and beer. All those things are prepared from cassava only. These are cookies prepared from cassava rava also. Sabudana, this is a very popular dish. People are aware. This is coming from cassava only. People are not knowing this one. Then sweetness also prepared from that. Biodegradable plastic, of late it is getting importance because this is the main source for preparing biodegradable plastic. And we have a technology, many of adhesives also coming from this one. From the cassava stem, we can prepare particle boards like fly boards. Particle boards can be prepared, and from the leaf and seed extract, biopesticides can be formulated. Very good biopesticides. It's against the sucking pest and borers we have tested. It's very effective. Weevils and other things. And mealybugs, papaya mealybug. This is a plant, biopesticide extracting plant. And this is a Dioscorea. You will be knowing very well. This is elephant putiam, colocasia, and yambin. These are some of the product. Just I am giving conclusion in brief. This tuber crop, the tubers can be consumed by fresh, boiling and making some vegetables. And you can go for fresh tubers. You can prepare chips. You can go for making flour. You can extract starch. Some of the extractants, vitamin A, that is a beta carotene, can be extracted. And from the leaves and stems, you can go for biopesticides. From the extractants, pigments, starch, you can go for multiple things. Bioethanol, biodegradable plastic, glucose. And from the flour, bakery products, snack food, and extruded product. This is the nutshell I want to give. It's a very good food. It is a future food crop, and try to have a few plants in your backyard that will save you during flood and drought. Thank you very much. Thank you. At least find a time for eating tubers. Thank you very much.
Thank you so much, sir, for finishing on time, and we are running time constraint now. So moving ahead with Engineer Chaitali Mahatre, ICAR-CIWI Bhubaneswar. She'll be presenting a virtual presentation. So I guess, can we connect her on Zoom call? She's there.
I'm here, madam. Am I audible?
Yes, ma'am.
Okay. Kindly permit me to share the screen.
Give me a few minutes now.
Screen is visible?
Give me a few minutes. They're connecting. Yes, it's visible.
Yes. Okay. So, first of all, welcome to everybody. Are the slides moving?
No.
Yeah.
Yes.
Yeah. So, first of all, I would like to thank the team of Agri Vision 2024 for allowing me this opportunity to present my work here. Today, I'll be talking on the aspects of rural women entrepreneurship through establishment of custom hiring centers. I belong to the Central Institute for Women in Agriculture. And working with rural women is a predominant activity for our institute, and my expertise is in farm machinery and ergonomics. So I'll be incorporating those aspects also, how they are important while establishing a custom hiring center. So first of all, one second, let me just hide this bar. Yeah. So first, when we say about custom hiring centers are equipped with the farm machines or process equipment machinery. Now, what is the need of farm mechanization? As we know, there is a male and youth migration in two sectors, in two dimensions. First, they are migrating from the villages to cities, and there is a sectoral migration also. They are migrating from agriculture sector to service sector. So what happens is that in the home, a family labor is only remaining as a main source for source of labor, the family people, and they are comprising of women, children, and elderly. So what is the need for farm mechanization? Because farm mechanization can reduce the labor requirement and it will increase the labor productivity. It will also help us to eliminate child labor and drudgery. Farm mechanization is needed because climate change is reducing the number of workable days. The time period between two viable windows of harvest or of planting or of spraying is getting reduced, and it's challenging to finish off this operation without machines. So we need machines to tackle the climate change also. Then again, there is another hurdle, that is seasonal labor, because agriculture is not a continuously monitored or continuous labor bearing activity. Whenever there is a transplanting season, whenever there is a harvest or threshing season, at these 2 points we need a huge amount of labor. So when everybody needs labor, then the labor becomes scarce. So the unavailability of a seasonal labor is another factor which can be countered by farm mechanization. Another issue is that these farm laborers, when they do not have a work during this lean time period, it causes a social distress or social issues for them. The positive aspect of farm mechanization is it enhances efficiency. In what way? Efficient usage of energy, input, reduction of losses, increasing productivity, and increasing cropping intensity. The last but not the least is that it creates entrepreneurship opportunities. How it can be done? It can be done through custom hiring centers, through manufacturing and service sector. Ultimately, all these things will reduce, will lead to increase the farm income. But what is observed is that there is a wide gap in technology absorption capacity across various farm categories and farming system. That means a large scale farmer can adopt higher quality of technology, whereas the marginal or the small scale farmer will not be able to adopt the advanced technologies in a similar manner. Also, farming system, in wheat, in rice, we have good quality of farm mechanization available. But if we go towards horticultural crops like fruit orchard, still many operations are done manually. So we need to address this gap of absorption of technology. So how we can do that? Okay, so I would also like to bring into notice the major need for mechanization is addressing the drudgery, and especially women's drudgery. You can see wherever women are involved in the agriculture, they are one of the most drudgery prone activities: transplanting, weeding, sowing, load carrying. So the women have to stand in knee-deep waters while transplanting paddies. They have to work in bending or squatting postures while weeding. They have to work in scorching heat conditions. They have to work during the rainy conditions. So we need to address these kind of issues also with inclusion of farm mechanization. Now coming to the topic, what is the need of custom hiring centers? So women cannot afford, but they also cannot avoid the usage of machinery, as I have stated previously. If we think about heavy machinery like tractor or power tiller or combine harvester, these heavy machineries are not scale-free technologies. And the individual ownership is not a viable venture for farm women because they do not have capital to purchase machine, and sometimes they do not have freedom to invest time to operate the machine or to develop a skill for the machine. But usage of machinery is unavoidable due to climate change. Hence, you can see that we have found a niche here, that there is needy farmers and there are machines which are sitting idle in some of the houses because they will be sitting idle after working in one field only. So when we can overlap these, we come into concept of custom hiring centers, that if someone has an idle machine sitting in their home, they can give it on hire to the needy farmer. This was the actually origin for the for the concept of custom hiring. Custom hiring is not a new concept. It has been since 19th century, and even in India it has been set up as agro service centers from 1971. Major was to provision of farm maintenance, hire, spare parts, agriculture inputs, soil testing. So we see that this is not a novel approach. It has been there for several decades, and the benefits of which are profound. So, and especially small marginal farmers have benefited from the concept of custom hiring center. Now we need to extend this proven benefits to women farmers by making the farm tools and equipments available to them at affordable rents, for timeliness of the operation, by solving the labor shortage, by spreading the ownership of the machine over large, because women mostly they work in collectives, and when they work in collectives, they can pool their resources and they can spread out the risk. So spreading of ownership over a larger area or larger capita, then they can also avail better matching equipments to them. They don't have to be only restricted to use machines or small tools and equipments which they only own. With the availability of a custom hiring center in the vicinity, they can be getting a better matching equipment as per their need. Now, how can we turn this need of farm machinery or need of machines into an opportunity? We can do that by transforming a women farmer or a women labor to entrepreneurs. For this, we need some of policy, execution, and social approaches, such as inculcating gender concept through planning and execution of policies, that is making women-friendly policies, gender sensitization of implementing agencies, gender-friendly process of availing credit, gender-sensitive market environment. Women should be included as a development partners, not just beneficiaries. That we just don't want them to be only benefiting from. They need to be there from the start of the process. Gender sensitive climate at a community level, and amalgamation approach of women into collectives, as I said before, SHGs, FPOs, etc. So we in ICAR-Siva have been associated with the projects where we have established custom hiring centers throughout Odisha, especially in the Puri district, in the Mayurbhanj district, and in Koraput district. Through this establishment process, I have come across certain points that I would like to highlight. While establishing a custom hiring center, we need to assess the village where we are going to establish a custom hiring center. First is the crop. What kind of crops are taken? You cannot just go and give a list of machinery which will be able to handle only one crop, like paddy. We need to see what kind of a secondary or tertiary crop is being taken, like whether they are taking rabi crops of vegetables, whether cucurbits as a summer crop is taken. And then we have to also make a complete package, annual package of machinery for that particular village or for that particular district. Then we also have to consider the geography, because we have established a custom hiring center in Puri districts, and we have also established custom hiring center in Mayurbhanj, Similipal area, for forest area also. So these two are having completely different landscapes. So what we observed is that when we have a hilly area, we cannot drag a heavy machinery up the slope. So we need to advise them with a with a less weight or a simple machinery. Like in place of tractor, we need to advise for a power tiller. The other factor is that they don't need tractor also in that area, because the land size is smaller due to the hilly terrain. So the geography also makes an important point. Another thing in geography is that whether it's a rain-fed area, whether it is a drought area. So what kind of maintenance will go in place of these machines? That also, geography plays a role in maintenance also. Next is we have to ensure availability of electricity, fuel in the area, because this we learned through an experience. Actually, while we were working with Koraput and Mayurbhanj Similipal area, we developed a general package of machinery and we established custom hiring centers. 2 months later, when we went there for taking an impact analysis, we came to know that they have not been able to use the electric thresher. The reason for that was the intermittent supply of electricity, and they cannot take the electricity lines out to the fields because of the terrain. So, and the intermittent supply of electricity was happening, and the three-phase motor thresher that we had supplied could not run on this kind of conditions. So then we rather replaced it with a manual pedal operated thresher, and the pedal operated thresher was much more user friendly for them because they could actually use it. So these kind of things also we have to take care. Then availability of spare parts, because we are not giving them a machine or we are not establishing a custom hiring center for where the machine will stay indoors. These machines are going to work outside. They are going to work in muddy terrain, in muddy fields. So there is going to be a wear and tear. So we need to make sure a good quality spare part availability and maintenance facility. If not, then we need to create a spare part and maintenance facility available. The next and the most important factor is the human resource. So we need to assess what is their knowledge level, what is the skill level, and what attitude level, and what kind of access they have. So if we have a village where the knowledge is high, where the access is high, and where the attitude is very positive towards machinery, only the skill is lacking, then we can pool our all resources towards only developing a skill upgradation or capacity building kind of a training module. But where we have that access is low, attitude is poor, knowledge is moderate, and skill is moderate, then we need to pool our efforts towards first changing the attitude and increasing access towards maintenance, spare parts, machinery, and generating a knowledge by giving operational and skill enhancement training. So that is the way you should first enter the village. We need to do this is a kind of a PRA technique that we need to do. The next is when the training module is generated, we need to first create awareness regarding the tools and imparting knowledge of operation. Then we need to do the frontline demonstrations of machinery, training of master trainers, assembly of the tools. We need to give them basic training regarding assembly, because if they are not able to repair and maintain a little bit of their tools, then every time the women especially cannot run towards, because they have a mobility issue, they cannot every time go and hire somebody. Other thing is that we need to set up a particular management structures. If we are going to establish a custom hiring center in an SHG mode, then they already have a president and member secretary among themselves. But still, in place of that also, we need to make sure that there is a financial person to take care of financial requirements, a person to take care of wear and tear requirements, and so the management structure should be well devised. Another thing that we need to teach them is a very important thing, means all things set apart. This one thing is very important, that is logbook keeping. That logbook keeping will help them to maintain the financial aspects, and it will also help them to maintain the wear and tear aspects by logging in how many hours of work has been done by a particular machine. Next is tangent businesses to custom hiring centers. As I said before, spare parts supply and repair and maintenance are very important sectors. So we can encourage the women from that SHG group or from that group who is establishing a custom hiring center to start a spare parts supply or a repair and maintenance shop. This custom hiring center can also become a training center for other women and men of the village, where they can learn about these machineries. The women that have established a custom hiring center themselves can go as a skill on hire for an additional income. These are some of the glimpses of the trainings that we had imparted to women while establishing custom hiring centers. First, we have to interact with the farm women to choose which kind of women we will be inculcating in our custom hiring centers, which are more motivated and who can be a manager, who can be a finance person. So through these interactions, we can gauge that. Then we have to give them training from assembly to repair and maintenance. We have to train them how to assemble at least simple machineries they need to assemble themselves. We have to also tell them about the safety while operating farm tools, because obviously safety is of paramount important. We need to give them correct on-field training regarding operation of various machinery. So this is one of the success stories that I had told. We have established 44 custom hiring centers in Lampaput block of Koraput and Jassipur block of Mayurbhanj, and they have generated substantial amount of revenue. They are pro farm tools now. They have access and control over farm tools. They have adopted even more improved practices like SRI. The tribal pockets also women are using machinery. Then drudgery reduction is obviously there because the machinery is being used. Women farmers are enjoying the farm tools and it gives them leisure time. This is another aspect, because when you use machinery, the operation is completed in various is completed in lower time. So these leisure active time can be used in other remuneration generation activities. Another thing is that recently the institute has been given a SCSP fund. So we have pooled this fund together and executed by establishing 26 custom hiring centers in Puri, Khorda, Balipatna, Jagatsinghpur, Cuttack, Kendrapada, Ganjam districts. As you can see, these are some of the equipments that we have done given in custom hiring center, and they have been the farm women have been using it very successfully. So outcomes of CHC for women entrepreneurs is, first of all, it's an income generation. So it gives a financial independence. It enhancing women's access and control over productive resources, development of emotional intelligence, stress-free lifestyle, confidence building, and leadership qualities. Thus, it leads to socio-techno and economic empowerment of farm women. So thank you.
Thank you, ma'am, for the wonderful talk. So we are not having any questions as of now because we are running short of time, but they'll definitely connect with you if you could share your mail ID and all. So the next session, Dr. Jyoti Nayak, she is not available on the video conferencing.
Is she there? Okay. हाँ जी वही है डॉक्टर ज्योति। 5 मिनट में हो जाएगा ठीक है। आपका वर्चुअल था ना?
Screen change, please.
Respected chairman and co-chairman and also rapporteur and all the delegates. Now I am going to present the topic that is rural women conserving environment and sustainable development. नहीं हो रहा है। मैडम स्लाइड नहीं चेंज हो रही। हाँ 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 Introduction current. Huh. You all know that women play a vital role in the sustainable development of the nation, because you know that basic unit of the society is the family, and the women acts as an agent who transfer the culture, heritage, norms, various values. क्यों नहीं हो रही यार? Culture and heritage of nation, various values in society, and tradition and knowledge also. Sustainable development of a country determines poverty, eradication, and also environmental conservation. You should know that in a developed country, that gender equality is a major fact to get sustainable development. Developed countries are providing equal opportunity and platform for both gender and ensuring women participation in national progress. But in case India also needs to concentrate more and more to empower women by means of educationally and financially. Yeah, or yeah. Oh. Okay. Oh, next. If you see the rural women, they are engaged in multifarious activity. Starting from the household activities, they are engaged in agriculture and allied sector also. If you see the statistics, इतना problem हो रही है यार. If we see the census here, we see their percentage of during 2024, their share, women's share will increase to be 60%. So the women involvement in not only in agriculture but the allied sector. In Indian agriculture, women perform 4 different types of roles. First, as a worker, a source of power. As an operator, is a controller. As a manager, a farmer. As an entrepreneurship, a business person. So, women not only doing the household activities, but also in the rural women. I am not talking about the urban women. Even rural women, they are acting as a farmer to till entrepreneurship. If you see, there are so many technologies available in the market, but all the technology are not reached at their point. Means rural women are not getting almost all technology, not because due to some many reasons. The first reason is the technological barrier. They don't know what are the technology available. And second, the passive attitude towards the modernization. Though there are so many training are given, but due to passive attitude towards modernization, they are not adopting. And marketing system, due to marketing system, they are not getting. And also training, lack of training as skills, they are, for which they cannot operate the technology. And also credit facility, and the most important thing in our country and also in Odisha, due to social barrier, they are not getting the technology. Next. So now the question arises empowerment. Empowerment means moving from a position of enforced powerless to power. To make them power, it is most important. Today, the need of the hour is to make them empowered. Empower means we have first skill empowerment, but training in the area of various operations like field operation, conservation of biodiversity, nutrition, biosecurity, vocational training, organic farming, some of the training, they should know the skill empowerment. We should enhance their skill. Second. Next. Technology development of women, designing tools for various field operations. Previously, the male folk, they are going to the agricultural field. So previously the scientists, they have developed according to the structure of the male folk. But now the involvement of the women is in the agriculture field is increasing. So according to their health status, we have to design the tools which will be the women friendly and they can operate easily. So also animal husbandry, their involvement is increasing. So they should training, we should give training to the artificial insemination and also then creation of self-help group, which is very much important. And next. So also for side income, mushroom cultivation, floriculture, that should be given training to them. And self-help group, which is very much important for the farm women, that you know, for financial support, for generation of employment and projection of contribution of women by collecting, analyzing data. Data collection is also very much important. Collection and display of data, projection of successful women in agriculture, representation of their contribution in economic terms can be displayed, and providing financial power, which is also important, giving them representation in the land holding is also the need of the hour. So importance and involvement of women in sustainable development is very much important because women's contribution and participation have high significance in the substances of the nation. Nowadays, our country is using male human resources directly and mostly female human resources indirectly. If we use these female human resources also as a direct manner, easily our country can become developed. If women have to contribute in national development, first we need to empower them through educationally and also economically. Education and economically self-sufficiency are the two keys for the empowerment of the women. You know that our Siva Central Institute for Women in Agriculture, the main mandate is the gender mainstreaming in agriculture and allied sector. So our institute, we have one project, that is Nutri Smart Village village program is going on in the 10 district in the 10 villages of the of Odisha in the 4 district, and we are giving them intervention, that is development of the Nutri garden for livestock and the mushroom. Mostly we are doing for the small and marginal farm women, for the technological and economical empowerment for the farm women. Next. These are the local of the project we are doing in the 10 villages of the 4 districts of Odisha, and from 10 villages we have taken 30 households, total 300 sample size we have taken. Next. We are given, first we have identified the need and also structure questionnaire developed, and we identify the need, and according to their need and demand, we have given them input for developing Nutri garden and also rearing poultry and also mushroom cultivation. And these are the input we have given. Next. These are some of the inputs, and also we are giving the hybrid rice variety also we are giving. Next. training sufficient. Next, madam. Training interface continuously, we are imparting training to the farm women at the village level as well as in the institute level, and we are giving them continuous, we are storming their mind setup, we are changing their, motivated them to change their mindset and behavioral changes, attitudinal changes. We are continuously interact with the farm women in the village level. This is the impact after intervention. We have seen their production in mushroom fishery and nutri garden poultry is increasing. Not only the production is increasing, but the income is also increasing, and nutritional security for mitigating the malnutrition, we are focused for the vulnerable group also we are doing. Not only the production, income, nutritional security is increasing, but one changing, that is their mindset, that is transformation. They are engaged group, and they are cleaning the ponds themselves for fingerling stocking. Next. So also the unutilized space, they are themselves cleaning and establishing the community nutritional garden. So also they have developed a group in group, self-help group, they are developing the kitchen garden. And next. So from this, what we have observed that not only their production is changing, not only their income is changing, not only their nutrition security is enhancing, but also a transformation, that is their mindset up and the behavioral changes, which is very much important for them, because now the time is coming that we should involve the farm women in all aspects, because in the first slides I have seen showing that during 2025, the participation, their share is increasing, that will be near about to 60%. So the first sustainability development is the development that meets the need of the present without compromising or without harnessing the ability of future generations to meet their own needs. The ultimate age is conservation of earth and different species. Various systems are involving in the development, such as ecological, social, and cultural, and political, etc. So, one thing that the policy, the political person, state government, central government, all should give equal right for equality in both the gender. If the farm women not involved in all the programs, then we will not achieve, never achieve the sustainability, because women are the counterpart, and they are the main source for our sustainable development. In developed country, which I mark that in their country, there is no disparity between men and the female folks. So we should give equal importance to the women folk, then only we will definitely reaching to the sustainable goal. Sustainable development and the women empowerment and economic development is closely related. Female economic power also enhances the wealth and well-being of the country, and to advance women economic opportunity include financial self-providence, women and agriculture, enterprise growth, technological assets, capacity building, and business leadership. By ensuring women empowerment, we not only promote gender equality, which Sustainable Goal 5 meet the Sustainable Goal 5, but contribute to other goals like good health and well-being that will meet the SDG 3 also, decent work and economic growth, SDG 8, and zero hunger, that is SDG 2, by improving food security. Political empowerment is a key driver for achieving SDG 16, that is peace, justice, and strong institution, and no poverty, that is SDG 1. So for any society, if we will not involve the women, that will prove that in our research project we have that in initial stage They are not doing anything, but now they themselves calling us, madam, please come, and we are doing all these things. So there is a drastic change transforming between them. They themselves, we never told them to clean the those on those land, but they themselves preparing a group. They themselves developed a nutrition and called us to, madam, please come and see. So this is the drastic change. If we'll give them scientifically, technological skill development and credit facility, definitely our country will definitely will reach to the sustainable development. Thank you all.
Okay, now I'll request Mr. Ghanshyam Lal Vyas, GTUV Renu Land, Indian Private Limited, Noida, Government Inspection and International Trade is his title. Hi, no, ma'am. He has to leave early. He's requesting me since a long time. Yeah, na? Government inspection. Yes, sir. कर दिया? मेरा काम तो है।
ये नीचे करने के लिए इसमें डाउन करने के लिए ये है ना. Yeah. Good evening, friends. My name is Ghanshyam Lal Vyas. I am from TUV Land India Private Limited. So it's a nice experience with all of you. Here, lot of the professors, scientists, doctors, and as we are delivering this lecture in Research Institute, so of course this is expected. Most of the professors and the scientists are there, but I am not from this category. However, I am having the engineering background, so my topic is different here. Most of the professors and scientists discussed about the rice, agriculture, crop, how to use the fertilizers, all these enhancement, and the women entrepreneurs has been addressed by the speakers. So it was very nice interaction. So now I will tell you about something different. So for the agriculture products, we need the testing, we need the certification, we need the inspection, which is very important to maintain the quality. So today we are talking about the this testing, inspection, and certification. This is also the one of the most important requirement. As for any of the product, when we are going to purchase in the market, we always seeing, is it having the any quality mark? Is it BIS registered? Is it CE marked? Then only we will purchase that product. So this is the today requirement. Every single person seeing the quality mark on the product. So these quality marks provided by our company and our organization. So TUV Rhineland is the German company. Our head office is in Cologne, and India head office is in Bangalore. We have different business fields. We are working to the mechanical products, textiles, food products, electricals, chemicals, and we are also working on this, green fuel, carbon footprint. So all these are our services. Our organization is 150 years old organization. It is founded in 1872. So last year we celebrate the 150 years of the anniversary of our organization. And these are the main ethics of our organization. We believe into the integrity, excellence, customer satisfaction, performance, and agility. Agility means prompt. Prompt response is required by the today customers. Nobody want to wait. And in India, we have the 17 locations in different cities, 29 laboratories, and
total 760 plus employees we have in the India for this particular TIC industry. TIC means testing, inspection, and certification. This is the building of our head office, which is situated in Electronic City, Bangalore. And I am especially taking care for the government inspections and international trade. So in this, we are providing the mandatory certificates for the various countries. If you want to export your product for the Saudi Arabia, Uganda, Egypt, Kuwait, Morocco, and Qatar, you need the mandatory certifications for the particular regulated products. So in further slides, you will be able to see in details. So this is the very general procedure. Whenever you are planning for the shipment, you will share some application for the certification requirement, and along with some packing list and invoice, we will review these documents and will revert back to you with the further requirements, like as the testing is required or not, inspection is required or not, any marking requirements. And then after reviewing of our comments, if you are able to fulfill the requirements, then only we are able to issue the certificate of the conformity, which is the final document which you required to export your shipments. So Saudi Arabia, we are talking about the rice, spices, buffalo meat. These are the main products going to the Gulf countries from India. So for these particular products, if you are planning to export to Gulf countries, you need the SFDA requirement, SFDA COC, Certificate of the Conformity. Saudi Food, Drug and Authority approved our laboratory to provide such type of the certificate. And these are the particular products for food and cosmetics. Cosmetics products comes under the FASE. FASE is the software from which you are able to see your certificates online on the government website. Another country, we are providing the certificates for the Kuwait. For Kuwait, various products are going to export from India, like as the electrical products, refrigerators, washing machines, household appliances, ceramic tiles. So for all these products, we are providing the certificate of conformity so that your shipment will clear the destination formalities. Egypt. For Egypt, lot of the products going from the India, like as the textiles, ceramic tiles, food products from the various part of the countries. So we are providing that certificates for the Egypt requirement. These are the main products in this list. Textiles, garments, linen and textile linens, dyed and colored yarn, carpets, home textiles. Most of the products comes into the category of the textiles, mainly going from the Ludhiana, Jalandhar, this Kerala, Bangalore, Delhi, and Agra. These are the main locations from where product is going for the Egypt. Morocco is the another requirement for which we are also issuing the certificate of the conformity for the various products. These are the product categories for the Morocco: automotive spare parts, construction materials, chemical products, children toys, electrical products, oil and gas appliances, leather textiles and clothing, plastic products, PPE. All these are the various products categories going to the Morocco from India. Our company is authorized to issue the certificate of conformity for Uganda as well. Uganda, most of the grocery products going from India, mainly from the Gujarat part of the country, like as the spices, some snacks, furnitures, used products, food and food products. These are the main products going to the Uganda. Last but not least, we are also providing the certificate of conformity for the Qatar. For Qatar, mainly rice product or buffalo meat product is going from India, and we are providing the certificate of conformity for this Qatar requirement as well. Another country also added into the new list of our accreditations, Cameroon. We are providing certificate of conformity for the Cameroon, which aims to certify that products imported into the Republic of Cameroon comply with the applicable Cameroon standards and technical regulations before shipment. So clients need to contact with us if they are planning any shipment for the Cameroon market. These are the product categories for the Cameroon, almost same like as the Morocco: electrical, vehicle spare parts, textiles, furnitures, medical or musical instruments, food and food products, tobacco, petroleum. We are also handling the commercial inspections for the bulk commodities from this Pipla port, this Paradip ports, from JNPT ports, Mundra, Gandhidham, all ports we have our inspection team. They are doing the commodity inspections for the bulk shipments, mainly for the pulses, for the cooking oils, grains, and minerals, fertilizers. We are arranging these commercial inspections over there. In commercial inspection, we do the inspection as per the customized requirements, mainly for the quantity verifications. They nominate the inspection agencies to further investigate the shipment. In this, we generally check for the delivery for the completeness, proper packaging, visual inspection of goods, loading and offloading supervision, and tailor-made inspection requirements. Tailor-made inspection requirements means the client sometimes need to verify some details, as he never believed the second party. So they need the third party to verify the requirements. So in these cases, we can help the client for different testing, inspection, and certification requirement. Thank you, colleagues, to listen to me. If you have any question, then please come back to me. Thank you very much.
Thank you, sir. So can we have Dr. Lakshmi Priya Sahu, ICAR-CIWA Bhubaneswar, improving the quality of life of rural farm women through sustainable agricultural interventions is her topic of discussion. Screen change.
Good afternoon, Chairman. Good afternoon, the co-chairman, rapporteurs, and all the delegates present in this hall. So myself, Dr. Lakshmi Priya Sahu, and I am working at ICR Institute, Central Institute for Women in Agriculture. At Women in Agriculture, we are mostly working on the empowerment of women and particularly on their life and livelihood. So it's a topic which is very less discussed when we are talking about the women-centric developmental initiative of government, which are mostly meant for the improving the livelihood of farm women and bringing empowerment. So as we all know, the farm women or rural women, they are doing many activities which are unpaid activities, like the caregiving activities, home management. So by imposing more of developmental initiatives,
through different programs, which are mostly family oriented. So women has to give time. So there is a very delicate balance has to be maintained between the livelihood of women, the development and the life of women. So I can tell that the quality of life for women, and what is the quality of life for women and why we are talking about that. So whenever I am talking about the quality of life of rural women, we have to see some indicators like health and nutrition, their gender equality, their access and control over resources, cultural and social inclusion. All these things has to be come under the quality of life, but it's not possible. As a researcher or as a developmental worker, whenever I'm going to village, it's not possible to address all the things which implies the quality of life for women. But these are crucial. What if we are increasing the income? What if we are giving them empowerment? At what cost? They should have bad health. They have very weak family nutrition. They have drudgery. They have illness. And what cost? And how the programs will be sustainable? And how we can tell that we are doing the sustainable development? So for that, we have to definitely go for the quality of life. So quality of life being a very qualitative aspect, the qualitative aspect that if my quality of life is good, if I am feeling good, if I am enthusiastic to do something, if I am healthy to take over something, then only I can say that my quality of life is good. But whenever as a researcher, I have to see the quality of life, I have to devise some tangible things which I can measure, which I can measure that the life quality is good. So besides this, all factors, I am only taking few tangible things like what is the participation? What is her participation in farm, non-farm, social and other activities, and how the efficiency of the rural woman is managed. She has to do a lot of things, and how this efficiently managed. Does she have some gadgets? Does she have some new ICT technologies? How she is managing her caregiving, her farm, her developmental aspects, children, education, and all things. Because a woman has to do more than 100 things. Then what is the economic gain? Whatever she is doing in participation for social development or other programs, policies, then what is her income, economic gain? Is she getting money? Is she getting the fair share of benefit? Is she a partner in getting the outcome? Or she is simply doing or undertaking the tasks and the result is taken by others. So that is also important. Then what is the social empowerment? Is she recognized in the society? And what is the social empowerment? It comes from technological empowerment, which gives rise to economic empowerment. So if she is socially empowered, she also takes part in politically things and gets more empowered. So these are the tangible pathways by which we can measure the quality of life. So for undertaking this work, we took it in a project mode and we selected 4 locations at ICAR-Siva, then Ludhiana, Jodhpur and Jabalpur. We covered the arid and semi-arid areas of the country. So what are the interventions? What are the interventions? Because now only I have discussed that for measuring the quality of life or improving the quality of life, we have to focus on few tangible things only. So accordingly, we gave intervention on health and nutrition, drudgery reduction, income generation, then awareness through health camps. So for building the scenario, we took the baseline survey on health and nutritional status, resource availability, drudgery reducing activities, and need assessment for income generation. And we took 4 collaborative centers. And in each collaborative center, we have taken 40 farm women. And on them, we applied the interventions to measure their pre-project quality of life and post-project quality of life so that we can compare the impact. So we found that 52.5% women are normal as per their anthropometric measurements. So for measuring the health and nutrition, we did some anthropometric measures, whether their body status is they are normal or obese, malnourished, and we found that more than 52.5% are normal and others are not normal. And they have 37% women were facing chronic energy deficiency from mild to severe degree. Because nutrition deficiency nowadays, we all are talking about hidden hunger, so that we are deficient in micronutrients, vitamins and proteins. But still in country, we have the energy deficiency. Chronic energy deficiency is still persistent in rural women. So they have the disease and health situation which was assessed, and it was found that tuberculosis and diarrhea affected the families from 15 to 30%. As large number of population were suffering from chronic energy deficiency, malnutrition, initial interventions has been initiated among 100 farm families. So we started intervention in 160 farm families. So while this is the data of chronic energy deficiency of the selected sample, and this is the disease and health situation. So we saw that 52.5% are normal and more than 17% are malnourished. So for after studying the nutritional situation and the other nutritional analysis, we went for intervention for nutritional improvement, because nutritional improvement is very important. So we developed 160 nutrition gardens to reduce the malnourishment, and in those nutrition gardens we focused, because nutrition gardens were developed in the backyard of the farm families, so very less area. So we focused on protein rich and vitamin rich vegetables which will give them nutrition, particularly colorful vegetables, protein rich vegetables we developed. So third intervention was skill enhancement training. We conducted a series of skill enhancement trainings for giving them awareness, for interventions, for making nutrition gardens, for business management, for nutritional things, everything we gave a series of trainings. And we developed linkage with Central Food and Extension Unit, Government of India, and we imparted 5 days comprehensive trainings on food and nutrition. So nutrition education was given mostly on milk production and its utilization, because in rural areas, people, they produce milk, they have cows, but women hardly drink milk or utilize milk. Then importance of kitchen gardening, importance of green leafy vegetables, then conservation of nutrients during cooking, hygiene, sanitation, and low cost weaning food for children. So these educations were given very comprehensively to farm women. So the 5th intervention is drudgery reduction of farm families, and we provided them different tools, trained them how to use very small tools and very low cost tools, but that will reduce their drudgery to some extent. So revolving stool for milking the cows, then cow dung collector, lightweight hand cultivator, trowel sprayer, and improved sickle. All these things we provided and measured the impact. So 6th intervention was empowerment of farm women for income generation through backyard poultry, then another off-farm activities like stitching bags and purses and animal management. So for this, we have given some demonstrations, that is value addition to agro and animal-based demonstrations. So in this, we have grown different crops and found that the production, then gross return, net return, and BCR, benefit cost ratio, and it's in the farmer's field, and so in some places the BCR is more than 2.6%. So while giving, I'm summarizing the interventions. So fruit plants we have grown, then vegetables we have grown, then kitchen gardening, distribution of inputs, then income generation activities. So the impact is they saved approximately rupees 2,000 for family through vegetables, because they used to buy the vegetables and from kitchen garden they collected the vegetables. So this has different benefits like reduction of cost, then organically produced vegetables, so it supplemented to health, and they were empowered technologically for their own nutrition, family nutrition. So they have income, generated income 2,000 to 3,000 through small candle making, wax making, all these things. So by empowering them in different technologies, giving them a community support system, we created an enabled platform for them. So in that platform, they get support, institutional support, hand holding, and we tried to develop and strengthen their local level institutions like SHGs, their federations. So these create a multiplier effect and empowered them. So though we started with just improving their quality of life, we ended up with income generation and giving them satisfaction. They ate their own food, they got income, and for very petty expenses, they don't have to depend on their husband. So these type of effects were found, and which was multifaceted and which has also good spilling effect. Seeing them, other women from other villages were interested to also come for these type of endeavors. So what are the checkpoints? Whenever we went for this improving quality of life, first is community. So community mobilization, their exposure, strengthening the grassroots organization is the first point. Then their awareness creation. For this, we created outreach programs. Then we created a platform for them which they can participate, and their participation behavior can be positively manipulated. Then interventions that should be location specific, and the location specific, need based, women friendly interventions were very important to bring a positive change in that endeavors. And then impact. For getting the impact, we did concurrent evaluations. At all stages, all points, we did evaluations and we measure corrections. So this is the checkpoints where we regulated our interventions endeavors to study. We wait a little, study it, then we proceed. In spite of going in a linear manner, in every checkpoint, we evaluated ourselves. So what is the way forward? Because this is the reality that we are going for village-centric development, and more specifically, we can tell that we are going for women-centric developments. So women-centric development, what is their time use pattern? What is the leisure? And what is the developmental participation? So we have to very intricately design this phenomena so that along with care be giving, they can do some income generating activities and get some leisure. For getting this, their rest and leisure, which is very important for their quality of life, we have to introduce them with new gadgets, time-saving gadgets, so that they can manage their efficiency. We have to give a community platform where they can exchange their ideas, where they can clarify their doubts, and where they will get motivation to go ahead. So this is very important. So policy matters should be like that. So policy considerations for delicately balancing rural women's time used for economic activities with caregiving responsibilities is the need of the hour. And women-centric rural development initiatives need to be taken care of women's quality of life indicators. Definitely, this is the thought of the day. So what are the wow factors? Because in this research, what I found that the quality of life, yes, we can address it. Yes, we can measure it with tangible forms. Yes, we can quantify it. So while doing developmental initiatives side by side, we have to ensure the quality of life the women. So the recipes being community mobilization for motivation, support and hand-holding, and institutional linkage and enhanced mobility is the with informed decision making, not decision making, biased decision making, informed decision making will definitely add flavor to the recipe. So we can cook the recipe very nicely and we can provide women a better quality of life. So to conclude, so what we have to do, we have to first overcome the socio-psycho barriers through mentorship and support system, then cultural barriers through sensitization, inclusion, participation, then mobility barriers through internalization of opportunities. The women cannot go out always. So we have to provide the required things at the vicinity of their village. Let the inputs be produced by women in their village and let the women be the input dealers. They must be the input producers. So this will remove lot of mobility barriers and the market barriers through linkage and ICT. So thank you. So I'll be very thankful if everybody will think a little about that quality of life for women is also important. A woman is a human being. So by putting more and more and more work in the name of livelihood, in the name of income, we have to look back little and we have to see, we have to stand a while that what is her quality of life? Should I look it before doing anything? So this is my thought of the day. Thanks for patient hearing. Thank you.
Dr. Tanya Sheth. Screen change.
A very good evening to all the respected chairman, co-chairman, rapporteur, and all the delegates present over here. Myself, Dr. Tanya Sheth. I'm working as a scientist in the discipline of vegetable science at ICAR Central Institute for Women in Agriculture, Bhubaneswar. Today, I'm going to give insight about the NutriGreens, the superfood for nutrition and income. We all know that women have to play dual role. Maybe she is a farmer or a woman, but if she is a farmer, maybe she is a rural, maybe she is placed in a rural place or urban or peri-urban area. She have to do a dual role. His or her farm work is an extension of her household work. So and at the same time, due to rapid urbanization and also male migration, tend the most agricultural work to the shoulder of women. So women-centric woman-centric technologies for improving their, for reducing their drudgeries and other research is already going on. And for women, mostly they are having so many constraints. They does not have proper land rights. They does not have much time in their hand also. So for developing any woman-friendly technologies, some important things have to be kept in mind, that the technology must be available in a very, it must be utilized a very small area. It should be less input intensive. It should be less time consuming, so that the participation of women in farming will be more. And women and malnutrition and gender are interlinked issue. So many aspects are interlinked for the malnutrition, and particularly women and child are the more affected. So coming to the rationale, now our Indian diet is changing towards a sustainable diet. We are now thinking of some nutritious food. And according to Thompson and Amoroso, food-based approach is the most viable and cost-effective approach to improve the nutritional quality of diet. And nowadays, people also thinking about nutri-rich diet. Nowadays, customers are also ready to pay a vegetable requirement for a particular adult per day is 300 gram, but the availability is around 200 gram. But if we see the consumption pattern, then as on an average, Indian man was consuming only 143 gram vegetable per day, whereas a woman is consuming around 138 gram per day. So our recommendation is around double than our consumption. So nutri-greens, mostly we can classify it in three groups. First one is the sprout. At sprout stage, it's also having a high nutrition. And from sprouts, it's mostly required in the dark condition for their growth. And all know that sprouts are also very, very much healthy. So microgreen is the transition stage between a sprout and a mature green. Microgreen is mostly as its edible part is a central stem, a cotyledon leaf, pair of cotyledon leaf, and pair of true leaves. And microgreen require light for their growth, and it's rich in all my vitamins and minerals as compared to its mature part. And mature leafy vegetables, all are known, it's a rich source of vitamins, minerals, and all these things. So microgreen is mostly edible seedling greens. It's grown from seeds of vegetables, cereals, then your, in fact, millets also, or some herbs, and it's having a distinct color, flavor, aroma. As it is consuming raw, so its nutritional value is keeping intact, and its fresh and crisp appeal increase the value of the food. As compared to seed and mature plant parts, it has rich source of phytonutrients, antioxidants, essential minerals, and at the same time, it is low in fat and carbohydrate content, and also anti-nutrient factor is much lower as compared to mature part. And it can be grown in several conditions. That means in open field condition, in greenhouse, in high-tech phytotron laboratories, and even it's the possibility to grow in higher altitudes also. And even for space farming also, microgreen is nowadays gaining importance. And it can also be grown in different medias. So microgreen is not a new technique. It's started back in the 1930, but in 1990, it's from California. It is mostly popularized as a vegetable contifi. Then in 1998, the word microgreens is actually come in pictures. And from 2010 onwards, the popularity of microgreen in the grocery shops and reputed restaurants is day by day increasing. And these are the crops which are mostly used for microgreen cultivations. And it is a very simple technique. It can be grow easily. Anyone can grow it at their home also. So 5 main things have to be taken care. Flat tray is required, and it can go for any type of soilless media. and untreated seed is used, then soaking of seeds and then sprinkling of water, and after germination, when they are true after cotyledon leaf, first true leaf appear, that is the perfect stage of harvesting. And sometimes some variety or some types of crops are having the tendency to again regrow from the same cuttings, but some are again have to resow. So after immediate, as it is a live part and having high moisture and very minimum shelf life, so it should be marketed within 1 or 2 days, but it can be stored in refrigerated conditions for 7 days. Our Indian diets are very, very much diverse diet are there. So there is also chances to incorporate microgreen in our everyday meal. We can, as it is mostly used as a topping or garnishing. As it is not cooked, so its nutritional value is also being intact. So in our Indian dishes, we can enrich the value with microgreens. And recently in the G20 summit also, the chefs are also decorating, also preparing millet products, millet dishes, and also decorating with microgreens. So microgreen is not only a technology which can be adopted only in the urban or peri-urban area. Our rural farmers also in a community approach, they can also be grow microgreen and can become an entrepreneur in this direction, because as it is required very low investment and very less space is required. Also vertical expansion can also be done. So it is a very, very much woman-friendly technology, and it is also having the possibility to grow round the year. Off-season cultivation is also there, and as we are not using any type of chemical pesticides, so organic produce, organically grown produce can be say it is one, and from 1 month a woman farmer can earn more profit. But at present, it's having a niche market, mostly the supermarket, reputed restaurants, and VIP colonies. It is there, but we are hoping that in future it is having a good market. As like the example, we can take a site from broccoli or mushroom. Previously, when it was there at the first time in the market, nobody is there to buying it. But day by day, when the people is aware about its nutritional value, they are going to adapt that crop. So, and also microgreen growing kit is nowadays a very popular in online platform. Also, you can search about microgreen growing kits, and it needs only 2 to 3 things, only cocoa peat or a growing tray and only seeds of 2 or 3 crops. So a woman farmer can, if we can create awareness and proper skill to them, how to grow and how to be licensing them with the marketing agencies, then they can become an entrepreneur in microgreen cultivation. So I can say that our Indian women farmer are already growing sprouts in their kitchen and also consuming it. And they, at the same time, they are also growing leafy vegetables in their kitchen garden, and also means ensuring their nutrition for their household family members. So why not they can also take the intermediate stage also as a better life income generating source. And it's also having a bright future in the near. So coming to the conclusion, as the changing climate, as per was the changing climate and dietary transformation of urban pupils, so it's maybe a good profitable enterprise, and also as it is also can be grown in off season, so high market price can be obtained by farming families, and proper packaging and branding can enhance the value of the products, and farmerman can also get more profit from this. And it would also provide prairie urban farmerman a substantial source of employment, as well as financially and environmentally sustainable means of subsistence. Thank you for your patience hearing. And if questions any are there, then I am happy to be answered. Thank you.
Dr. Divya Sharma. Okay, Dr. Biswajit Mandal.
I'm here online. Divya.
Okay, ma'am. Okay. Yeah, please connect her on the Zoom call. Yeah, you're visible, ma'am.
Okay.
Hello everyone. Today I'm going to present about one methodology which I have done for like a research in Uttarakhand in Haridwar district in a village level where I have tried to understand that how decisions are made to grow a crop at a village level. And because it has always been for farmers, it has always been a matter of concern that maybe for this crop cycle what can be grown, and what are the factors that are there to see into that. So this is the study area, basically Uttarakhand, and there are two major regions in mountains and plains. And we have seen that in plains, normally there is intensive cropping in the area of Haridwar and Udhamsinagar. And this study is based in Haridwar. And previously in 1980s, 1990s, farmers used to grow urad dal, arad dal, moong dal, millets. But now they have, the cropping pattern has changed and they have shifted their crops to other crops. Hello, I'm audible? Am I audible? Yeah, so the basic research question was, what are the different cropping strategies, coping strategies that has been taken up by farmer household, and how the factors and what are the factors involved in the decision-making process at the household in the area? So the methodology which I have considered is I have done household survey at 30% intensity, and then used a hierarchical agglomerative clustering technique to understand what are the major cropping patterns in the area. And then when I have understood, after understanding what are the major cropping patterns, I have tried to understand the variables which are behind that kind of cropping pattern in the area. And then I have done social network analysis to understand if the networks are also, like, people connected to other people are also influencing their decision to grow a certain kind of crop. And then I have like concluded that what are these factors and what are the major factors and what are the major concerns. So to understand that what all were grown in that area, I made a list, and there were around 15 crops in the area. And after surveying, what I have found is that there are many variables that actually affect their cropping pattern. So what I have taken is caste, economic class, land, soil type, water access, monthly income, and the labor ratio, primary occupation, and the water which they having. And after running the clustering technique and after doing the multi-regression model, what I've understood is there are basically 6 cropping patterns which were there. And if you see this frequency table is basically showing how many farmers are using that kind of cropping pattern, following that kind of cropping pattern. So the first cluster, they were growing poplar trees. The second one was they were growing cucumber, fodder, paddy, and wheat. And they were taking 4 crops in a cycle. Third one is again fodder, paddy, sugarcane, and wheat. And few of them were taking only 3 crops. So there was an intensity in, like, they have increased the farming cycles there. And what I tried to understand is, after considering these three major cropping pattern of having 4 crops at a time and 1 crop at a time, like poplar, or 3 crops, what I've tried to understand is how these what are these networks, and how every kind of farming activity is affected by people who are involved in that kind of farming. So what I did is I used a social network technique, where what I tried to do is created a network around that farmer, and from where they're taking their inputs, where the outputs are going, from where they are getting the information, and all those things I have mapped. And then I've tried to understand what are the effects on that kind of network. So there were 3 major crops. One was poplar. So there were 3 major farmer types who were growing poplar trees. So poplar were grown with fodder, sometimes with wheat, sugarcane, and then paddy plus wheat. These were the actors which were involved in those networks, like farmer, local relative, distance relative, mills, seller market, private bank, government bank, Krishi Vigyan Vibhag, private shop, beet society, Krishi Vigyan Kendra, agriculture district office, Ministry of Agriculture and Farmer Welfare, and government. So all these different actors were also were there in their network. So after doing the analysis, what I found was this was the network for the sugarcane farmers. This one, these are the poplar farmers, and these were the wheat and paddy farmers. So the analysis shows that for the sugarcane farmer, the network efficiency or the density was less as compared to poplar farmers and paddy farmers. So that means that actually there's a lack of social structure, because there are much more dependent on support from the mills and from the government. And the people who are actually growing paddy, wheat, they have much more interlinked with their own social structure. So maybe in a stressful situation, if I feel that if there is a sudden shift in somewhere, people who are more dependent on only few sugar mills or only on government subsidies or government interventions, they might be affected more, because if there is a change in policy, then it'll be difficult for them. But people who are into a different kind of other farming activities, they were actually a little more resilient than only depending on just one type of cropping. So I would say that at this, because this is just a village level study, this is actually catering to a kind of methodology which can be taken or considered for understanding nuance, in a nuanced way, what is happening in a village. Because sometimes we see that even for climate change, or even when we talk about sustainable agriculture, we want to understand that if in a place, not everyone will take a similar decision. If we have to have an intervention, even with the same climate, even with the same kind of stress, every person will not be able to take that kind of decision. Because even to adapt and to cope or to shift to another kind of crop, there are constraints. They are constrained by their line size, they're constrained by the soil type, they're constrained by even caste. Because in this village, most of the village vegetable farming was done from a very different caste. And for like a person who doesn't have the ability or doesn't understand how vegetable farming is done, it's difficult because then he needs to have training, or he and she needs to have training, and then to have that kind of labor. So when I went into the field and asked people, why you don't want to shift from, even if dependency on sugarcane is such, why don't you shift to other kind of crop? And then say that we don't have skills, and then we have to hire people, and labor is also not available. So it's a I would say it's a complex structure even at village level, to introduce new things or to cope to a new situation, like because now we are facing climate change, and we might see a lot of crop failures, or we want to introduce new seeds maybe, but then again the adaptability or the way a farmer would want to adapt to all this, or want to take this technology forward, will also depend on a lot of background factors. So this was a small study which actually shown has that potential, that actually we can go in a village and try to understand their own social structure as well, so that we have some people there where we can identify who are these people who can actually take up technology faster than others, and how we can support the adaptation of a technology or adaptation of an intervention in a holistic way, and not just through technology, but how to embed that technology in a social structure, because we cannot separate technology or intervention or any farming activity from the social structure as well. So we need to understand how their structures are, how their networks are, even if they are growing different kind of crops, because what I have observed is when these farmers, they grow something, there is a lot of things that goes behind, not only as technology or not only as the resources, but how they are influenced through their networks also. So yeah, so I think I would just want to say that it can be a very good method to understand what is happening, maybe at village level, or if you want to see it at a district level as well, that how a farmer shift the decision. So if we want to introduce something, how he will be encouraged to do that. It's not only the technology or ease with the ease of use of that technology, but also how he or she is influenced by a social structure or a network around her or him that also influences. So yeah, that is all from my side. Thank you.
Thank you for the wonderful talk, ma'am. And we will refrain ourselves from questioning anything as we are running short of time. So moving ahead with the next one, Dr. Biswajit Mandal, ICAR-National Rice Research Institute, Enhancing Livelihoods Through Module-Based Interventions and Farmer First Program, Impact of 7 Years Journey. That's his topic.
Good evening to all.
My presentation is enhancing livelihoods through module-based intervention in Farmer First program, impact of 7-year journey. Actually, ICAR has started a flagship program in 52 ICAR institutes as well as SAU in the name of Farmer First program. Here, first is farm innovation, resources, science and technology, where farmer will innovate themselves. So our NRRI Cuttack is one of the center. It started during 2016-17 with 4 villages initially. Then afterwards, 4 more villages adopted. That is total 8 villages we have covered. And the area is on Salipur block of Cuttack district, which is situated in the delta of 2 rivers. Total area is total area is around 1800 hectare. Net cultivated area is 1000 hectare around. And mean rainfall is around 1400 mm. Where major occupation is farming only, that is agriculture and livestock. The project was initiated with 4 major modules, that is crop-based module, and we have adopted rice and pulses mostly in crop-based module. Then second one is horticulture-based module, that is vegetable and fruits. Third one is animal husbandry-based module, that is poultry, duckery and aquaculture. And fourth one is enterprise-based module, where we have taken up mushroom cultivation, vermicomposting, and later on some other enterprises also we have taken up. During these 7 years, under various modules, number of interventions we have mentioned here. And in crop-based and horticulture-based module, we have taken up more number of interventions. Under crop-based module, mostly we have given that improved varieties of recently released varieties of our institutes for demonstration. Then apart from varieties, we have given different other interventions like transplant, other package of practices like transplanting of young seedlings, then line transplanting, then integrated weed management, then integrated pest management, integrated nutrient management, likewise. And here we have shown result of only 1 year, where we can see that average yield in different interventions, how it has been increased. Then we have demonstrated other improved practices also, like seed treatment with various bioagents. These are few practices. Then we have adopted drone technology for pesticide application. Under horticulture-based module, we have demonstrated high yielding variety of vegetables. Then mulching techniques. Then we have provided agro shade nets for protective vegetable cultivation. Then adoption of these practices increased the income of vegetable farmers up to 45%. These are the different vegetables, different varieties. Yield advantage after adoption of this, improved varieties.
it was found more than 70% for all the vegetables. And we have calculated the economic ordinaries of improved varieties, and it was found that the benefit surpassed about 3 times over cost. Then one of the important practices we have introduced, that is using potted seedlings and polymulching, then trellis in case of commercial vegetable cultivation practices. And it was observed that farmers are getting around the year income of up to 5 to 6 lakhs per year. Likewise, another practice, that is relay cropping in trellis system. In that case, farmers are getting around 7 to 8 lakhs per year, per acre per year. And one assessment was made in comparison to pre-farmer first program, and it was found that benefit cost ratio during pre-period was 2.48, and after the intervention it increased to 3.42. Under animal husbandry based module, emphasis was given to balanced feeding of dairy animals and introduction of poultry breeds, that is Banaraja, Rhode Island Red, and Karaknath, and demonstration of mineral supplementation in case of cow, goat, and poultry. And they have accepted, farmers accepted poultry over Dakari and Banaraja breeds over Rhode Island Red. And benefit cost ratio was calculated to be 3.72 in case of backyard poultry intervention. Under enterprise-based module, that is mushroom cultivation, was demonstrated for 34 farmers, and vermicomposting for 30 farmers. And when we have calculated its economic benefits, we found that around 2.23 benefit cost ratio in case of cultivation of mushroom. And in case of vermicompost production, by considering three cycles per annum with 6 beds, benefit cost ratio was 2.45 with net returns of around 1,36,000. Various training, demonstration, and technical backstopping was done for commercial mushroom cultivation. Then under enterprise-based module, another intervention, that is beekeeping, was done for 20 farmers. There are few groups, and we have given body making machine to such group, and they are producing body and selling to local market, and they have earned net return of rupees 15,000 per month with a benefit cost ratio of 1.5. One mushroom production unit was developed at our institute. Here a few indicators we have shown that how much percent of that indicators increased over pre-project period. In case of paddy yield, increment was 37.5%. Then in case of horticulture, it is 38.72%. Animal husbandry, it is 100%, and enterprise, it is 100%. And we have assessed also with the perspective of doubling farmers' income, and we could find that in case of high yielding varieties, rice, then rice and pulses, vegetables, poultry, and fisheries, income was increased more than double. So we have listed here some of the measurable outputs. So around 1,000 farmers from 8 villages got benefited from this program. 11 high-yielding varieties paddy replaced the earlier grown varieties in the villages. Then farmers are growing vegetables through commercial scale by using relay cropping as well as trellis system and earning around 6 to 7 lakhs per acre per year. Then 15 young entrepreneurs, they have incubated and promoted in mass production of mushroom and they're selling it. Few farmers, they're growing fish fingerlings. 2 women groups, they are making bori and realizing gross returns of about 30% of our total cost. And a mushroom spawn production unit developed at ICAR-National Cuttack. Here we have shown how many number of demonstration conducted over the years, as well as how many farmers adopted that particular intervention, that is particular interventions. Then linkages developed with various organizations over the period. These are few media coverages. Training programs conducted for the farmers. Few farmers awarded at national level at Pusa Krishi Vigyan Mela. So overall, there was significant increase in knowledge, skill, and awareness of farmers, farm women, and youths in the cluster, and their income has been increased substantially. So the livelihoods through Farmer First program at ICAR-NRRI Cuttack. Thank you.
Thank you very much, sir, for the excellent presentation. Any queries, anyone? If not, moving ahead, once again, give it up for Dr. Biswajit Mandal for the excellent presentation. Now, moving on, let's welcome Dr. Sudipta Paul from ICAR NRRI Cuttack, and he is talking on insights into paddy farmers' price safety net through micro-level field investigations. Please welcome, sir.
Good evening, everyone. So I'm here to talk on insights into paddy farmers' price safety net through micro-level field investigations. And here I'll take the example of minimum support price, the government policy of India. So as we all know that MSP has been long serving as a very important price safety net policy, not only for rice, but also for other crops. At least 22 crops are covered under this price policy. But it has also provoked debates time and again. It says that price policies, particularly this minimum support price policy, has propelled crop specialization at the expense of crop diversification, and it has promoted rice and wheat at the cost of oilseeds and pulses. Secondly, it is often told that minimum support price policy has an indirect bearing on the environment in terms of, first of all, crop residue burning, and secondly, the depletion of groundwater level. Having said that, it cannot be overlooked or undermined that the fact that MSP has been instrumental in serving the smallholder farmers through the years. So that is why we take here the instance of MSP to state the importance of price policy to support particularly the smallholder farmers, and also we'll try to conclude on the sustainability of this price policy through our experiment. We had two objectives. First, to identify the factors of access to and availing MSP. And second one was to evaluate the impact of MSP on paddy cultivation at the micro level. So we conducted this study in 6 states of India: Chhattisgarh, Maharashtra, Odisha, Madhya Pradesh, Jharkhand, and Bihar. And all these states are major paddy procuring states as far as the central pool for paddy procurement, the Kharif marketing season, is concerned. So household level, secondary data, disaggregated level, at the disaggregated level were generated through personal interviews and household surveys using a pre-tested semi-structure interview schedule. And although we followed the randomized selection procedure, but our initial screening suggested that the control and the treatment here, by means of treatment, we mean the farmers or the households who availed MSP. And by means of control, we mean the farmers or households who did not or could not avail or did not decide to avail the benefits of MSP. So when we compared these two groups, we found that there are differences at least in 4 parameters, 4 baseline covariates, which prompted us to go for first setting up statistical equivalence between these two groups, because unless and until we establish statistical equivalence between the control and the treatment group, our conclusion will be misleading. So in physical or biological or other sorts of studies, means getting or achieving such sorts of control is easier compared to such social studies. So that is why one of the prominent and most popularly used techniques, which is called the propensity score matching technique, which is proposed by Rosenbaum and Rubin. That technique we used to set up first the statistical equivalence between the MSP and non-MSP farmers as far as their baseline covariates are concerned. Then to reach the second objective, we use the analytical hierarchical procedure. And our results suggest that we found some interesting results. That is, we found that multiple sources of income, farmers who are having multiple sources of income are more likely to go for MSP or to go for disposing their paddy crop through MSP in the APMC market mandis. So why is it so? It may be due to the fact that multiple sources of income also increases the cosmopoliteness and multiple sources for information of the farmers. And as we know, that various other studies have also informed earlier that multiple sources of information has a direct correlation with availing MSP. We thought that it may not be individual. At the individual level, these decisions may not be taken. It may be at least a neighborhood or community-based decisions. That is why we also went for focus group discussion followed by an AHP-based analysis, and we found that convenience of disposal, lack of awareness about MSP, and dissatisfaction with the amount of MSP offered, these 3 factors are the topmost 3 important factors which determine whether a farmer will avail the benefits of MSP or not. So these findings are in concordance with the very famous Santa Kamar Committee report, which suggests that only 6% of the farmers in India can avail the benefits of MSP. And secondly, it also is in concordance with some earlier reports like Aditya et al. And some famous reports are there, which also suggests that yes, farmers, only 1/4 of the total farmers in India are aware about something called MSP exists. Only 1/4. That means 25% of the farmers. And as far as paddy crop is concerned, for common grade paddy, farmers receive an MSP of rupees 2,183 a quintal. And for the grade A paddy, they get 2,203 rupees. So you can also understand that whether this amount is sufficient as a support price. So our finding, the dissatisfaction with the amount of MSP offered may be occupying one of the topmost 3 places. That is why. So coming to the impact of MSP, we find that as we selected 6 different parameters: average selling price, that is price realization, then average volume of marketed surplus, percent marketed surplus, acreage under paddy crop, and average rice yield and varietal diversity. And in terms of all these 6 parameters, compared to the non-MSP farmers, the MSP farmers had a better result. So, so as for the first indicator, as you can see, that price realization, the MSP farmers get at least 545 rupees more than the non-MSP farmers. That is our estimate. And as far as the marketed surplus is concerned, 10% at least maybe the marketed surplus for the MSP farmers compared to the non-MSP farmers. Similarly, for the other parameters also, you can see that there is a distinct impact of MSP on rice farming at the micro level. So how credible these estimates are? It is credible in the sense that we satisfied the criteria of what is called your statistical equivalence in between the MSP and non-MSP farmers, and it can be seen through these 3 figures. For example, if you see that before matching, there was a huge gap between the MSP and non-MSP farmers in terms of area, means acreage, but after matching, the gap reduced a lot. So this way, for the other matching variables also, we could establish this statistical equivalence, and this is clear from these 3 figures. So finally, to conclude, we must say that paddy cultivation at the micro level, MSP has a very positive impact. Although the benefits are not uniformly distributed across the states, only few states are getting, the surplus states mostly are getting better benefit from MSP. And finally, seeing the importance of paddy in a country like India, it must be said that significant amount of more care should be taken while declaring MSP in paddy. With that, I conclude. Thank you very much.
Thank you very much, Dr. Paul, for the wonderful presentation. Moving on, now let's welcome Dr. Shushmita Munda with a round of applause. And she is talking on enhancing soil health and sustainability, comparative analysis of conservation agriculture in rice pulse cropping system. Let's welcome her. Please, ma'am.
A very good evening, all of you. I'm Dr. Sushmita Munda, working as agronomist at ICAR-National Rice Research Institute. I'm working on conservation agriculture. Conservation, the word is a buzzword today in the present context, and any technology that has the word conservation actually is considered the technology of tomorrow. So conservation agriculture is also considered as technology for tomorrow. Conservation agriculture relies on three principles. Basically, minimal disturbance of the soil, crop diversification, and maximum residue retention or soil cover. So based on this, we conducted experiments on rice-based cropping system, and we took green gram as part of the cropping system. The treatments, actually, the objective of my work was to compare how zero tillage rice or how conservation agriculture, the rice grown under conservation agriculture, is different or comparable or better than the conventional rice growing. And we know that in rice, there are two types of rice. One rice is direct seeded rice, the other rice is transplanted rice. In direct seeded rice, zero tillage or minimum tillage, minimal soil disturbance, people are aware of it. A lot of research has been done. But when it comes to zero tillage or a non-disturbed transplanted rice, transplanting in undisturbed soil, people don't know about it. Not much of research has been done on this aspect. We at ICR NRRI Cuttack, we conducted many experiments and we standardized the package of practices for non-puddled or unpuddled zero transplanted rice. That means transplanting done without tillage. So we wanted to know whether the results are comparable with zero tillage direct seeding. We also wanted to know whether we are getting any advantage over the conventional tillage system rice. So we had three treatments in the main plot, that is zero tillage direct seeded rice. And we followed this, zero tillage rice was followed by green gram in the Rabi season. The second was zero tillage transplanted rice in the Rabi season, followed by green gram in the Rabi season. Then conventional system of rice green gram, rice conventional tillage rice in the Kharif and conventional tillage green gram in the Rabi season. We have taken three pulse variety, three varieties of mung bean, that is a pulse, IPM 2-3, IPM 2-14, and a local Czech variety. These varieties were taken to check whether the tillage system followed in rice had any effect on the different varieties of mung bean. And all the mung bean varieties, for all the mung bean varieties, the package of practices for management were kept similar. We found that in the soil, there was improvement in a lot of soil properties after following this cropping system for 5 years. In the zero tillage systems or the conservation conservation agricultural rice-based systems, rice pulse system, we found that the available nitrogen, phosphorus, and potassium content was higher. Similarly, organic carbon also increased in the conservation agriculture rice-based system. Apart from nutrient availability, we also saw the physical structure. And we also found that conservation agriculture systems improved the macro, meso, and micro aggregate formation significantly over the conventional system. This signifies that the soil is well conditioned after following the conservation agriculture. Now, carbon sequestration is an important part that agriculture is considering today, and all the management practices revolve around how much carbon we can conserve. And our management practices under zero tillage system, both in the transplanted rice as well as in the direct seeded rice, we found that the total organic carbon was significantly higher. Total organic carbon was 16.9 and 17.04% higher in zero tillage DSR compared to conventional DSR and zero tillage transplanted rice. Over the zero tillage DSR, overall, the zero tillage DSR has a much higher amount of permanganate oxidizable carbon. We also calculated the energy balance. And the overall energy balance, we found there was 32.2% input energy that was required in the conventional tillage system or the conventional rice growing was saved. The diesel and machinery can be reduced, machinery use can be reduced drastically in the zero tillage systems or on the conservation agriculture. So to conclude, it can be said that conservation agricultural based systems increase the nutrient availability, soil organic carbon stock, water stable aggregates, and microbial activity. And based on these findings, it may be suggested that comparable yields along with the added advantage, particularly in zero tillage transplanted rice in terms of soil and environmental health necessitates the scaling up of the conservation agriculture rice pulse system. With this, I conclude my talk. Thank you.
Thank you very much, ma'am. A round of applause for her. Moving on. Now, our next speaker is Dr. Raghunath Satpathy, who is going to join us virtually from Gangadhar Meher University, Sambalpur, India. Let's welcome him. And he is talking on exploration of Agrimone mexicana plant through RNA sequencing data assembly, focusing on annihilation of genes associated with secondary metabolite production. Let's welcome, sir.
Good evening. Is it audible?
Yes, sir, you're very much audible. Please share your slides. Dr. Raghunath Satpathy is with us. Ladies and gentlemen, please welcome him with a round of applause. Sir, please share your slides and continue. Sir, am I audible to you?
Yeah, it's audible. I'm just trying to.
Yes, sir. Yes.
Some network issue is there. I'm just.
Not an issue, sir. Take your time.
Is it visible?
No, sir, not yet. Sir, first of all, open the file, then share.
Yeah, it's already open. It's already open here. It is not coming. 1 minute. PDF. Now,
Sir, please open
now
the file first, then you can share.
it's already open.
Now, you can share that. There's a sharing option.
Yeah. The document things are there, but it is not coming. Sharing the screen. Now it is coming, madam.
No, no, sir, not yet. Dr. Shushmita Munda is with us. It's a pen drive. So
It's actually that particular option. 1 minute, just a minute. I'm just joining with a different thing so that can share.
sir, if you will permit us, we will
Yeah.
proceed with another presentation right now. Till that time, you can check it out and find it.
Yeah.
Should
can
you do that?
Yes, sir. It's after one.
Is it convenient for you, sir?
Yes, sir. After one, one talk.
Yes, please take your time and check it out so that you can share the slide. Thank you very much, sir. Now moving on, please welcome Dr. Shrikant Lenka from ICAR-NRRI Cuttack. Let's welcome sir with a round of applause. He is talking on evaluating in vivo efficiency of new and commercially available fungicides against seed blight disease of rice. We welcome you, sir.
ଫୋଲ୍ଡର ଅଛି 21 ସେଟା ଖୋଲିଲେ ନା କୋଉଟା 21 ଏଇଟା ହଁ ପ୍ରଫେସନାଲ ସୋସାଇଟି ଯେଟା ପ୍ରଫେସନାଲ ସୋସାଇଟି ଦେନ୍ ଏଗ୍ରି ଭିଜନ ପ୍ରେଜେଣ୍ଟେସନ ସ୍ଲାଇଡ୍ ବନ୍ଦ ମ୍ୟାଡ଼ାମ ଏକ ମିନିଟ୍ slide. Slide. Madam. Sorry for late. Thank you very much. Good evening, respected organizers and beloved delegates, international and national, having reputations. So myself, Dr. Srikant Lenka. I am working as a principal scientist, plant pathology in crop protection division, National Rice Research Institute of ICAR. I am very happy to present the oral presentation on occasion of a 6th International Conference on Agriculture for Sustainable Development, and my research work on evaluating the in vivo efficacy of new fungicides, new and commercial available fungicides against the devastating rice disease, that is seed blight disease in rice. Madam. Next. Oh, yeah. Huh. Oh, yeah, then the next. Will.
Just keep it here.
Huh. Yeah. ये भी नहीं अच्छा हाँ as you know that after introduction of hybrid varieties of rice and also hybrid varieties by the rice breeders in association with the plant pathologist and entomologist, so many varieties of rice have been developed in our country and also world. And due to introduction of those hybrid varieties, the rice diseases which are prominent in rice crop and which were minor, then due to climate change and introduction of hybrid varieties, they have been now come to the status of major diseases. And our scientist, Paidwick 1950 and Chakrabarti, one plant pathologist working in at that time Central Rice Research Institute, Cuttack, had reported 30 diseases of rice. Then coming to the devastating diseases due to favorable climate and also favorable situation, then these diseases were now coming to above 60 numbers. So out of a major diseases of rice, the most devastating disease of rice, that is the seed blight disease of rice. It is caused by the fungus Rhizoctonia solani cone, and its a perfect stage is Thanatoporus cucumeris. It is a basidiomycetes fungus. And you astonishingly know that this disease has no resistant genotype. Though researchers are working again and again, and also in the morning time, I also raised the question while one of our experts was deliberating on genome editing. I was also putting questions, and we are taking a lot of efforts in our institute and also other institutes of ICAR and also other rice breeders to develop the resistant variety. And we hope that we are also having done the QTL mapping of taking the most susceptible variety Sorna Sub-1, and we have been succeeded and think that and we have trying to develop resistant varieties. And now the option is with us that we have to depend on the host plant resistance or resistant varieties, and another option is fungicides. And till we obtain the resistant variety, we have to depend on the fungicides. And again, by repeated use of the older traditional fungicides, this disease, seed blight disease, has developed a resistance to those diseases. So the plant pathologists have tried and taking in the research trials different new fungicides and also comparing with the available commercial fungicides to know that which new fungicides or combination of fungicides will be significantly reducing or inhibiting this devastating disease. And according to reports by different plant pathologists like Rajan and Rai, the yield loss due to this disease in India has been reported up to 54.3%. And here, various cultural practices combined with the use of fungicides are the most common option for managing this disease. In my study, I am presenting the effect of a new and commercially available fungicides for controlling the sweet blight disease. Madam? Madam? Madam, hello? Babu? Babu, hello, madam? Next, suno. डॉक्टर साहब वो जो छोटा लिया था ना वो उसको दे दिया ये मैडम थोड़ा डॉक्टर साहब दीजिए हाँ यही होगा मैडम उसको दे रहे ठीक है चलो सॉरी सर 1 minute प्लीज वेट मैडम एक्चुअली ये सेंसर वो पेन ड्राइव नहीं है वो सेंसर है इसका ये सेंसर है इसका ये पेन ड्राइव उसका नहीं हाँ दीजिए ये करो अभी आया ना स्लाइड मोड हाँ स्टार्ट है देन सेकंड अच्छा स्लाइड मोड था वो अच्छा देन लास्ट नहीं देन सेकंड में स्लाइड पे नेक्स्ट हां नेक्स्ट अच्छा ठीक है हाँ नेक्स्ट ठीक है then some combination of fungicides were also tried with the hope that a synergistic action and it was found that mancozeb mixed with thiabencarb and eprodion mixed with the carbendazim were found better than several single formulations in disease control and yield protection. While mancozeb mixed with the carbendazim tank mix was not effective, then here in my experimentation, I had done the experiments for 2 consecutive seasons during Kharif and Kharif season 2021 and also 2022. Here in this experiment, I have taken 7 new and commercially available fungicides. One is difenoconazole 25 EC at the dosage of 1 ml per liter of water. Sorry, 0.5 ml per liter of water. Then second treatment, isoprothyllene 40 EC at the dosage of 1.5 ml per liter of water. Third one is casugamycin 3% SL at the dosage 2 ml per liter of water. Then coming to treatment 4, it was ketazine 48 EC at the dosage 1 ml per liter of water. Then treatment 5, propineb 70 WP at the dosage 3 gram per liter of water. Then treatment 6 was a tebuconazole 25.9 EC at the dosage 1.5 ml per liter of water. Then treatment 7 thioplorazamide 24 AC at the dosage 0.8 gram per liter of water. And treatment 8 was control. That means without application of any fungicide and only distilled water or clean water was sprayed in the rice plants. Here I had taken the formula for calculating sheath blight disease severity and sheath blight disease incidence. Here are two formula for disease severity and also disease incidence. And I had also followed the disease reaction 0 to 9 ACS scale as adopted by the Philippines. Here, I had taken the most susceptible susceptibility variety, that is the Tapassini, and here the rice plants were inoculated with the most virulent strain. Here I have taken, and by placing the sclerotia with the mycelia inside the leaf sheath of the every treated plots, and after inoculation of this susceptibility pathogen, watering was regularly done, and after 10 to 15 days, the susceptibility symptoms occurred. And after 10 to 15 days of symptom appearance, all the 7 fungicides, that means new and commercially available fungicides, we are sprayed in the inoculated rice plants 2 times in the gap of 10 to 15 days. And after the disease occurrence, then the disease recordings were taken for 2 times at the interval of 15 days. And then susceptibility disease severity and susceptibility incidence, that is PDI, was calculated. And after harvesting of the the grains from every treated subplots, the grain yield was taken and compared with the control plots. Then coming to my result out of the experiments for 2 consecutive seasons, here I found the best treatment was isoprothyllene 40% EC at the dosage 1.5 ml per liter of water, which showed 18.3% disease severity, 22.7% disease incidence, that is PDI, and production of a grain yield of 5.3 tons per hectare. Then another fungicide, second one, the significant one was difenoconazole 25 EC at the dosage 0.5 ml per liter of water, which showed 18.4% disease severity, 23.7% disease incidence, and produce the grain yield of 5.26 ton per hectare. But you can see that in unrated control, that means where no chemicals or no new fungicides have been sprayed, there the disease severity was found to be more, that is 67.9% disease severity, 70.4% susceptibility disease incidence, and here is very least grain yield, that is 3.4 ton per hectare. Here I have presented 2 years data, 2 season data, 2021 and 22, and average the figures, and you can see the isoprothyllene 40% EC reduced the susceptibility disease severity by 73% and reduced to susceptibility disease incidence by 67.7%. But in difenoconazole 25%, that is the second most important fungicide, new fungicides, where it is reduced to susceptibility disease severity by 72.9% and reduced the susceptibility disease incidence by 66.3%. In the treatment, best treatment in isoprothyllene 40% EC, the grain yield was maximum, that is 5.3 tons per hectare or 53 quintals per hectare. And in the second best treatment, it was 5.26 ton per hectare or 52.6 quintals per hectare. Third best one was tebuconazole 25.9% EC, where 4.74 tons per hectare was the grain yield. Then coming to my conclusion of 2 years experiment, I advise here the delegates and also the farming community and also development persons in state government or other agriculture department officials. I advise that the fungicides, these have been tried all over India supplied by the All India Coordinated Plant Pathology Trials. And all the plant pathologists in different state corners have tried these experiments, and here I advise, and also it will be presented in the, already we have presented the forum, And these two fungicides, that is isoprothyl 40% EC and difenoconazole 25 EC at the dose of 1.5 milliliter and 0.5 milliliter. It should be advisory for the farmers, and also we also researchers and developers should apply against this devastating disease to reduce the fertility incidence and disease severity, and also we can get the satisfied grain yield out of for that. Till we get the resistant fertility variety, we have to depend on the fungicides. And I in this forum, my conclusion is that these two best fungicides can be spread. Thank you very much. Thank you very much. Namaste. Thank you.
Thank you very much, sir. Indeed, an excellent presentation. A round of applause for him. Moving on, now let's connect to Dr. Raghunath Satpathy from Gangadhar Meher University, Sambalpur, Odisha.
Okay, madam, I am just sharing my screen.
Yes, sir, please.
It's okay?
Yes.
Okay. So, good evening. So today I am going to talk about the exploration of Argemone mexicana plant through RNA-seq data assembly, focusing on the annotation of genes associated with secondary metabolite production. It's a computational work, and it contains many of the computational pipelines to analyze the next generation sequencing data about this Argemone mexicana plant. So I'm just skipping this particular slides. I'm just going to my objective, so that, okay. So the basic objective is the in silico study for the assembly of transcriptome data of Argemone mexicana plant and annotation of for the secondary metabolite gene. The Argemone mexicana plant is prevalent and it's an annual wheat, and it is basically widely used for various therapeutic, the phytochemicals, whatever is produced by this Argemone mexicana, are widely used for the therapeutic approaches, starting from anti-cancerous, anti-asthma, pain, and even if antibacterial, and also anti-parasitic, anti-malarial, and so on. There is a important thing to address. So what are the genes and the interaction about these particular genes that is available in this particular plant that are responsible to produce these secondary metabolites. So what we did, this is the system configuration we have used, and the raw sequence data we have taken from this NCBI SRA database, and this is the ID that is for this Argemone mexicana plant, and we have taken this, and this raw data we use the particular computational tool that is called FastQC to check the quality, and there are so many parameters we computed for these particular sequences. Then we use Trimomatic version 0.39 for the trimming purpose, and finally the COT adapt application tool is also used. All we used for this cleaning purpose, and finally the quality of the process sequence we again checked with the FastQC tool. Then after satisfactory things, then we went for this de novo sequence assembly by the Trinity program 2.15.0. It's actually contains so many sub programs that takes this raw processed data to make a complete assembly. And once we got the assembly, then we use this last text program to find out, to annotate these particular things. That means what are the genes are present. We downloaded all this UniProt review database for the as on the date particular 15/1/2023, and a local blast was basically done by using the blast to go software tool. And after this, whatever the blasted or whatever the blasted contigs we found, then we used another tool for annotation, functional annotation, that is called WEGO 2.0. So distribution of the genes and functions in this particular things. So this is the result. This is the quality control. Before filtering, this is the data, and after filtering, the quality has been increased. So this is the contig measurement about how many number of contigs we obtained, and what is the median contig length, what is the average contig length, and total assembled basis. These are after the assembly. Then this is the blast test result, where we obtain this total number of blasted sequences 21,970, and this is the interpro annotation, and this was the sequence for and 7,621 was no blast hit we obtained. Then this is the major plant species we got from this. Major hit was from Arabidopsis thaliana. Then next is Oryza sativa and so many other plants. These are some major category of plants where we got homology from this particular plant. Then this is the annotation of this particular thing. So total number of annotated gene, what we mentioned in this previous slide, is 19,780. So these were annotated things. So we used this WEGO tool to find out what is the gene ontology terms or biological function, cellular function, and this is the distribution details. And out of this, we found 44 contigs. These are characterized for the secondary metabolite production, and these are measured for the phytogelatin and phenyl propanoid metabolism. These are the contigs that we obtained for our result. And in the conclusion, we can say the computer-based methods are very preferable method to analyze this complex and large genomic data. And finally, we obtained these genes involved in biosynthetic pathway that can be used for this production of these metabolites, important metabolites in this particular plant. And we got 44 number of secondary metabolite genes involved in the secondary metabolic pathway, and this work can be extrapolated to study metabolic pathway network, the protein-protein interaction, as well as further we can also see for the metabolic engineering to enhance the production of these specific secondary metabolites. So this is some of the reference, and thank you.
Thank you very much, Dr. Satpathy, for sharing the highlights of your work. A round of applause for him. Thank you very much for your valuable time, sir. Now,
thank you.
Now we come to the end of the scientific session. After a small setup, we would go for the felicitations. I would request each one of you to kindly stay back with us. Thank you.
Thank you. Thank you very much.
Check. Once again, very good evening, everyone. As you all know, for the second day, we are done with the excellent presentations by eminent researchers. Let's give them a round of applause. As you all know, each day on this platform, we are felicitating few eminent personalities. And today, it is our utmost pleasure to felicitate another very renowned person to give away the award. May I welcome on the stage Professor John Adinson. Let's welcome sir on the stage. I request sir to kindly come up on the stage to do the honor. And I would also request Engineer Dibyanshu Prasad Das to kindly join us for the felicitation. With utmost honor and pride, we are giving this award for the exceptional contribution in the sector of fishery and aquaculture. With a huge round of applause, let's welcome on the stage Shri Akshay Kumar Sahu, esteemed CEO of Kailas Fisheries and Aquatics. We are awarding him with the award of Market B. Let's give him a huge round of applause. Can I see some hands up in there to congratulate Shri Akshay Kumar Sahu, everyone? Let's have a big round of applause. We congratulate you, sir. Thank you very much for your exceptional contribution in the field of fishery and aquaculture, and also for the market positioning. You are getting this award of Market B. Give it up for Shri Akshay Kumar Sahu, esteemed CEO, Kailas Fisheries and Aquatics, ladies and gentlemen. One of the leading face in the field of fishery and aquaculture. Once again, let's congratulate him with a round of applause. बहुत-बहुत अभिनंदन सर आपको. Today is the second day, and tomorrow we are going to witness the closing ceremony, and also tomorrow we are having lot more awardees, and all the awardees are going to join us tomorrow. I would request each one of you to please join us tomorrow as well for the scientific sessions, for the closing ceremony and the cultural session tomorrow. Thank you very much, Professor John Adinson, for doing the honor. Thank you. Thank you very much, sir. Please take your seat. Once again, I would like to thank each one of you for your kind presence and patience hearing. I would request each one of you to join us for the dinner from 7:30 onwards. Thank you very much. I would request each one of you to please proceed and join for the dinner at 7:30. And it's a request to all of you to please join us tomorrow as well. Have a good time. Thank you.
Sí.